the support from lecturer and alumni of Universitas Ahmad Dahlan. Ladies and gentlemen, it is a wonderful and precious chance for me to be your master of ceremony in this beautiful morning on Saturday, 18 July 2021, in our big event, National Pharmacy Conference Universitas Thailand 2021, Major Challenges and Trends in Pharmaceutical Science 2021, from natural product, genomic perspective, and applied technology to pharmaceutical product. Ladies and gentlemen, before we begin this event, let us pray together by reading the Al-Fatihah letter. May Allah Subhanahu wa Ta'ala make this event beneficial for all of us, and this event can run smoothly. Al-Fatihah. All right, ladies and gentlemen, I would like to inform you that to get SKP from IAI at today's event, participants are required to fill in attendance and attend the event from beginning to finish and do a post test at the end of the event. And we will close the post test at 6 p.m. West Indonesian time or GMT +7. On this special morning, we have several agendas. So allow me to read several sequences of our agenda. The first is the opening, which we previously had read together. The second is singing the national anthem Indonesia Raya. The third is recitation of Holy Quran. The fourth is remarks. The fifth is documentation. The sixth is the core event, the webinar. The seventh is poster and oral presentation. The eighth is announcement of the best oral presenter and the best poster presenter. And the last is the closing and post test. Ladies and gentlemen, step on to the second agenda is singing the national anthem Indonesia Raya. Ladies and gentlemen, if you can, please stand up. Thank you. Indonesia Raya. Next, on to the following agenda is reciting the Holy Quran, who will be recited by Ulfi Yandi Rizki, student from 4th semester Faculty of Pharmacy, Universitas Ahmad Dahlan. To Ulfi Yandi Rizki, the spotlight is yours.
Assalamualaikum warahmatullahi wabarakatuh. أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم سبحان الذي أسرى بعبده ليلا من المسجد الحرام إلى المسجد الأقصى الذي باركنا حوله لنريه من آياتنا إنه هو السميع البصير وآتينا موسى الكتاب وجعلناه هدى لبني إسرائيل ألا تتخذوا من دوني وكيلا ذرية من حملنا مع نوح إنه كان عبدا شكورا وقضينا إلى بني إسرائيل في الكتاب لتفسدن في الأرض مرتين ولتعلن علوا كبيرا فإذا جاء وعد أولاهما بعثنا عليكم عبادا لنا أولي بأس شديد فجاسوا خلال الديار وكان وعدا مفعولا ثم رددنا لكم الكرة عليهم وأمددناكم بأموال وبنين وجعلناكم أكثر نفيرا صدق الله العظيم. السلام عليكم ورحمة الله وبركاته.
Waalaikumsalam warahmatullahi wabarakatuh. Thanks to Ulfi Yandiriski, who has recited the Holy Quran for us. May Allah always give us guidance and help. Amin. The next agenda is remark. Ladies and gentlemen, I immediately invite the Dean of Faculty of Pharmacy, Universitas Ahmad Dahlan, Professor Dr. Diah Aryani Perwitasari, MSc Ph.D. Apotheker, to give her remarks. To Professor Dr. Diah Aryani Perwitasari, the time is yours.
Okay, thank you very much, Mas Ricky, as the Master of Ceremony. Good morning. Assalamualaikum warahmatullahi wabarakatuh. Our distinguished guests, the speaker of this seminar. The first is Dr. Phennapa Charoenwiwattanakij from the Faculty of Pharmacy, Siam, University, Thailand. And then the second is the Dr. Henry Sung-Ching Wong from the College of Pharmacy, Taipei Medical University, Taiwan. And then the next speaker
Recording
is
this meeting is being recorded.
Professor is Irwandi Jaswir from International Institute for Halal Research and Training, International Islamic University, Malaysia. So thank you for accepting our invitation to be the speakers in the seminar, and also for all of the participants, lecturers, and students, both from the Universitas Ahmad Dahlan or maybe from other universities. I also say thank you for your participation in this seminar. Firstly, thank you to Allah Subhanahu wa Ta'ala, the Almighty, for blessing us and giving us the opportunity for joining this event. Of course, I hope in a healthy condition. Prayers and peace upon our Prophet Muhammad Sallallahu Alaihi Wasallam, his family and companions. And then secondly, I'm proudly present to you all that the participant registered for this event reached around 200 participants, meaning that the scientific meeting until now is still needed, especially in current situation to get the new knowledge and experience across the country, which we know that we always conducted the seminar or event in virtually way. Also, I announced to you that this seminar is under the collaboration between the Pharmacy Higher Education Association for Muhammadiyah and Asia, and many participants also as the member of this association. We have some interesting topic which related to the challenges and trends in the pharmaceutical sciences, such as natural product chemistry, genomic research in pharmaceutical interest, and halal pharmaceutical science. And of course, you will have an opportunity for learning from the expert of this area, like I mentioned before, from the Dr. Phennapa, Dr. Henry, and also Prof. Irwandi from IIUM Malaysia. Unfortunately, the speaker from Universitas Ahmad Dahlan cannot join with you today because of health problem. But you still can learn from many things from the oral and poster presentation, which will be presented by the students and also by lectures. So please enjoy the seminar, and if possible, please make a networking, because even though the seminar conducted virtually, networking still can be developed without the boundary. Finally, formally, I open this national seminar and have a great seminar. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Professor Dia, we still have time to for you to open the event today. So the time is yours to open this today event.
Okay, I think I already mentioned before in my opening ceremony that formally I also opened this seminar. So what can I do with the opening? Should I make some knock or just say Bismillahirrahmanirrahim? So formally I opened this national seminar with saying Bismillahirrahmanirrahim.
Bismillahirrahmanirrahim. Thanks to the Dean of Faculty of Pharmacy, Universitas Ahmad Dahlan, Professor Dr. Diah Aryani Perwitasari, for the remarks and for the opening of the webinar today. Ladies and gentlemen, the next agenda is documentation. So speakers, participants, invited guests, please activate your camera because we will take a group photo together. Again, speakers, participants, invited guests, please activate the camera because we will take a group photo together. For the committee, please help us with a screenshot of today's documentation.
Yeah, okay.
There's 6 page. I will say it have done in each page. Okay. Page 1.
It's done. Okay.
Okay. Then page 2. Then page 3.
Okay.
Then page 4. Then page 5. Then and the last page 6. Then thank you for everyone.
Thank you, Miss Prita. Thank you. So let's skip on to the next agenda, which is the core event, that is the webinar. Before that, let me introduce our moderator in this session. This session will be moderated by Apotheker Verda Farida, Magister of Biotech. She finished from Faculty of Pharmacy in UGM in 2005. And in 2009, she gets her professional degree in Faculty of Pharmacy, also in UGM. In 2010, she finished biomedical engineering degree in UGM. And in 2017, on Philipps-Universitat of Marburg. She also has experience as a staff of Department of Pharmaceutics and Biopharmaceutics in Philipps-Universitat Marburg in 2017. And right now, she is lecturer in Faculty of Pharmacy, Universitas Ahmad Dahlan. Her research is focused in nanolipids, nanocrystal, and nanoketosan. Some of her publication is like ultra small lipid nanocarrier loaded with Q10 and comparison of bulk and nanocrystallized dry powder of medicinal plants. And so on. So let's we say hello to Miss Verda. Morning, Miss Verda. Assalamualaikum.
Good morning, Mr. Ricky. Waalaikumsalam.
How are you today, Miss Verda?
I'm fine. And you? How are you?
I'm fine too, Alhamdulillah. It's sunny but cold in here in Batu Licin. How about in Yogyakarta, Miss Verda?
It's sunny and cold too. It's here.
All right, Miss Verda, to shorten the time, the spotlight is yours.
Okay, thank you, Mr. Ricky, for the introductions. It's really nice to meet you. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. My name is Verda Farida, but you can call me Verda. Now we are on the main course of the conference, and I will take over to get you to get through this session. The Honorable Professor Apotheker Diah Perwitasari, Ph.D. as the Dean of the Faculty of Pharmacy, Universitas Ahmad Dahlan, Dr. Apotheker Hari Susanti as the Vice Dean of the Faculty of Pharmacy, Universitas Ahmad Dahlan, all the Honorable Deans and Heads of Pharmacy Program study from APTFM, Pharmacy Higher Education for Muhammadiyah and Asia, representatives from IAI, Ikatan Apoteker Indonesia, all the preceptors from Apothecary Program of the Faculty of Pharmacy, Universitas Ahmad Dahlan, our distinguished speakers, Professor Dr. Irwandi Jaswir, Dr. Phennapa Charoenwiwattanakij, and Dr. Henry Sung-Ching Wong, and our honorable guests. Welcome to our national conference in Major Challenge and Trends in Pharmaceutical Science 221, from natural product, genomic perspective, and applied technology to pharmaceutical products. It's a pleasure to have you here. Okay, there will be 2 conference sessions in which our outstanding invited speakers will share their knowledge. In the first conference sessions, there will be 3 sub-sessions. The first sub-session will be a presentation from Dr. Phennapa Charoenwiwattanakij from Siam University, Thailand. And the second sub-session will be from Dr. Henry Sung-Ching Wong from Taipei Medical University, Taiwan. Each presenter will deliver their topics for 40 minutes. And for those who have questions, you can write it down in your notes, and we will give you the opportunity to ask the questions in the third sub-session, which is question and answer session, for about 20 to 30 minutes. The second session will be in just 2 sessions. As Professor Diah mentioned before, Dr. Arif cannot join the event today. The presentation will be from Professor Dr. Irwandi Jaswir for about 40 minutes and followed by the question and answer session for about 15 minutes. You are highly recommended to ask in English, but of course you can ask, you can use Bahasa Indonesia and our team will help you to translate it. Okay, now let's move to the first subsession, which is a presentation from Dr. Phennapa. Before we will jump to listen to her presentation, I will read the CV from Dr. Phennapa Charoenwiwattanakij. Give me time. I would like to share my screen. Wait. But I cannot do this.
I will help you to sustain what Ms. Verda.
Huh? Sorry?
You have difficult to sustain? Okay.
Yes, I cannot share my screen.
Okay. We will help you. This is my share screen.
Okay. Okay. Okay, Dr. Phennapa Charoenwiwattanakij is a lecturer and researcher in the Faculty of Pharmacy, Siam University, Thailand. She got her doctoral philosophy in Pharmaceutical Chemistry and Phytochemistry, Halal University, Thailand. Can you move down? Her research interests are in natural product chemistry and Thai traditional medicine. She had an experience as a special research student at the Laboratory of Biofunctional Molecular Chemistry, Graduate School and Pharmaceutical Science, Osaka University, Japan, and move to the publication, please. Yeah, and these are 2 of her publications about protein FTS set. And today, it's enough, thank you. And today her topic in this conference is current issue on natural product chemistry. This would be really related to us since we in Indonesia also has an abundant resource of natural product. It will be interesting. Without further ado, Dr. Phennapa.
Good morning, can you hear me?
Yeah. Yes, we can hear you. Your time is 40 minutes. And I would like to apologize in advance if I had to interrupt you if the time is almost over.
Thank you, please do.
Yeah, the time is yours.
Thank you. Good morning, everyone, and good morning, Professor Dr. Dear. And
Morning, Dr. Phennapa.
We never met for a very long time.
Yeah.
It's very an honor to be here. And also, thank you very much for Dr. Mufida for contacting me.
Yeah. You're welcome, Doctor.
So today, the topic that I responsible for, let me share my screen. Okay. Is the slides on the screen? Is my slideshow on the screen already?
Yes, we can see your slides.
Okay. Thank you. Okay, so the topic that I'm responsible for today is the current issues on natural product chemistry. And I believe that there's nothing more current than the COVID-19 right now. So I'm going to have a talk about the natural products that will involve in this, and also it's from Thailand. But before we go into that, we're going to go through the topic of drug discovery from natural products, which is like the basis for everything that we're going to talk today. So talking about drug discovery, we're going to starting with identifying the drug target and finding the compound which you can find from the traditional medicine or any other source, or maybe the new drug or from the synthetic source. And then you just like go to taste whether it's in vitro or in vivo or in silico studies. But I would like to say not only in the first part that the chemistry involved in the natural product is involved all the way into the clinical trial as well, because you're going to, if you know the chemistry of the natural product that you're interested in, you're going to know more that these chemicals what it's going to do in the patient's body. So the first one, Okay. So talking about the chemical that involved in natural products, we're going to think about active substances and toxic substances. And how can you know that this is active or toxic substances? For sure, you need to test it. But what I would like to mention in this slide is different in the plant species, different plant sources, different in parts, different in doses, and different administration method. And also the time for collecting or harvesting the herbal product also involved in the differences in active substance or toxic substances. So when you want to do the research on the active or toxic substances in the plant that you got interested in, this is very important that you need to think about before you go to. Also, sometimes we think about the only compound, which is the active ingredient in that herbal product, but you need to keep in mind that it's not only the active ingredient available there. There's a lot of compounds available in the herbs. And even you extract it into the crude extract, there are also a lot of compounds there. So there are more than one for sure. The thing is, you need to standardize before getting it into the pharmaceutical product or launched out for the patient. So making sure that the active ingredients is at the level that can treat the patients, and the toxic substance keep lower than the toxic dose. Like I said, starting from the plant itself, there's a lot of compounds, and maybe the part of the ingredient or active compounds that you got interested in, maybe only a little. But then when you collect it, it will be more concentrated for the active ingredient or toxic substance. And when you go on, like extraction or purifying this, it will be more concentrated. So every step leads to different dosage, due to different compounds available in the extract itself. And also you need to keep in mind that if you use extract or you use the isolated compound, like purified compound, the dose also different. So this is what you need to think about it as well when you want to deal with the natural product. So that's just the introduction I just would like to let you think about when we work on this, I mean the natural product itself, you need to think about the chemical available there. But not only that, the chemicals come from the natural source, which have a variety of them. So you need to think about species, you need to think about the method to get it. And also you need to control it by the standardization. Next, we come to the main topic that I would like to talk today, which is the natural product of COVID-19. For sure, it is the problem globally for like 1 or 2 years already, and the scientists can try to find a way to treat the COVID-19 patients. And in Thailand, I need to say that right now we have the cases keep increasing. And it's going to be a problem for the hospital and the public health system if we cannot find a way to suppress it. But if the cases of the infected patients keep increasing, natural product is another way to help the patients. I can tell you later what is the result that we find in Thailand. So today we're going to have like 2 herbs that involved in the COVID-19, and one is very interesting because it's launched out in the Thai natural drug list that you can use for the COVID-19 patients. The first one, which is the first one, Endoglossus paniculata. I'm not quite sure that, ladies and gentlemen, have you ever heard about this herb? But it's very famous in Thailand. The plant here belongs to Acanthaceae, which available not only in Thailand. We believe that originally it's come from India and get spread, like we get it from India and also apart from China. And also this plant used a lot in China as well. There's a lot of name here, which is Endoglossus, and in Thai, we call it Pha Thalai Chon. Or make it easier, we can call it King of Bitter. Like when I study in bachelor degree, there's a course that we need to identify the plant, and there's also the test. So if you just look at the plant and you're not quite sure that this is Endoglossus or not, my teacher said you just test it because it's super bitter. And in Thai words, if it's bitter, it is the drug. So we think that it should be very good to deal with. This is the active ingredients available in the Endoglossus. The main one is the endoglopholine here, and also related substances which you can see all of them, not all of them, but most of them are diterpene, as you can see in the structure here. And a part of them also available with the flavonoids itself. So looking at this, you can think about at least the antioxidants from this plant, right? But we're going to talk about this later. But right now in this slide, what I want to show is remember endoglopholine. This is the active ingredient or active compound in Endoglossus paniculata. In Thailand, since we use this for a long time and also available in the natural drug list, firstly, it's available as the drug, herbal drug for common cold treatment or anti-diarrhea. And we use this for a long time, and there's a lot of product available in the market, and it's really varied. As you can see on the slide, that number under x-axis is the code for the product. This is a research done by the Faculty of Science of Mahidol University in Thailand. The endoglopholine and the derivative itself is very varied from product to product. So there's a question that how many of the endoglypholide should be there, or how many of the diterpene should be there in order to control the quality of the endoglyphis product. So like I said, is extract itself and the purified compounds are different, especially in doses. So the thing that we can control is also different, which is total lactone. Like I said, endoglypholide is the active compounds, and looking at the structure is lactone. So we're going to control for the total lactone for the extract itself. But if you purify it to endoglypholide, you're gonna compare it with the standardized endoglypholide, which you can use the technique like HPLC to check for the quantity of it. The part also very important. Usually in Thailand, we use this for the part upper ground. So as you can see, like the study to find endoglypholide and total lactones in same leaf and total herbage, there's like they have a lot of them there. There also the study that when you should harvest it, because like if you grow it up, there also endoglypholide available there, but at which state you should harvest it. We find that the endoglypholide available or the quantity is the maximum in the flower. But the size of the flower is really, really small. So it's like for the farmer itself, it's very hard to harvest it to get the dose of the endoglypholide. So the thing is, the recommendation was done by, okay, you just like let it bloom for 40% and harvest the whole plant upper ground. So you can get the endoglypholide as much as is possible. So there's a requirement. When we look at the product or when you want to deal with it, you should making sure that you work with the extract or the purified compound. If you work with the extract, you should check that the diterpene lactose or total lactones are over 6% weight by weight, or at least 6%. And if you work with endoglypholide, it's going to be more than or at least 1% weight by weight. So you need to make sure about that. This is not only the requirement from Thai natural drug list, but also the WHO monograph on selected medicinal plants as well. So for the pharmacological activities of the endoglyphis paniculata, there's a lot of them. In traditional one, we use it for relieve the fever. That's the main one. When we look at the traditional medicine, I would like to say that the way we look at the patients are different from the modern medicine, because we look at the patients at the whole body. And the type of the medicines as well. We have like subtypes like this drug is the warm or hot herb. This one is the cold herb. Endoglyphis paniculata fall into the group of cold herb. It does not mean that you touch it and you feel cold. This means that you take into your body, make your body cooler. So is health in this when you explain it in traditional way, but in modern medicine also we prove that it's help relieve the fever for sure, and it have the activity for as an anti-inflammation agent. Also, we have the test that it can treat with the virus infection, so it can be an antiviral activities. Also, this is one of the very important thing, is improve the immune, but not only the humoral one, but cell-mediated or innate immunity as well. So this is the idea that very important. On the picture here, you will see there's a lot of the activity that involve in the thing that I'm talking about, especially the antivirus activity of Endocarpus paniculata. Okay, this is the indication and those. Originally, we use this for the common cold and recommended for only the powder drug. Sorry, this is also healthy here. The powder drug, which recommendation is 1.5 gram, and you need to take it for 4 times per day. But that's a lot. And consider is even is in the capsule. When you take it a lot, you feel like the bitter flavor in your mouth, and it's very hard for the patient sometimes to take it. So later on, the standard dice extract also available. And the compound that we standardize is endoglycolide, which is the active compound, like I said. And you need to make sure that every time you take it, you get 20 milligrams per dose, and you take it 3 times per day for the common cold itself. The product available in Thailand is not like 20 milligrams. Sometimes you can see it's like 6 milligrams, 5 milligrams, 9 milligrams, or 10 milligrams. So making sure that it's 20 per dose. Next is for non-infectious diarrhea. We use it for a long time as well, but usually we use the powder drug. So it's going to be 500 milligrams to 2 grams 4 times per day for non-infectious one. And lately, the last test recommendation on COVID-19 just launched out in May this year is the COVID-19 for mild to moderate or intermediate condition patients. Recommendation is using the standard dye extract with endoglycolide, but the dose is increased. So as you can see, is 180 milligrams per day. So you need to separate it into 3 times. It's 180 milligrams per day. So separate into 3 times. So it's about 60 milligrams per dose. And that's a lot. But they also like the proof that it's okay. And it can help the patients, especially with the mild stage. Many cases that using this, we find out the condition of the patients like recover very, very fast, and also the protection for the disease to be progressed is less as well. So just to prevent the patient to go to the clinical stage. It's very good. So talking about this one, I already talked about the uses, but this time we're going to find the information that is proof. How can we prove it? This is the in silico. We're going to talk about the in silico test first, but before that, we going to identify the target for the medicine itself, which is the S protein, which we know that is the spike protein that used to for the virus to infect into the cells or for the viral entry to the cell. And S2 protein, which is the cell available, especially at your lung cells. So it's the S protein and S2 protein will bind each other right before it's getting to the lung cell itself. And we don't want that to happen. Another thing is the M protein, which is involved in viral RNA synthesis. So if we can block all of this target, it's going to be very useful to deal with this disease. And this is the list of the drug that test for the screening by in silico method or docking method. In this study, they use like S2 receptor and M protein. And looking at the docking score, for the people who deal with this, you're going to know that if they're less energy, it means it's good, because the ligand, which is the drug, and the target can bind tightly. For the screening, even androgalfalide is not the top 10 list, but Thai scientists are very interested in it because it's available in Thailand a lot, and we use this for a long time. So we go with this. This is another study for the for in silico. The previous slide is just androgalfalide, but this slide proves that other compounds available in endogalfis paniculata also have the good effect in the silico test, in the in silico or docking study. So we assume that all of these compounds might be active against the COVID-19 or SARS-CoV-2 virus as well. But this is just in silico. So it's just a study that's very good for the screening, because you can do it for a very, a lot of compounds together. But you need to prove in real life as well. You cannot use just this result and go to the conclusion that it's good. So this is also from my colleague who work with this as well. She's collect all the data and also to prove that there's a lot of data concerning the in silico screening, and find that the androgalfalide and its derivatives can bind to the target that I mentioned. Okay, so this is the slide that we're going to talk about the in vitro study, because like when we get the result from the in silico study, we should go with this to prove that it's really work. So the first one is they're going to check for the pre-treatment, for inactivation, and for post-treatment. What does this mean? For the pre-treatment, they mix the cell and the drug, which is endogalfis paniculata and androgalfalide separately together first, and then add the virus in it. So the result is, these results show that it can prevent the cells from the infection or not. Which result is not, because there's no activity there. Next is the inactivation. For the inactivation, they use the cells, and they use the, firstly, they do pre-treatment for the virus mixing with the endogalfis and androgalfalide, and then they add it into the cells, incubate it, to see that they can just like kill the virus or not. And they got the result, which is, you can see here, that endogalfis and androgalfalide can work very well on this. And for the post-treatment, mean that after the virus infection, they can treat, the extract and the androgalfalide can treat the patient or not. So they infect the cells with the virus first, and then add the sample into it. And you can see the IC50 here that also have the result as well. So from this slide, mean that they cannot prevent it, but they can treat it. So this is another study done by Mahidol University as well. They do the experiment on the human lung epithelial cells and the PREG assay together. In this slide, you see that after increasing the dose, the upper one is the extract and the lower one is the endoglypholine. After increasing the dose, the green color of the fluorescence decrease. So it means that it can suppress the virus itself, and also is the same trend, both in extract and endoglypholine. And then they confirm it by the PREG assay reduction. So if it's reduced, it's good. So as you can see here, they also work in the same trend. So from the study here, you can see that both the extract and endoglypholine can treat can inhibit the SARS-CoV-2 in dose-dependent manner. And also they have like selectivity index show that is the cytotoxic effect also very low. Next is the preliminary study in my COVID-19 patient. As I mentioned on the slide, it's only 6 cases, but it's in the preliminary study. And I need to mention this because I think that this study is at the end of the first wave of the infection in Thailand, and it's done without control group. So, but it's good for just like finding the data first. So they use the first one is to the standard diet, endoglypholine for 180 milligram per day for 5 days, and together with the standard treatment. So this is like the supportive treatment. And if everything is good, there's no cytotoxic effect, they go to increasing the dose, but there's nothing that different. So we're keeping with the 180. So the result is showing that they're going to check about the severity of disease, and check for the pro-inflammatory cytokine, biochemical profile, and safety. I would like to say about the inflammatory cytokine a little bit, because in the progression of the disease, sometimes patients will face cytokine storm, and it means that the inflammation that they keep increasing, and that make many patients go into coma and can die from that stage. As a result, they check for the viral load, and they cannot find it later, after the treatment. There's one out of, because the result is in error. The severity of disease, actually in cough, sore throat, phlegm, and headache, reduced as very fast after the treatment, comparing and anti-inflammatory marker. As you can see, some is decreased and some has increased. Because the cytokine storm occur because of the imbalance of the inflammation marker. So you need to check all of them to see that the balance is keeping on track or not. And for the safety, they check for ALT and AST, which is the marker for the hepatotoxic. And it's still okay. Even there's one case that is increased after the treatment for 5 days, but after you stopping the medicine, everything recovered to normal. So they go on to another preliminary with more cases later, because in the second wave, we have more cases in COVID-19, and they get the same trend of the result. Within the 5 days and recover, comparing between the patient who get pneumonia, which means that the virus can infect the lungs. As you can see here, the group that get the sample is just 3 cases, comparing to another is 77 cases. And there's no severe ADR. And there also this study for preliminary study waiting for the publication. So I are still also waiting for the full paper after this. And there also another study they're going to do the randomized control trials on this. But I think that they already go on with the test, and the test is launched out in the government level. So we have the recommendation that we can use this for COVID-19 right now. Okay. Not only that we know that we can use, we need to know that it have the contraindication or the precaution as well. We have a lot of cases reported in the case of allergic to Endocarpus paniculata. Not only the urticaria or something like that, but sometimes it's the allergy that's very severe, like swollen in your throat or your airway, something like that. So you need to be careful if you take it for the first time. And it's contraindication for the pregnancy and lactation. For sore throat, even it recommend for the common colds, like sore throat is one of them. But there are also some of them that you should be careful as well, like the bacterial infection or rheumatic heart disease, because this is already proof that endocarpus cannot treat them. And if you don't treat it correctly, it will lead to a very severe condition. So before taking it, you need to make sure that you don't get this condition. For precaution, even in the study, they proved that it might be okay for liver, but please keep in mind that you use it for a long time. Liver and kidney impairment is one of the conditions you should not use it. Also, I mentioned that it's cold medicine, so the numbness and the weakness in the peripheral area, like your finger, your hands, or your feet, can be found. So sometimes you feel like paralyzed as well. And also, if after the treatment of 3 days, if your condition does not better, you should like stop it, because it does not help, and it means that you got other condition. Also, we find that they have the drug interaction with the anticoagulant and antiplatelet. So if you use it for a long time, you should not use it. Also, anti-hypertensive drug is also the precaution. I mentioned about the derivative of the compounds in Endocarpus paniculata. It's not only the endographoline, but it also have other compounds like neo-endographoline and many of them. So also this small amount of compound, which is the derivative, does this effect as well, anti-hypertensive drug. So you should not use this together. And also the drugs that pass the metabolism, like cytochrome P450, like CYP1A2, CYP2C9, or CYP3A4. So making sure there's a lot of medicine available there and pass through CYP3A4. This is the adverse effect that you can find. I mentioned the numbness and weakness in peripheral area, and also GI disorder. Sometimes you find it as well. Even it's treated the diarrhea. But if I explain it in terms of the Thai traditional medicine, it's the cold medicine. So it makes your body lose balance. So this might be the case. Sometimes dizziness, palpitation, and urticaria can be found as well. So that's about Endocarpus paniculata, which we mainly use in Thailand right now. And it seems like I have about 8 to 10 minutes for this. I think I can make it in time. Next is Bosenbergia rotunda. Bosenbergia rotunda is a plant in family called Zingiberaceae. So mainly we use the part underground. And here, as you can see in the picture, in Thai, it looks like a finger. So we call it finger root. In Thai, we call it grachai. And I just found out in Indonesia, it's called temuk kunci. I'm not quite sure that I pronounce this correctly or not. If not, I'm very sorry. So it's a plant that are viable in Asia, especially in Southeast Asia. And we use it in our food as well. This is a compound that are viable in Bosenbergia rotunda. The compound that we very interested in right now is Panduratin A. There are also other as well, like pinostrobin and pinocrambin, but it's under the study right now. I heard that there's a big study on pinostrobin right now, but it's on the way. So I think the publication will be launched out soon. For this plant, they have a lot of pharmacological activities on your left-hand side, like antimicrobial, anti-parasitic, and a lot of them. Usually in Thailand, we use it to release the gas in your GI tract. Since they have a lot of this, we go on with the other activity that we got interested in, which is the antiviral activities. This is the study finding the Thai medicinal plants that have the activities on SARS-CoV-2. The study was done with over 100 Thai natural and Thai plants finding this. And as you can see here, there are also endocalphalide and endocalphalide extract here as well. And percent inhibition here for endocalphalide is very good. Also, you can find the percent inhibition on the extract as well. But for panduratin A and the extract from Boswellia rotunda, as you can see, percent inhibition is about 99, it's almost 100%. And that's very interesting because not only the purified compound, but the extract itself also show the same strength. The study was done on the pre-entry and post-infectious phase as well. This one using the fluorescent-based of the SARS-CoV-2 nucleoprotein and use the viral E6 cells. Together, they study with the PrEP reduction assay. So almost the same way with I mentioned in Endocarpus paniculata. For the pre-entry phase, they use the compound to get mixed together with the virus and add it on the cell. The post-infectious phase, they mix the virus first and then add the compound later and check it. This is the result. For Boswellia rotunda, the extract is on your left-hand side. On your right-hand side is the isolated compound. As you increase the dose, you can see that the fluorescent color is phase. This means that it can inhibit the virus both in extract and in the purified compound as well. And you can check this on the andrographis extract and endocalphalide. The result also show this that it can inhibit it as I mentioned in previously.
Dr. Phennapa?
Yes?
5 minutes left.
Okay. It's almost at the end. So thank you for the mention. This also the IC50 done by PrEP reduction assay to check it. And they say, as you can see that this give out the IC50 that a very good result. Okay, that's for the screen. This is the pre-entry that I mentioned. For the pre-entry, you can see the IC50 of the result that show the same way with the screening one. And the fourth infection phase, which is, like I said, that add the compound or extract later, also show that it can inhibit the virus after entry, after infect to the cells with a very good IC50. So this is the summary for Boswellia rotunda. For the pre-entry, the post-infection, it gives out the IC50, a very good one, and the cytotoxic CC50, cytotoxic concentration. And if you compare it to hydroxychloroquine, it might be a little bit like cytotoxic or not quite sure about that. But when you check for the safety by finding the ratio of cytotoxic to the inhibition concentration, inhibitory concentration, is very similar to the control, hydroxychloroquine. So these two herbs is the thing that I get at want you to know about the Thai herbal medicine. So the thing that I want you to get back home with the take-home messages is endographic spanuglata, which have we use this both for the powder and the extract itself, and also the endoglypholine and the derivatives. We use this for a long time, so the safety is quite okay, but you need to monitor as well, especially the new indication like COVID-19, where we use like a very high dose comparing to the normal, so 180 milligram per day, to help the patients, not only the treatment, but for the progression of the disease as well. And don't forget that dealing with this, you need to monitor. For both Bergia, with right now the penduratin A is the compound that's very interesting. But I found out both of the extract and the compound itself are both interesting because they give out a very same result on the inhibition of the SARS-CoV-2 infection. Okay, so this is what I want you to know, but not only this, don't forget about the quality control, the part of the use, the species. All this you need to check before you're doing not only the experiment, but also when you want to treat the patient or you get the product to use by yourself. Okay, so this is going to be the end of my talk. If you have any questions or any comment, please share with all of us. I really appreciate that. Thank you.
Okay, thank you very much, Dr. Phennapa. That was a nice explanation. Herbs for COVID, right? It's really important in this current pandemic as we are struggling to find a cure. I'm sure that there will be interesting questions from the audience, but we will keep it for the question and answer sessions. Thank you very much, Dr. Phennapa.
Thank you. I'm waiting for the Q&A session.
Okay. Okay, now we will go to the second subsession, a presentation from Dr. Henry Sung-Ching. I will read his CV before we start the presentation. I will try to share again my screen. Okay, this is the CV from Dr. Henry. Dr. Henry is a postdoctoral fellow in the College of Pharmacy, Taipei Medical University. He pursued his Ph.D. Maybe you can go down. Yes, here. He pursued his PhD degree at the School of Pharmacy in the same university where he is currently working at, Taipei Medical University. Go down again. His specialties, are in the field of pharmacy, epidemiology, genetics, genomics, pharmacogenics, immunogenomics, and cancer genomics. Precision medicine, translational medicine, also in bioinformatics, computational.
The recording has stopped.
Microarray analysis.
This meeting is being recorded.
Generation sequencing analysis. From the CV that I got, his publication list is long. Yeah, as you can see here, there's professional activities. He also got many awards. Yeah, and there are the list of his publications. Maybe we can focus on the news, the third news, the last one. Yeah. He already published 25 papers. Okay. Yeah. Okay. And his topic is applied genomics in pharmaceutical research. Without further ado, Dr. Henry, are you here?
Can you hear me?
Yes.
Now, I'm going to share my slide.
Okay. Dr. Henry, your time is 40 minutes, and I also would like to apologize if I had to interrupt you when the time is almost over.
Sure, thank you.
The time is yours.
So, good morning, everyone. My name is Henry Sung-Ching Wong. I'm a postdoctoral researcher from the Professor Wee Chao Chang lab, so-called the WCC lab in pharmaceutical research. Here is my brief CV. So, my research fields is majorly focused on pharmacogenomics, cancer genomics, and bioinformatics. And here is the select publication within 5 years. So now I will introduce the research field of the WCC lab. This is the Professor Wee Chao Chang. He is my supervisor. So in our lab, majorly we have 3 fields. The first one is about the calcium signaling genes. The second one is about the genomics and pharmacogenomics. And the third field is about the immune repertoire sequencing, so-called the immunopharmacogenomics. So regarding the calcium channel-related genes, this is one of the research field in our lab. So it is about studying the roles of calcium signaling-related genes, such as STIM1 and ORAI1 in human disease. So we study the biological or the genetic roles of these genes in autoimmune diseases and cancers. So previously, our lab has published a paper in Oncogene in 2015, and discovering that the STIM1 overexpression can promote the human colorectal cancer progression and the cancer cell mortality, and also the COX-2 expression. Next, our lab also focuses on the human genomics study. This study is to determine how we could apply human genetic information to the precision medicine. Through the genomic analysis, actually we expect to identify some useful biomarkers that could be translated into the clinical tools. And we hope the genetic information that identified from the population, especially Taiwanese population, can help us to improve the clinical diagnosis and to facilitate the development of some disease therapy. For the immune sequencing part, we aim to determine the disease-relevant T-cell receptors or B-cell receptors. T-cell receptor repertoire analysis actually occupied an important position in the T-cell related research. Nowadays, next generation sequencing, the NGS technology, has allowed us to simultaneously detect the TCR sequence with high throughput. So the potential of the TCR repertoire as a biomarker for immunotherapy efficacy is also widely studied, as TCR receptor repertoire represents different baseline within the people and shows dynamic change during the disease treatment. Now I will briefly introduce the concepts of pharmacogenomics. Pharmacogenomics pharmacogenomics is the definition of the pharmacogenomic is it is the study of variations of DNA and RNA characteristic as related to the drug responses. Pharmacogenomics can be defined as the technology that analyze how the genetic makeup of a people or an individual can affect his or her response to drugs. It is also the technologies that deal with the influence of genetic variation on drug response in patients by correlating the gene expression or single nucleotide polymorphism SNP with a drug's efficacy or toxicity. So by doing so, pharmacogenomics aim to develop some rational means to optimize drug therapy for the patient's genotype to ensure maximum efficacy with minimal adverse effect. Such approach can promise the advent of the personalized medicine or precision medicine in which drugs and drug combinations are optimized for each individual's unique genetic makeup. So another term related to the pharmacogenomics is pharmacogenetics. The pharmacogenetics is a subset of pharmacogenomics, and it is defined as the study of variations in DNA sequence as related to the drug response. So basically, we can think that pharmacogenomics combines the knowledge of pharmacology and of genomics. Here is the genetic factors of the drug response. So some of the factors that may determine the result of the drug response is which can be categorized into intrinsic and extrinsic. For the intrinsic factors, it's including like age, like the healthy status, and the genetics, which is the mainly focus in the talk today. And on the other hand, the extrinsic factors is the factors that may affect the drug pharmacokinetic or pharmacodynamic parameters, such as the diet, the use of the concomitant drugs, and also the patient adherence are all included in this category. So some of the examples of the genetic variants that may influence the drug response, including the single nucleotide polymorphisms, we call SNPs. SNPs is actually a DNA sequence variation occurring when a single nucleotide ATCG in the genome differs between members of a biological species or the pair chromosome in human. Another types of genetic variants, including the insertion and deletions, which we call indels together. Take the word IN and the DEL, so-called the indels. For example, here, the original variant in the population is like A, and then maybe an insertion may occur and become AACC. So this is an insertion of the nucleotide or the genetic materials. Another one is copy number variation. It is the longer sequence patch of copy of the genetic material. So there has many pharmacogenomics related genes has been identified, and they can be categorized into these four categories based on their function. That is how they affect the drug response. The first one is the genes that are relevant to the drug's pharmacokinetics. That means that these genes affect the drug's response through the ADME process. The second one is the genes that encode the drug targets and their associated pathway. So these genes can affect the drug response through the pharmacodynamic process or mechanism. And the third one and the fourth is the genes that influence the disease itself, the disease susceptibility or the adverse effect of the drugs. So here is the figure showing summarize the above idea. So basically, for example, there is the variation of the gene in one location. So because due to the difference of the nucleotide it carries, so it may affect the enzyme activity, the drug enzyme activity, like the SIG family enzyme, or like the P-glycoprotein is all including in this category, or the efficiency of the protein transporter, or the metabolic rate of the drugs. So finally, turns out it may affect the retention time of the drug, or the blood concentration of the drug, and the efficacy of the drug. So here is the example. Actually, this is only a very small amount of the example. So if you want to have more example, actually you can find it in many papers or the review articles. So I just briefly give you some example so that you can know that people already identify many genes that can affect the drug responses. For example, through the genes that may affect the ADME process, so-called the pharmacokinetic process of the drug. Here is the example, like the SIG family gene is very famous one. Okay, so also including the SLC family. For the pharmacodynamic part, some famous example is about the cancer drug, like for the patient with the BRAF mutation, especially we know that the V600E mutation. If patient has this mutation, then they can use the drug called vemurafenib. Also for the KRAS mutation, cetuximab can be used. But if patient didn't have this kind of mutation, this is not really suggest. Okay, so this is one example of the pharmacogenomic that we can subscribe the drug, the suitable drug for the patient based on their genetic context. So another example regarding the disease susceptibility or the progression is about the 5 for the HIV disease and the HLA-DRB1, especially this one, HLA-DRB1 for the rheumatoid arthritis is all the very, very famous example in the pharmacogenomics field. Here is another carbamazepine. We know that carbamazepine is an anticonvulsant and a mood stabilizing drug. Also, the carbamazepine is the main cause of the severe Stevens-Johnson syndrome and some of the related diseases like toxic epidermal necrolysis, we call TEN, in Southeast Asian countries. So the carbamazepine-induced Stevens-Johnson syndrome is strongly associated with the HLA-D1502 allele. This is the study published by Taiwanese group, another Taiwanese group. This is about the result and the evidence. And this study is from the Taiwanese group from Academia Sinica in Taiwan. So in this study, they study the incidence of the Stevens-Johnson syndrome of 10 after the genetic screening. So basically, they want to see whether they prescribe the carbamazepine after the genetic screening can reduce the case of the Stevens-Johnson syndrome. So this is basically the result of this study. In this study, they collect 4,000, more than 4,000 patients. So based on the estimate historical incidence, actually there is a 0.23% of the patient who takes carbamazepine will induce the Stevens-Johnson syndrome. However, in the 4,000 cases they collected, and then they all perform the genetic screening, and then if the patient has the HLA-D1502, they will not prescribe the carbamazepine, and for the patient who didn't have this gene, this allele, they will prescribe carbamazepine. So finally, it turns out if based on this incidence that estimate from the population data, they expect in the 4,000 patients, they will have more than 10 patients will have the Stevens-Johnson syndrome. But after genetic screening, actually none of the patient, none of the case has either Stevens-Johnson syndrome or 10 in the study. So this differed significantly from the historical incidence. So this study actually showed the importance of genetic screening in the prevention of the severe ADRs. So although the pharmacogenomics is very powerful, however, the path from the gene discovery to the effective clinical applications is very complex and challenging. And hence the efficacy in healthcare, health delivery, and quantifiable improvement in human health are the major goals of the genomic research. So the discovery, the development, and the application of genome-based drug discovery require both an interdisciplinary science strategy and the pragmatic implementation strategy for successful translation. Our lab has thus focusing on the developing of these strategies and capabilities to enhance our ability to diagnose and predict patient outcomes across the continuum from health to disease. Nowadays, genome research, especially conducted through the genome-wide association study, the GWAS, have identified numerous disease susceptibility genes. Now I will briefly introduce what is a GWAS study, the genome-wide association study. Genome-wide association study, the GWAS, is a study that looking for the SNPs that associated with a specific phenotype or trait. By doing the GWAS, it can help us understand the mechanisms of a trait and provide therapeutic insights. Also, GWAS can further suggest the way of interventions or preventions of the trait. So, as the knowledge of the disease gene associations accumulates, it becomes increasingly important to translate this knowledge into the clinical practice. This challenge involves finding some effective drug targets and estimating their potential, often result in the failure of the clinical trials. So one way to identify the potential drug targets, the useful drug targets for the clinical implementation of the pharmacogenomics is direct association study. The direct association study means that we directly collect a sample, including the case and the controls, with using the drug response as the phenotype, like whether the drug is effective or not, or whether the patient has the side effect or not. So this is called a direct pharmacogenomic association study. However, this kind of study is very difficult to conduct and due to the limitation of the small sample size. So there is another research field, so-called the genomics-driven drug discovery is proposed. It including some methodologies, which is the drug reposing, Mendelian randomization, and multi-omics integration. So I will introduce them one by one. So the genomics-driven drug discovery is actually one kind of the drug discovery approach that is different from the traditional drug discovery approach and the traditional genomic drug discovery approach. For the traditional and the traditional genomic drug discovery approach, actually they are focusing on the disease, and then they use the they collect the disease samples and then they try to find the drugs. But for the genetics-driven genomic drug discovery, it is focused more on the genome. So by using the genome profiles of the diseased patient, it can give us some hints about the effective drug targets that can be discovered by the traditional approach. So the first effective approach for enhancing the clinical practice is drug repurposing or the drug repositioning. So drug repositioning is a strategy for finding novel indications for the existing approved drugs or drugs in the clinical trials. The rationale of the drug repositioning is because the safety of the drug, the approved drugs has already been confirmed in the early stage clinical trials for its original purpose. So we repurpose the existing drugs, require less cost for testing the safety and the development and implementing the novel drugs. So here is two research examples about the drug repositioning approach. The first one is from the Professor Yukinori Okada from Japan, and by using the drug repositioning approach, actually they found that the CDK4 and CDK6 genes is the potential drug targets for the rheumatoid arthritis. And another one is from Imamura Sensei, also from the Japan, about the type 2 diabetes. So here is the two examples from our lab. That is that this study is conducted by our lab regarding the drug repositioning approach. The first one is about the drug repositioning to the familial short stature. And this study has been published in Journal of Biomedical Science. So for the familial short stature, actually we know that the genetic factors, the dysregulation in the endocrine system, cytokine and paracrine factors are implicated in the pathogenesis of familial short stature. However, the treatment choice of the familial short stature is very limited, with only the recombinant human growth hormone being available. Starting from the identification of 122 genetic loci related to the familial short stature, we use a genetic-driven drug discovery pipeline that based on the functional annotation to prioritize some potential biological genes. And these genes were suggested to be potential targets for therapeutics. And finally, we discover 5 druggable subnetworks, which contain in total 7 the familial short stature related genes and 17 druggable targets. So basically, this study provides a drug repositioning result that accompanied by corresponding targetable gene cluster, which is useful for the familial short stature therapy. The second study that also from our lab is about the drug repositioning to the COVID-19 infection. In this study has already been published in genomics. Because we know that it is in urgent need to rationalize some potential therapeutic targets to fight with the progression or the infection of the disease. So one of the effective and feasible way, faster way, is to use the pre-existing data about the SARS or the MERS virus as a surrogate for the SARS-CoV-2, since both lack the molecule responsible for the interferon antagonistic effects, which is the major pathological mechanism of this virus. So in this study, we finally prioritize 55 genes and 2,038 drugs to reposition currently available medications for the COVID-19 therapy. Some of the drugs that we found is already in the clinical trials, including the dexamethasone. So in addition, we also pinpoint 16 drug groups based on the ATC classification system with the potential to mitigate the symptoms of the COVID-19. So actually this paper is from last year. We done this study in last year. So at that time, the genomic and transcriptomic profiles of the COVID-19 is very little and is less. So we can just use the data from the SARS or the MERS as a surrogate to find the potential that may be repositioned to the COVID-19 infection. But until nowadays, there has many teams had released the whole genome or the transcriptome profiles of the COVID-19 patient. So our lab is now doing the research regarding the drug repositioning to the severity of the COVID-19 infection. So the second methodology I want to introduce is the Mendelian randomization. Mendelian randomization is actually it is a genetic epidemiological framework that used for the causal inference between exposure and the outcome. Here, the exposure can be the drug targets, and the outcome in the pharmacogenomic field is about the drug response, whether the drug efficacy or the drug side effects. So actually, it is a methodology that mimics the randomized control trial, so that it can be used for the causal inference. For the Mendelian randomization, we need to know that the suitable target for the therapeutic medications are not really limited to genes. But here the exposure in the pharmacogenomic field, we use the gene as example. So these drug targets do not necessarily, since the drug targets do not necessarily play causal role in the disease pathology, as they can be influenced by the disease states or other causes that affect the drug response. So one way to solving the causality problem with the help of genetics is the Mendelian randomization. Since in the population, the genotypes are assigned almost independently of the environment or the confounding factors when they are inherited from the patient. So those who have the genotypes that increase the exposure are in fact like assigned a high dosage of the exposure that is independent of other confounding factors. So that is why the Mendelian randomization is can be viewed as a mimic of a randomized control trial. And the third methodology is about the multi-omics integration. Since nowadays we have tons of genomics or omics data that is available, so we can try to incorporate those information together so that we can get more insight about the disease and identify the genes that may be used as a drug target, whether it is a novel target or actually it is has been discovered before, but for validation purpose. So here I just list some examples for you. Like the first one that we commonly do is about to incorporate the multifaceted biological research, including the protein-protein interaction network. This is, we can call this the proteome analysis. or the transcriptome analysis. The second type is we can combine the metabolomics data. By doing this, we expand the range of the candidate therapeutic targets, because the therapeutic targets not only about the gene products, but also about the gene products that are related to the metabolites. The third one is single-cell genomics. Single-cell genomics is recently a very powerful approach to reveal the cell type specific effects of the genetic variation. And it can also be used to fine-tune the genotype-phenotype relationship. So this is single-cell genomics. And there is also immunopharmacogenomics is another way to combine with the pharmacogenomic field so that can get more powerful insight about the disease and so on. So now I will give you a brief introduction about the immunopharmacogenomics, which is also one of the research field in our lab. So immunopharmacogenomics can be viewed as a combination of the immunogenomics and the pharmacogenomics. For the immunogenomics, we know that it is a research field that uses several tools, such as next generation sequencing, to analyze the complexity of the human immune system. Actually, this is not a very precise word, because in the field of the immunogenomics, there has many immune-related genes that can be studied, which including the HLA genes, or the KIR genes, or the TCR and BCR. So our lab, for example, in our lab, we majorly focus on the TCR and the HLA genes. And in the future, we are also trying to analyze the BCR. So this is for the immunogenomic parts. And for the pharmacogenomics, we already know that it is the research field that uses the genetic information for a better understanding of the drug responses. So the combination of these two categories resulting in the immunopharmacogenomics. So the immunopharmacogenomic is the major aim is to characterize the disease-related immune factors. Okay. And finally, to identify the genetic determinants for the efficacy and effectiveness of the immune therapy or the cell therapy. So maybe this is the final slide. Okay, so here is the three major aspects of the immunopharmacogenomics. Actually, in immunopharmacogenomics, any disease or any study that related to the immune genes can be viewed as one of the category of immunopharmacogenomics. But here I propose three major aspects about the immunopharmacogenomics. The first one is about the precision medicine. Actually, this is in this field is very challenging, because we know that if we want to profile the HLA or the TCR, BCR, actually is very difficult, because like for the HLA or the KIR genes, there are hyperpolymorphism across the population. And for the TCR and BCR, we know that because the combination of the VDJ genes, so they are extremely great complexity at there. So it is really difficult to profile them and then to analyze them. So this is the major challenge in this, in the precision medicine of the immunopharmacogenomics. The second field is about the cancer. In the cancer-related immunopharmacogenomic study, actually we need to characterize the neoantigen with using many omics data. So basically this experiment is very expensive and also need to have computationally intensive skills like to do this kind of analysis. because we know that to characterize the neoantigen in the cancer patient is important for the future cell therapy or the immunotherapy of the patient. So this is the major challenge about the cancer treatment in the immunopharmacogenomics study. And the third field is about the autoimmune or allergic disease. For the autoimmune and allergic disease, the challenge is about we need to provide a special temporal, it is like the space and the time of the immune system and the microbiome together. So this is also really challenging. Okay. Okay, here is my reference. And thank you for your listening. So I have finished my speech.
Okay, thank you, Dr. Henry. It's a nice presentation. I'm sure that there are questions from the audience. Therefore, we will move to the questions and answer sessions. Thank you very much, Dr. Henry. Okay, for those who have questions for Dr. Phennapa and Dr. Henry, you can raise your hand and we will unmute you so you can ask them directly. Please mention your name and your institution. You are also pleased to ask in the chat box. The time for the session is 20 minutes or 30 minutes. And to make it more interesting, there will be a chance for you to ask questions to get a door prize vouchers 50,000 in the form of pulsa for 5%. So please prepare your questions. Okay, now is there any question from the audience? Please raise your hand if you have any questions. For the committee, can you help me to find the raising hand? Okay, so there is Mr. Ikhwan Ridwan Rais and Mr. Rizky Rahmadi Pratama. I will give the chance first for Mr. Ikhwan. Okay, please, you can start asking your questions.
Okay, thank you very much, Mrs. Verda. Excuse me, so sorry. Okay, I think you can hear my voice, right, Mrs. Verda?
Yes, I can hear your voice.
Okay, thank you very much. Sawasdito Chao, Ajahn Phennapa. Good morning, Ajahn Phennapa. So I would like to ask about your presentation. Since the Thai government put the andrographis and Weissenbergia rotunda to the drug list for COVID-19, I think it's going to be huge amount to be needed in the marketplace, right? So how did your country fulfill the necessity of these two species because the needed of the huge amount and how you cultivate these two plants to fulfill the necessity? That's my first question. So then the second one is for the Weissenbergia rotunda. I still didn't heard about the dosage form, such like the andrographis. You explained about the dosage form for the andrographis clearly, but for the Weissenbergia rotunda, I think I didn't hear yet. I mean, for example, how many milligrams for the extract or the chemical active. Yeah.
Okay. For the first question is how can we get the species? I mean the plants, right?
Yeah, yeah. How to fill the necessity.
So, okay. So for the questions, I need to clarify first that For Rosenbergia rotunda is still in the study stage. So it's still in experimental study. So we don't still recommend it for the patient with COVID-19 yet, but it shows a very great result. So a very near future, I think it's going to be a pharmaceutical product launch out, and it might be the recommendation later, but there's a lot of room for the study because after the experiment, you need to test for toxicity, but the screening also show, the screening that I show in the slide also show the screening for cytotoxicity as well. So kind of in the first stage, it's clear that it's work and it's safe. Then the next stage is going to the clinical stage. If you remember my first slide, there's a study, right? Separate into experimental part and clinical part. So in the clinical part, we're going to start with the healthy patients first to find the good dose that we should use in the patient later. So after that, you go to the second phase or third phase for the clinical study, but that's in the future. For the first question, that how can we find it? For endographis and Rosenbergia rotunda, we use it a lot in Thailand already. Not only the medicine, but as the food as well. So generally, we use it in our daily life. So you can find it everywhere in Thailand. And for example, endographis paniculata, right now, the requirement is a lot because the recommendation just launched out just 1 to 2 months ago. So we have a lot of hospitals and pharmaceutical companies that invest in this type of the medicine. We have the herbal, the traditional hospital that focus on the Thai traditional medicine as well, and they're working for the agriculture. So sometimes the hospital or the company going to sign the agreement with the farmer and help in the control that the condition of the agriculture should be condition A, B, C, in order to help controlling the amount or quantity of the endographis. Does that answer your questions?
Yep, it's very clear. And after the government decided that endographis can use for this COVID treatment, so how technically, how do this endographis used by the patient or the people? Is it just by the prescription by the doctor, or the people can consume it by themselves and go to the drug store or any others?
Okay. Usually, like I said, it's in natural drug list, so it can be prescribed by the doctor, or you can go to the pharmacy and buy it. Generally, we use it for control. The dose, 20 milligram per dose, 3 times per day. So it's like maximum at 60 milligram per day is the recommendation one because it's already safe. Then for the case for COVID-19, that we increase the dose to 180 milligram per day is still under the study. Like I said, we need to control a lot. So for the case of COVID-19, you should under the investigation of the healthcare provider.
Okay, so your government is accepted for the treatment of COVID, but still also under investigation for the complete information about it. One more question for the last one. The side effect endographis, as I know, is like maybe you ever heard that some food or some herbal that have bitter taste, it can increase our appetite, right?
Yes.
You know it? Yeah.
Yes,
So it's,
yes, sure.
on the other hand, so it's make us want to be eat much more. So it's also maybe have another information they have to fill. Yeah, for example, for people that have maybe with a overweight patient that, yeah, this is a side effect that as I know, maybe. Is it important to investigation for this side effect for the increase the appetizer?
What can I say? DAR or adverse drug reaction and side effect is like a little bit different. So sometimes you need to monitor something that will lead to, the sign that lead to a condition that showing a severe condition. For example, the liver or kidney impairment. So you can monitor some marker like ALT or AST. For side effect, it's something that is might occur. But if you can tolerate that, it's worth to use the medicine. You mentioned about the loss of the appetite in some of the patients. For the bitter taste of herb, sometimes we use it for, what can I say, it's like stimulate the appetite as well. But in case of endographics, I mentioned in the side effect that it might make the patient lose appetite. That is side effect because we use in different dose. And some of the side effect is effect on the GI tract of the patients that make the GI move differently. And that means GI disorder. So some of patients might face with the condition of loss their appetite as well. So under the label, not in the label, sometimes try traditional medicine might prescribe for the patient to eat it, take it before meal. But usually we prescribe it after meal to avoid the GI disorder. But for the patient that lost their appetite, just sometimes, and under the investigation of the traditional medicine practitioner, they prescribe it before to increase the appetite as well.
Thank you very much for the answer, Doctor.
Thank you for your questions as well.
Yeah.
Okay, thank you, Dr. Mr. Irfan and Dr. Phennapa. We will go to Mr. Rizky Rahmadi Pratama for the next questions. We will unmute you and you can introduce yourself. Okay.
Okay. First, thank you, Mrs. Verda. I want to ask a question to Mrs. Phennapa. My question is, as we know, let me myself. My name is Rizky Rahmadi from University of Muhammadiyah Banjarmasin. And as go to the question, I want, as we know, a lot of drug target, we know. Example, like spike protein, main protease, or like cysteine-like protease, or papain-like protease, or LDRP, RNA-dependent RNA polymerase. I just done my research about plant that is Andrographis paniculata and Pisidium guajapalin. That the Andrographis paniculata can inhibit the LDRP. The drug target is LDRP or RNA-dependent RNA polymerase, and the Pisidium guajapalin can inhibit the 3-cysteine-like protease, the drug target. Now, I want question about drug target. In your opinion, or in your POV or point of view, what is the best drug target that from you, that POV of you? That is my question. Thank you.
For SARS-CoV-2 or something like that?
Yes, for SARS-CoV-2, for the drug target.
In my opinion,
yeah,
if the drug can inhibit all of them, that's the best. It will inhibit every step that virus involved in the progression of the disease. But if it can inhibit at the first step, viral entry, that's also the good, if you need to choose. Viral entry is my recommendation because after infect, you don't know that, like if you test that, it might not be 100% that you can block it, right? Some of them might get entering into the cells, but the less, the better, right? But if you focus on the RNA replication, RNA synthesis of the virus, that might be okay. That's good, or that's good as well. But thinking about that is cannot inhibit 100%. Some of them still have like the replication, right? So that's my answer. If you can block that both, that's the best one. If you can choose, I recommend viral entry.
Oh, viral entry, like spike protein or like S2 receptor?
Like S2 receptor or spike protein.
Okay, thank you, Mrs.
Welcome.
Okay, thank you, Mr. Rizky. Is there any other questions from the audience? Okay, while we are waiting for the next questions, I would like to ask to Dr. Henry. Yeah, Dr. Henry, I'm trying to connect your presentations with Dr. Phennapa. I just want to know, is it possible to apply the genomics in the development of natural products? As we know that in natural products consist of many substances. Thank you.
Yes, actually, the natural products in the context of the genomics-driven drug discovery approach is can be viewed as one part of the ligand, because we know that in the pharmacogenomic study, we need the disease and we need the genome information, that is the gene, and then we need the ligand. The ligand is actually the drugs. So whether it is the approved drugs but a reposition to another indications, or whether it is the drugs in the clinical trials, or whether it is still is not a drug, it is maybe a natural products, or it is some ligands in the animal experiment. So it is all included in the category of the ligand part. So basically, if we want to connect the natural products to the pharmacogenomic study, so the first step maybe is we need to profile which is the drug target or the gene target of the natural products. Like for one natural product, the compound, if we can characterize the genes that it inhibit or it activate, so that we can use this information to link to the disease, that whether given a disease, it is potential to treat it or not.
Okay, thank you. Okay, so is there any questions? You can ask in Bahasa Indonesia if you are not confident with your English. We can try to translate it for you. Or also you can use the chat box. I think there's no further question from the audience. So, if that's so, we can end this session. And yeah, okay. I would like to ask once again to the audience, is there any questions for Dr. Phennapa or Dr. Henry? Okay, there's no questions from the audience, and so we can end this session now. Thank you very much, Dr. Phennapa and Dr. Henry, for your nice presentation. And we will end this, but I hope you can stay here after the last presenter, because we will give you a certificate after that. Okay. So after this, we have a little interesting show for you that will be guided by our Master of Ceremony, Mr. Ricky. What will it be? I will give the mic back to Mr. Ricky to show you the answer. The time is yours.
Thank you, Mrs. Verda. I think we all agree that this is a very interesting presentation, a lot of information and knowledge that we can get from the presentation. And I hope this can be our inspiration and example, especially for the research to fight COVID-19 and for better method to maximize drug effectiveness via pharmacogenomics. Ladies and gentlemen, let's take a break for a moment. While take a break, let's watch a campaign video about vaccine made from the collaboration of participants today. So please enjoy the video. Thank you.
Kasus positif terkonfirmasi
dari sekitar
7
wilayah menjadi
pasien yang
setelah
sudah
terpapar
sembuh
COVID-19
dan pasien yang
Tepat pada tanggal 2 Maret 2020, untuk pertama kalinya pemerintah Indonesia mengumumkan kasus positif COVID-19. Bahkan hingga sekarang ini, kasus tersebut masih mengalami peningkatan.
Penyebaran virus corona memang sudah terjadi, tapi bukan berarti kita harus pasrah dan berdiam diri. Jangan panik, tetap siaga dan jaga kesehatan diri dan keluarga.
Penyebaran virus corona bisa kita kurangi dengan selalu menjaga kesehatan, salah satunya dengan menjaga imun kita agar selalu sehat.
Salah satu upaya pemerintah untuk menekan peningkatan angka kasus COVID-19 di Indonesia yaitu dengan melakukan penyediaan vaksin.
Menurut Menteri Kesehatan Republik Indonesia, setelah dosis kedua disuntikan, tubuh masih memerlukan waktu 2 hingga 3 pekan untuk membentuk antibodi. Setelah terbentuk antibodi itulah, resiko tubuh terpapar COVID-19 menjadi kecil.
Hindari isu-isu vaksin COVID-19 yang sumbernya tidak jelas dan tidak terpercaya. Kalian dapat mengakses berita-berita terkait COVID-19 pada sumber-sumber terpercaya seperti berikut ini.
Dan perlu diingat juga bahwa upaya pencegahan COVID-19 tersebut tentunya tetap harus disertai protokol kesehatan. Misalnya dengan selalu menjaga jarak fisik, mengenakan masker, dan rajin mencuci tangan.
Ayo, jangan takut divaksin. Tak kenal, maka tak kebal.
Hello, my name is Philipps-Universitat from Faculty of Pharmacy, Siam, University, Thailand. Since 2019, SARS-CoV-2 infection got spreading throughout the world and has turned into a pandemic. This is an urgent global health problem affecting everyone's lives. To help solving this problem, it is our duty to take part in it. There are several ways studying for ourselves by wearing masks, washing hands, and staying physical distance at least 1 to 2 meters away from other people. However, since the pandemic keeps continuing, the most effective solution is to get the vaccination as fast as we can. Fortunately, various vaccines are available and that are identified and it is continually developed for better version. As a part of the social and a pharmacy student, I would like to invite everyone to get vaccines. Not only preventing your body from COVID-19, but also protecting our family and helping our social back to normal.
Vaksinasi merupakan salah satu upaya pemerintahan Indonesia untuk memutuskan rantai penyebaran infeksi virus corona dan menekan angka kasus COVID-19. Vaksinasi COVID-19 bertujuan untuk menciptakan kekebalan tubuh herd immunity agar masyarakat menjadi produktif dalam menjalankan aktivitas sehari-hari. Namun faktanya 7,6% masyarakat Indonesia tidak mau divaksinasi dan 24,2% menolak dengan alasan takut efek samping yang akan muncul.
Berdasarkan Fatwa MUI nomor 2 tahun 2021, vaksin COVID produksi Sinovac dinyatakan suci dan pastinya halal. Seluruh masyarakat boleh divaksinasi dengan ketentuan 1. Lebih dari 18 tahun, 2. Sembuh dari COVID lebih dari 3 bulan, dan yang tidak boleh divaksinasi adalah 1. Wanita hamil, 2. Tekanan darah di atas 180/110 mm/Hg, dan yang pasti masyarakat yang menderita penyakit kronis.
Ingat, karena vaksin tidak membuat sebal 100%.
Maka harus tetap mematuhi protokol kesehatan seperti
mencuci tangan dengan air dan sabun,
memakai masker,
menjaga jarak,
dan hindari kerumunan dari masyarakat sekitar.
Ayo, dukung pemerintah
untuk mensukseskan vaksinasi COVID-19.
Vaksin COVID halal kah? Yang jadi pertanyaannya, apa sih komponen yang membuat suatu vaksin itu halal atau tidak halal?
Yang paling kritis itu adalah pertama itu enzim ya, dan tripsin itu sangat sensitif ya dari segi kehalalan karena kebanyakan tripsin itu diproduksinya dari babi. Lalu juga gelatin ya sebagai bahan dalam pembuatan vaksin. Kalau dari segi kualitasnya gelatin yang dari porcine atau dari babi ini memang secara kualitas lebih bagus.
Assalamualaikum warahmatullahi wabarakatuh. Kita dari Prodi D3 Farmasi dari Universitas Muhammadiyah Lamongan. Yuk, mari kita ketahui apa sih itu vaksin. Vaksin merupakan antigen yang dimasukkan ke dalam tubuh untuk kekebalan tubuh terhadap penyakit. Manfaat vaksin 1. Mencegah terkena COVID-19, 2. Mencegah penyebaran COVID-19, 3. Menambah kekebalan imun tubuh, 4. Melindungi generasi selanjutnya. Efek samping yang mungkin bisa terjadi adalah demam, menggigil, dan bengkak atau nyeri pada area suntikan. Penanganan pada efek samping. 1. Perbanyak minum air putih. 2. Konsumsi paracetamol sesuai dosisnya. Yang ketiga, kompres dengan air hangat apabila terjadi demam. Dan gunakan pakaian yang nyaman. Selanjutnya, kompres dengan air dingin pada area suntikan yang bengkak atau nyeri. Vaksin tidak 100% mencegah COVID-19, tetap patuhi protokol kesehatan dengan memakai masker, mencuci tangan dengan sabun dan menggunakan hand sanitizer, tetap menjaga jarak minimal 1 meter, dan hindari kerumunan. Ayo, dukung pemerintah mensukseskan program vaksin COVID-19. Terima kasih. Wassalamualaikum warahmatullahi wabarakatuh.
Protect yourself with vaccination. Protect yourself and others by getting vaccination very soon. Protect yourself and others by getting vaccine very soon. Protect yourself by vaccination.
Protect yourself and others by getting vaccination soon.
Protect yourself and others by getting vaccination soon.
We should raise our awareness together to fight this pandemic, especially the vaccine, which help us end the pandemic that has been running for 2 years. Ladies and gentlemen, let's continue our webinar session to Mrs. Verda. Back at you, Mrs. Verda. Are you ready?
Yes, thank you, Mr. Ricky.
Thank you, Mrs. Verda. The time is yours.
Okay, it was a nice video about vaccine, isn't it? We hope we can remind you how important it is to vaccinate yourself and your beloved ones, especially in this pandemic. We'll come back with me again in the second conference sessions. In this session, the presentation will be delivered by Professor Dr. Irwandi Jaswir. Welcome, Professor Dr. Irwandi. He is from the International Islamic University, Malaysia. And this is a summary of his CV because his CV is 44 pages long. Professor Dr. Irwandi Jaswir is a professor in food chemistry and biochemistry at the International Islamic University, Malaysia. He is a former director and deputy dean, International Institute for Halal Research and Training, International Islamic University, Malaysia. He got his Ph.D. degree in food chemistry and biochemistry in University Putra, Malaysia, and he has experience as a Ph.D. exchange program, Department of Food, Nutrition and Health, University of British Columbia, Canada, and as a postdoctoral fellowship in Lipid Biochemistry, National Food Research Institute in Tsukuba, Japan. As I mentioned before, his CV is 44 pages long. Most of them are about his work in halal food, halal pharmaceutics, and natural products. His published papers are 156, and here are the 3 of them. This is the newest. He also writes 30 book chapters and monograph in halal areas and natural product development. The topic that he will deliver at this occasion is halal pharmaceutical research science, drugs, and biotechnical products. Okay, Professor Dr. Irwandi, are you here?
Yes, Assalamualaikum.
Waalaikumsalam warahmatullahi wabarakatuh. My name is Verda. I am the moderator in this event. Okay, Dr. Irwandi, the time is 40 minutes and now it's all yours.
Okay, thank you. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Alhamdulillahirrahmanirrahim. Wassalatu wassalamu ala asyrafil anbiya'i wal mursalin wa ala alihi wa ashabihi rasulillah ajma'in. Thank you, Madam Chairperson, for kind introduction of me. And first of all, I would like to thank the organizing committee for inviting me to share a little bit on halal pharmaceutical research sciences, and especially to the Faculty of Pharmacy, Universitas Ahmad, Dahlan. My name is Irwandi Jaswir. Currently, I'm attached to International Institute for Halal Research and Training, IUM, and also a lecturer at Universitas Ahmad Dahlan, Faculty of Pharmacy. Can you see my slide now?
Yes, we can see your slides.
Yeah, okay, thank you. The title given by the organizing committee is the Halal Pharmaceutical Research Sciences, Drugs and Biotechnology Products. We are talking about halal now, yeah. Why halal? Yeah, not only us, everyone now talking about halal, because the market of halal industry is huge, and halal is one of the the fastest growth of the industry nowadays. There is a data saying that the market of halal industry is about 3.1 trillion US. Okay, and then that's why, because it is a big market, and then everyone is talking about halal. In 2017, it showed that the most of portion of the halal industry goes to non-food, whereby in this case is pharmaceuticals or cosmetics, while others like services, including logistics, etc., contribute about 25% or more. If we look at here the amount spent by Muslim for halal products and services, we see that it is a very, very big potential for us as consumers, also as producers. I'm interested in the pharmaceutical and cosmetic, whereby here 221 billion predicted, yeah, to be 2023. We look at the global Islamic economic indicator. We see now Indonesia is ranked number 4, yeah. And Alhamdulillah is a big jump from number 10 or 11 in the previous years. Of course, yeah, we thank the government for putting initiative, attention to develop halal industry. But the real question actually is how much that we earn from this industry. Even though Malaysia, with being number 1 for many, many years, the same question is given by the stakeholders, how much actually Malaysia receive from this industry. We do not want to become among the top in the world, but at the same, but in reality, we do not get anything. Yeah, we may be ranked higher, but we do not get as per shown in by the ranking. Why? Because many countries do not actually do not think much about the ranking. They are not very, very particular about the ranking, but they make a lot of profit from the halal industry. The countries like Australia, New Zealand, Brazil, Korea, Japan, China, etc., they put emphasis to develop halal industry, but they are not ranked in this world. Yeah. So for meat, halal meat, for example, New Zealand, Australia, rank number 1. Yeah. But for halal chicken, Brazil is produces number 1 in the world. But they are not ranked here. So the intention why I show this indicator is that we do not want to become consumers, yeah, of those products coming from overseas. Of course, during the pandemic, yeah, some of the halal sectors increase, some decrease, many decrease actually, and then some just remain the same, yeah. And and halal food and halal pharmaceutical are among the one who increase or at least the same level as previous one. Why? Because no matter what, people still need medicines and also food. So halal industry evolved from traditional to emerging. And we see here cosmetic, personal care, and healthcare products are among the emerging one. Because people now talking about the quality of life, and halal is part and parcel of that. When people talk about halal, they are not talking simply because of religion obligation. No, yeah. Some of them or many of them maybe are non-Muslim, but they are very particular about halal products. For example, yeah, the top 2 countries who import Malaysian halal product in the world, both are non-Muslim countries, yeah, like China and UK. Yeah, so these 2 countries are the major importer of Malaysian halal products. While the halal meat from New Zealand, yeah, halal meat from New Zealand, 60% of them are exported to non-Muslim countries. Imagine, yeah. So people see halal product as a quality one. This is just give you an example. I was invited to this country in Seoul in front of members of parliament. So this is parliamentary building, yeah, National Assembly building in Korea. And if we see how serious the developed halal industry, mashallah, yeah. Every time, normally before COVID, we went to Korea every 2 months to educate the industry players and then to give them solution to certify the product, etc. So they are very, very serious about halal, and this is a warning or wake-up call for Indonesia. If we are not serious developing halal industry, we may be just consumers of those products. There are 3 main issues in halal product: raw material processing, authentication, and these 3 are also applicable for pharmaceutical products, drugs, and other biotechnology products. We know the holistic concept of halal from A to Z, yes, source of origin till end product. And then in every step, there is possibility that the product is contaminated through ingredients or through processing or through mix, what we call is cross-contamination, yeah, in every step. Yeah, I know that for pharmaceutical, the step might be longer, yeah, might be longer. Okay, this is a potential critical areas. The ingredients and additive are among the critical points in the halal pharmaceuticals. Pigs and its byproducts, okay, enzyme in the production of vaccine, for example. What about the trypsin? Yeah, used for the production. And then what about the ingredient used to grow the microbes? Because most of vaccine now are microbial vaccine. And then what about the emulsifier use? Emulsifier is used in all the products, yeah, not only food and cosmetics, but also in pharmaceuticals and others also, yeah, issues. For example, here the gelatin from pig, 65% of gelatin from pig in the world are derived from pig, yeah, and then we know that in the capsule, yeah, production of hard capsule, for example, 60% need gelatin, yeah, in the materials. Recently, we are given amanah here by a Committee National Economy and Keuangan Syariah under Ministry of Finance Indonesia to study what the ingredients that Indonesia has, but until now we are still importing the products. Yeah, the materials or the ingredients, and among them are gelatin, flavors, oleoresin, seasonings, enzyme, food colorants, the antioxidants, emulsifier, anti-foaming agent, anti-caking agent, humectant, glazing agent, and some other. Yeah. So this is the highlight of our findings, and the books was just launched by Ministry of Research and Technology last year in 2020. So for those who are interested, you may download this book for free, and then it can be taken as a guidance for us when we develop, especially for pharmaceuticals. We know that some of the ingredients used for food also used in pharmaceuticals. For example, colorants, or maybe oleoresins, gelatin, emulsifier, etc. What gelatin? It is a protein, a denatured collagen, the source normally from skin and bones of the animal. So it must come from the animals, otherwise the terminology become misleading. Multifunctional hydrocolloids. And then it is used, as I mentioned, in all products that we consume or we use here in our daily life. Food, cosmetic, and pharmaceutical. Of course, some other industry like photography, etc. And the demand for gelatin, halal gelatin, actually is very, very big. Indonesia import in 2017, 395 billion rupiah, 390, almost 400 billion rupiah. Imagine we import a lot. And then I may say that we do not have a big gelatin plant in Indonesia. We have very few, but maybe in the very, very small scale or industrial or home industry scale. In terms of research and development for halal authentication for food, pharmaceutical, cosmetics, we may use various types of instrumentations for analysis, like infrared spectroscopy, electronic nose, differential scanning, colorimetry, molecular biology, chromatography, GC, GC-TOF, GCMS, HPLC, etc., and biopotential telemetry. Okay, this is infrared spectroscopy, and then this is the spectrum. Of course, we make questions about the sensitivity, but at least it can be used for monitoring and the screening process. It works based on the functional groups, and then in this case, at the wave number 965, we can take it as a signature for the spectrum to distinguish the concentration of lab. Electronic noses can be used also for halal detection, especially to detect the volatile component. That can distinguish if we realize that halal and non-halal fats or oils, for example, can be distinguished by the flavor, by the smells. That's why if we can use something nose related to detect the volatile component, and then it is a good idea. Okay, this is the machine for electronic nose, and this is the spectrum. The challenge here, of course, how we interpret the spectrum. Luckily, nowadays we have computer software program that can detect, especially for this kind of analysis where we can make it in the spectrum like this, and then we can see the pattern for lard compared to palm oil, peanut oil, corn oil, coconut oil, sesame oil. Okay, this is what we do using difference scanning colorimetry, and then you see this the pattern for exothermic endothermic process, or we call this is energy, energy pattern. Okay, of course, molecular biology technique is the most reliable one nowadays because it can detect the fingerprint of the component. Using real-time PCR, we can distinguish a halal and non-halal animal, pork compared to chicken and cows. And then here, the concentration of the non-halal substances can be detected using real-time PCR as well. We have a study before, in Middle East country, whereby we identified the DNA, porcine DNA, we extracted from the commercial gelatin available in the market, and the commercial gelatin is used in the various halal, various food products. And then the same technology can be used to detect whether the medicine that we use can contain the non-halal substance or not, by examining the existence of DNA, porcine DNA. And to make us surprised at the time, upset, about 20% of the food here in that country, particular country, contains porcine DNA in the gelatin. This is how serious the issue is. This is a lot fatty acid maltol esters profile, and then we can see, identify, and for these three formula, three types of fatty acid, and then there are some differences, especially for a lot here. You see a lot, these three fatty acids are there, but not for other animals or halal fat. This is a lot. Okay, and then the alcohol and hammer can be used. Biotechnology can be used to detect the alcohol and hammer. Some of drugs contain alcohol. Some of the personal care, for example, mouthwash. Mouthwash. Mouthwash is contain a lot of alcohol, and then in one study we found that in Malaysia some of the mouthwash contain about 29% of alcohol, and then we distinguish this using GCGC12. Okay. And then this is a gelatin pharmaceutical cosmetic, and then using capsule, in the tablet, and the lozenge, and cough drop, and the stabilizers and application. And then the common non-halal ingredients, other than what we have discussed, is placenta. It is used in cosmetics and also some drugs. In placenta, in cosmetic, it is used as anti-aging. And then some of the placenta are produced in non-halal way, either from non-halal animal or from halal animal but not slaughtered according to Islam. This is one of the study that we conducted, rapid detection of porcine gelatin in dental materials. In dental materials, this is how we do this, and then using FTIR first, and then DNA analysis, and then after that we study using PCR. So this is specificity of porcine and bovine primers. I just would like to inform that the challenge for using PCR for study of halal basically is only for how to extract the DNA, and then the rest to me is not that difficult. That's not difficult. Okay, and then this is electronic nose architecture. How we do this using electronic nose, and the design, and then this is what I mentioned. detection of ethanol in mouthwash using portable electronic nose. So this is some they were finding more than 10%, yeah, but basically some of them are more than 29%, almost 30%, yeah. And then the past and current research on gelatin, and the most recent one is we use this gelatin. We studied the gelatin from local goat, yeah. For pharmaceutical capsule, we produce hard capsule using our halal gelatin extracted from kambing, yeah, local goat. And then we work together with University of Indonesia, UI, yeah, Faculty of Pharmacy. This is our pilot plant of gelatin and derivative. And then there's some of our students using gelatin, nanoparticle, and for drug release, for drug-loaded fish gelatin, yeah. So this is a 4 PhD student working on gelatin nanoparticles and gelatin production. And one of them studied for drug-loaded application, especially for salicylic acid, yeah, salicylic acid, and one for functional food. This is how we prepare the fish gelatin nanoparticles. Okay, we use still the conventional one, yeah, glutaraldehyde. Okay, this analysis, yeah, using just now using SEM, the TEM, and then atomic force microscope, and then the release, yeah. And we found that consistently, yeah, high molecular weight, yeah, contained gelatin was producing gelatin concentration, and then we get the size below 200, yeah, considered the nanoparticle. And then the 3 factors have significant effect on particle size, which is pH, acetone concentration, and glutaraldehyde volume. Okay, next one is a lot of detector pen, yeah, lot detector pen, or we call lead pen, yeah. And then this one is we receive a grant from the government, and then the grant was all handed over by Prime Minister at the time. And then we produced several papers from this study. And then lastly is about immune booster related to drugs. We, as our expertise in these natural products, we produce immune booster in order to fight, yeah, for COVID-19 pandemic, yeah. So our product name, immune booster, etc. We see this, and then this is totally from local herbs, yeah, from local herbs. It is based on our research, long research, about more than 25 years. Some of the herbal contain antivirus, some of them contain, of course, energy antioxidant to boost our immunity. There are 4 products in this, yeah. This is my own products, yeah. Number 1 is turmeric plus, yeah. It can relieve abdominal pain, cleanse body system, improve digestion, and reduce body heat. And then andrographis, yeah. Andrographis plus, or Indonesia, I think very famous recently, yeah. Andrographis can control blood pressure, strengthen immunity, enhance blood circulation, and energy recovery. And just for your kind information, that in Thailand, andrographis is taken by the government as the medication for COVID patients. Yeah, this is already announced by the government. Yeah, and then you can take this service very, very effective, efficient. Yeah, because it can. It has antivirus, properties, And then Momodica Plus, this is also can reduce blood sugar, remove excessive air in the body, boost body energy, and improve urinary system. This is what we have done, yeah. And then finally, I would like to introduce our Halal Research Journal. We call it International Journal of Halal Research. And then we welcome everyone to submit the papers, and also your contribution is very much appreciated. appreciated. I think that's it, yeah, ladies and gentlemen. Finally, this is my latest CV produced published by Gramedia. Yeah, I'm talking about halal in this book. With that, I end my presentation. I get back to the Madam Chairperson. Assalamualaikum.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Professor Irwandi, for presenting such enlightening topics. Now I will give the opportunity for the audience to ask questions to our halal expertise here. If there's any questions, you can raise your hands. Is there any questions? While we are waiting, I would like to ask about the use of alcohol.
Yeah.
If you know that in a food, for example, there is alcohol in the ingredients, will it make the products not halal? Well, the intention of the alcohol is for something else other than to make someone drunk. Because I ever found a product, a food product with halal logo on it, but it also contained alcohol.
Okay. Okay, thank you, Ms. Verda. Yes, I'm going to give you the fatwa. Okay, according to the fatwa Majelis Ulama Indonesia, in the food application, you can use alcohol, of course, a food grade, not the alcohol in the lab. Don't misunderstand. You may use a food grade alcohol as long as it is not khamar. Yeah, khamar is alcohol derived from fermentation of fruits or carbohydrate, and it is purposely produced as a drink. Okay, so for khamar, the fatwa says is haram. Okay, no matter concentration is, meaning that if you have wine or liquor or beer, they are khamar because they are produced purposely as a drink. Okay, and for khamar, the hadith said if it is haram in large quantity, in a small quantity is also haram. Meaning that if you have wine, okay, or somebody, not you, if somebody has wine and they put the wine into the food, for example, you go to Italian restaurant, they put the wine into pasta, right? So the pasta become haram, even though only one drip of wine. So the fatwa is for khamar is no tolerance, zero tolerance. But for other than that, like alcohol as a solvent, we call this petrochemicals. Yeah, it is produced. We call it industrial ethanol. It is produced from petrochemical pathway, not from the fermentation of the fruits. So for that, according to fatwa of MUI, you may use it up to 0.5%, okay, up to 0.5%, but in the final product, it is not detectable. So the fatwa says like that. So meaning that we can use it, normally this one for solvent, because some of the bioactive compounds are not dissolved in water. We need organic solvent. So this is what they can, the fatwa says. But of course, the limit 0.5 differ from one country to another. In Malaysia, only 0.1% and coming 0.2% because they are discussing now. And in the US, you can use up to 1%. That's the fatwa says differently. So for the fatwa said, for out external use, external use, let's say for or cosmetics, or for maybe for topical medicine, yeah, you may use it. Yeah, you may use it no matter, regardless the concentration. So this is what I know, because of course limited, yeah, but this is what I know about the alcohol. Thank you.
Okay, so you said that as long as not from hammer, it's okay to use it.
From hammer, not okay.
Yeah, it's not
yeah,
okay.
For industrial ethanol is okay. So as long as we follow the fatwa just now.
So it's a basic question. The hammer is a fermentation product, right? How about Taipei?
Yeah, that's why when we refer back to Taipei, of course, people may have different opinion. Yeah, this is what I say from the fatwa. So from the fatwa said that Taipei, the intention to produce Taipei is produced not as a drink. You know that Taipei is a food. You know, drink is in Arabic. Khamar means drink, referring to drink. So this is the reason why Taipei is considered okay. But some said it's still not okay. So I leave it. Yeah, but some ulama said okay because it is produced not as a purposely as a drink. Yeah, unless you produce Taipei and then you take the water and then you drink it, so it's become khamar then. Yeah, so intention plays important role in Islam. Yeah, intention is very, very important. So this is my understanding until now. Yeah, but of course there are some who are very, very careful. They do not take Taipei at all. Yeah, so we leave it to the person. Yeah, but this is the majority fatwa in this region said the Taipei is acceptable. Yeah, because it is not a drink. Okay.
Okay.
If any questions, yeah, you may ask question in Bahasa Indonesia, yeah, if you want. Yeah,
oh, yeah.
No problem. I will answer in Bahasa as well.
Professor Irwandi is working in Malaysia, but you come from Indonesia, right?
Yeah, I'm Indonesian.
Yeah.
I'm Indonesian, yeah. I'm from Padang, Bukit Tinggi.
So
Yeah,
if the audience wants to ask in Bahasa Indonesia,
yeah, no problem. I will try to ask.
Okay, there's Mr. Iwan. He will ask you questions. Mr. Iwan, please. You can ask.
Yeah, thank you, Mr. Verda. Assalamualaikum, Professor.
Waalaikumsalam.
Thank you very much for your nice explanation about halal product. So I interesting with your product at your product that you display at the last one for immune booster. It's show that you got the halal logo already, right? It's from Malaysian government. What I want to ask is about what kind of type for the halal test to get that halal logo from the government, maybe from Indonesia also, if you know. I mean,
okay.
Yeah.
Okay, thank you, Brother Ikhwan. Yeah. Halal, these products, we are still applying, JAKIM certificate. Okay, in Malaysia, halal certification is under JAKIM. We applied, our company applied for that. But we get the halal certification from Korea halal certification because it's easier for us to get it. Yeah, but basically this is a natural products. And then it is all are from plant based, dari tumbuhan semua. Jadi it is I know that it is confirmed halal, but you cannot tell everyone this confirmed halal without having that logo. So the process in Malaysia, first is for pharma. In Malaysia there is no such undang-undang nomor 33 tahun 2014. We don't have that undang-undang in Malaysia. We are not, it is not compulsory for us to get halal certification. Yeah, in Malaysia, the undang-undang still like Indonesia before undang-undang nomor 33 2014. Indonesia now every product must have certification logos, and because mandatory. In Malaysia, still voluntary. So normally during the halal certification, what we do, in Malaysia is that we submit all the ingredients. This sama prosesnya di Indonesia and Malaysia the same. Yeah, you submit all the ingredients online, and then the auditor will verify first. Yeah, with no such testing. Yeah, the testing only done if there are some dispute or there are some disagreement between manufacturers and then the auditors. Ya, kalau auditors kata, oh rasa perlu ada ini nih, kata, tapi manufacturer bilang tidak ada, then we go to the lab. Yeah, so normally in most cases we just send, we all be audited using the ingredient list that we submitted. But of course we have to submit with the evidence. Yeah, with the evidence. For example, we said that we use gelatin, halal gelatin, and then where do you buy from? And then what the proof of the receipt or the purchasing or the whatsoever. Yeah, so normally we do that. Not every time you get certification, you have to get your product tested. No, it will be tedious and then time consuming and more expensive as well. Yeah, very costly becomes. So normally we do this. And then in halal certification, there is also positive list. Positive list is the ingredients or material which is already confirmed halal. So you don't need to check anymore. Ya, jadi ada yang namanya positive list yang udah jelas ya. Contohnya misalnya apa ya, misal gula atau apalah contohnya, yang memang udah confirm halal, nggak perlu dipertanyakan lagi ya. In most cases nggak perlu, you don't have to question it anymore. Ya, tentu saja ada, oh nanti kalau gimana kalau terkontaminasi, ya itu udah lain cerita ya. Okay, so this is what we do. So far we applied, our companies applied, and then it takes some time also. Not you apply today and then tomorrow we get it. No, it takes some time. But since the pharmaceutical in Malaysia at the moment, not made compulsory. Yeah, for food, many of the industry go for certification. Yeah, but for pharmaceutical and supplement, not that many. Yeah. But of course, yeah, from our company, our intention is still want to get the halal logo from Jakim. Thank you.
Yeah, thank you, Professor, for the explanation. Ms. Verda, come to you.
Okay, thank you, Mr. Irfan and Professor Irwandi. Maybe is there any other question from the audience? If there's no questions, I have one more question.
Yeah, please.
Is there any way to identify? Is there any way to identify a not halal substance coming from halal sources? For example, maybe can we identify gelatin from cow that is not processed in halal way?
The method, thank you for the question. The method at the moment, to be honest, yeah, I think it's very, very difficult. Yeah, I don't see any research or finding, yeah, to see whether the animal is coming from halal animal and slaughtered according to Islam. Yeah, only proper audit can tell this. Proper audit, normally we go and check, we do how the process. And then normally we check whether the slaughtermen are Muslim and whether in the slaughterhouse, slaughterhouse must be certified slaughterhouse, halal certified slaughterhouse. But if you ask me scientific method to detect whether the animal is slaughtered according to Islam or not, at the moment it's very difficult, but not impossible. I believe it's not impossible because we have once conducted study for animal which is slaughtered properly and slaughtered beyond the voltage allowed. Meaning that ada stunning process, ya. Stunning process is the electric current which is passed through to the animal, and the animal become unconscious before being slaughtered. Jadi ayam yang pingsan dan ayam yang segar yang tidak pakai stunning, tidak pakai elektrik, ayam yang overstart, voltage listriknya itu terlalu tinggi sehingga ayam itu mati sebelum diassembly before being slaughtered. They are different in terms of protein profile in the neck area. So this is our study before, and then one of my our PhDs, I'm co-supervisor at the time. They can detect this. So I believe, yeah, but of course we need to do more research on this. Yeah, we need to do more research on this. Yeah, I believe there's some different in terms of microbial counts, in terms of protein profile, etc. So, and not only that, for chicken, for example, the texture of the meat are also different organoleptically. Yeah, so we do sensory evaluation, and then the what we call is the texture of the meat. Yeah, so will be different. Okay, but of course we need more study. That's why we need researchers from just Ahmad Dahlan to do this more. Thank you.
Okay, thank you for the answer, Prof. Irwandi. I will ask to the audience if there's any questions.
Tanya Bu Verda.
Who is this?
Aziz.
Oh, okay, Mr. Aziz, please.
Okay, thank you, Prof. Irwandi. I want to ask you about. Okay, I'm using to bahasa Indonesia.
Yes.
Okay. Oke, saya mau tanya nih Prof, apa ada perbedaan secara chemical ketika ayam itu disembelih pakai asma Allah sama yang tidak? Secara chemicalnya.
Ya, terima kasih Pak. Itu aja ya?
Iya, oke.
Iya, saya coba jawab ya,
Okay.
karena belum. Bismillahirrahmanirrahim. Pak Aziz, terima kasih pertanyaannya, dan ini juga pernah ditanyakan ke saya sebelumnya, karena saya banyak meneliti penelitian halal gitu ya, halal, tidak halal, ingredients, method, dan sebagainya. Bahkan ketika yang saya sampaikan barusan, ya kita pernah menguji binatang itu yang disembelih pakai stunning pakai listrik yang dialirkan ayam mengalirkan aliran listrik ya supaya dia pingsan. Itu tuh ada voltase maksimal yang dibolehkan. Ya, jadi kita beda ayam tidak pakai stunning, ayam yang dipakai listrik yang dibolehkan, dan listrik yang tidak dibolehkan, itu voltase ketinggian. Sehingga ayam ini sudah mati sebelum disembelih. Kita sudah pernah penelitian, dan ini berbeda secara kimianya, yaitu profil proteinnya dan microbial count-nya, itu berbeda. Tapi kita belum pernah penelitian tentang pembacaan bismillah, ya, bacaan basmalah. Jadi apakah, dan saya yakin pasti ada ya, saya yakin itu, tapi nggak tahu membuktikannya karena belum ada penelitian ke sana. Penelitian yang pernah ada oleh teman saya di Department of Veterinary ya di Universitas Pertanian Malaysia, itu penggunaan musik soft ya untuk penggunaan musik untuk ayam yang sebelum disembelih. Jadi beberapa ayam itu kan umurnya 28 hari ya dan 35 hari ya, tergantung varietasnya. Ada yang dia peneliti itu 2 minggu terakhir itu ayam itu dikasih hiburan lah. Jadi musiknya dari musik yang cadas, musik rock and roll, musik klasik itu tekstur ayamnya berbeda. Jadi ayam yang di setiap harinya didengarkan musik klasik kan soft banget ya. Ya, kita bisa tertidur dengerinnya mungkin ya kalau kita bisa. Itu ayam itu teksturnya berbeda. Kalau tekstur berbeda pasti komposisinya juga berbeda di dalam ya. Nah, tapi mungkin akan menarik ya kalau kita coba ayam setiap hari dibaca surah Al-Kahfi gimana gitu ya, atau surah ini atau kita mungkin akan ada perbedaan ya, insya Allah ada gitu. Saya yakin itu ya. Tapi sementara ini kalau dibaca Bismillah yang hanya sesaat itu wallahu alam, itu juga belum bisa menjawab saya ya, kalau baca Bismillah ya. Tapi kalau waktunya diprolong ya, waktunya agak lama misalnya 2 minggu atau selama hidup lah, kan cuma 1 bulan ayam broiler itu cuma 1 bulan. Itu mungkin akan ada signifikan ya, saya yakin ya. Kalau dibaca Bismillah kan juga sebentar banget ya, cuma 1 detik Bismillahirrahmanirrahim ya itu. Dan bahkan ada fatwanya sekarang kalau untuk hewan yang di penyembelihannya secara shift ya, artinya rumah potong hewan itu kan 3 kali shift itu kadang-kadang ya, 8 jam. Ada yang sudah memfatwakan baca Bismillah dan doa itu hanya di awal setiap shift ya, Bismillahirrahmanirrahim lengkap doa-doanya. Jadi enggak, karena misalnya ya di yang pernah saya kunjungi di Malaysia ini, rumah potong hewannya punya salah satu restoran fast food restoran ya, saya nggak mau sebut mereknya lah, tapi Anda tahu lah ya, fast food restoran ini 1 hari itu memotong ayam itu sampai 300.000 ekor ya, itu restorannya seluruh Malaysia. Jadi 300.000 ekor kalau baca Bismillah 300.000 kali kan juga capek itu, dan bisa jadi nggak betul lagi bacaannya ya, makanya ada keluarkan fatwanya. Jadi nggak Bismillahirrahmanirrahim nggak gitu caranya. Jadi Bismillahirrahmanirrahim. Jadi ada fatwanya untuk itu gitu ya. Jadi kalau udah seperti itu di awal shift ya, tentu saja nggak ada lagi efek ke ayamnya ya, ayamnya nggak dengerin karena dia di awal aja, mungkin di pagi hari gitu ya selama 8 jam. Tapi tetap ya menarik untuk diteliti ya, mudah-mudahan saja saya ada kesempatan lah menelitinya nanti, punya dana, punya anggaran ya, dikasih ada misalnya ya, mungkin akan menarik. Terima kasih.
Oke, thank you Prof.
Iya, sama-sama.
Thank you. Other questions? No. Okay. Since there's no other questions, we will end this question and answer session, and then we will soon have to end this conference sessions and move to the plenary presentation session. Before we end the session, I would like to say thank you for Professor Dr. Irwandi for the nice presentation and the nice discussion. And now I would like to call our head of committee, Mrs. Warsi, to symbolically hand over a certificate to our presenters as a form of appreciation from Mrs. Warsi, please.
Thank you. Hello everyone, my name is Warsi. I want to say thank you very much for all speakers for your time in this speaker. Thank you to Dr. Henry. Maybe Dr. Henry has left. Also, thank you, Dr. Phennapa. Special thanks to Prof. Irwandi Jaswir.
Thank you.
Prof. Irwandi Jaswir is my Ph.D. supervisor in International Islamic University Malaysia. Prof become your student, Prof.
Thank you.
Kindly please accept the certificate of speaker. Thank you.
Thank you very much.
The certificate to Dr. Phennapa, please accept this certificate, Doctor.
Thank you very much.
Next certificate. the certificate give to Prof. Irwandi Jaswir. Thanks for your participation.
Thank you, Madam Warsi. Thank you very much.
Welcome. Yeah, this certificate is given for Dr. Henry. Thank you for the attention.
Yeah, thank you very much for Mrs. Warsi. And we are really pleased to have the three of you here, and we look forward to having the next collaboration with you. Thank you very much, Dr. Phennapa and Dr. And Professor Irwandi. The next session is presentation session that will be held in English and in Bahasa Indonesia. You may stay if you want and choose in which room you are interested in to hear the presentation or want to see the poster presented by our presenters. Or if you have any other appointment, you can leave this event. So this is the end of the session. I would like to apologize if there's any mistake and shortcoming during my role as a moderator in the session. And I would like to say thank you for sticking with me and see you in other events. I will give the time to our Master of Ceremony, Mr. Ricky. Thank you.
Thank you for having me here.
Thank you very much.
Thank you, Mrs. Verda. Also, ladies and gentlemen, that was the explanation of knowledge delivered by our three great speakers. And of course, this will be additional knowledge that is useful for all of us. And before we step on the next agenda, ladies and gentlemen, we have another video about a vaccine campaign. And this video is made by Faculty of Pharmacy, Universitas Ahmad, Dahlan. So please enjoy the video. Thank you.
The highly positive cases of coronavirus in Indonesia has an impact in our daily lives, such as online learning, social life, working from home, and there are many new normal activities that still need to be done. We hope we can run the new normal activities optimally. As a society, we must be at the forefront for helping the health workers to fight COVID-19 and also comply with the government's advice to obey the health protocols. Let's we fight together the coronaviruses for our family, our friends, and our country. So please do our optimally job as a government, healthcare, student, and general public, we have to fight COVID-19 together.
According to the latest data, Indonesia reported 1.9 million confirmed cases of COVID-19, 54,000 deaths, and 1.7 million recovered cases from around 34 provinces. The prevalence of COVID-19 between January to June 2021 is still increasing. The government has been doing many prevention programs to protect the people by large-scale social restriction and implements a partial lockdown. In this kind of new normal era, to protect our health, we have to obey the protocol such as wearing a mask properly wherever we go, washing our hands with soap and running water regularly, maintaining the distance between 1.5 to 2 meters, staying at home unless for emergency matters, and avoiding the cold as much as possible. And then, don't forget to get the COVID-19 vaccine to save our life.
In this pandemic era, we have to keep our health by performing some protocols to prevent the COVID-19 infection, such as wearing a mask, physical distancing, and washing our hands regularly using soap and water or hand sanitizer. In another hand, one of the most effective ways to protect ourselves is to get our body vaccinated. Now, some of the COVID-19 vaccines are being developed in many countries. According to the latest data, the vaccination is quite effective to protect people from infection. Vaccine is a product that stimulates a person's immune system to produce immunity to specific disease, and eventually protecting people from the disease. Apparently, vaccine is the only way to completely solve the problem. We cannot fully control all 7.8 billion people to wear a mask every day and maintaining their distance to each other, right? So the only ultimate way is to make them immune from the disease by getting vaccinated. COVID-19 vaccines have been developed in many countries, and many of those vaccines have been approved by either World Health Organization or the FDA, and have been used by millions of people like Pfizer, Moderna, AstraZeneca, Johnson & Johnson, Sinovac, Sputnik V, and others. These vaccines are proven safe and have passed the minimum efficacy determined by the World Health Organization in many trials. And the progress of the development is giving us a hope to live a normal life again. There are currently 2 brands of vaccines which are being used popularly in Indonesia. These are AstraZeneca vaccine from United Kingdom and Sinovac vaccine from China. Both of them have been approved by Indonesian National Food and Drug Agency and Indonesian Islamic Halal Council and declared safe and halal. Indonesia is also currently developing COVID-19 vaccine called Merah Putih vaccine and predicted to be ready in the year 2022. Finally, let's pass this pandemic safely by performing the COVID-19 protocols and get vaccinated. If you love your family, if you love your community, start loving yourself and let's get our vaccine for the sake of human lives. Thank you.
However, some vaccines were reported to have a mirror side effect after getting vaccinated. You don't be afraid because some side effects are normally, such as feeling pain, weakness, or swelling in your arm when you got the shot. Or some people feel fever after getting the vaccine, and all of that may affect your ability to do daily activities, but that will go away in a few days. So don't be afraid, you can contact your doctor or healthcare provider if there are some serious side effects. And as we know, the critical point of vaccine are safe, effective, and halal. Let's take off our immune system by vaccinating and practicing health protocols.
Vaccine is safe and healthy. Vaccine is attenuated virus. It will fight virus if somebody exposed virus. So don't worry about getting vaccinated. Even though we have been vaccinated, we should to comply with our health protocol. Wearing a mask, washing hands, social distancing, stay at home. So fight COVID-19 together.
Dear all audience, as of December 2019, 27 cases of unknown pneumonia were reported in Wuhan, Hubei, China. Followed by January 11, 2020, WHO officially reported that it's caused by 2019 nCoV, and it's also declared as COVID-19 disease pandemic. This pandemic situation started as worldwide emergencies situation which burden whole world. To find a cure, vaccination is one of solutions to prevent the pandemic. We try to participate in the global campaign by article review research through the International Journal Publication Review. I hope this project will be our positive concern to the world pandemic situation. Vaccination is a government national program against the pandemic, and I've been vaccinated. Thank you.
Currently, COVID-19 and vaccination is the most popular issue in the worldwide. However, some communities still refuse the vaccination since they have doubt or confused about the halalness of the vaccine. Some vaccines have the halal certification from the halal authority and some of them does not. So as a Muslim, we can choose the halal one. The certification of halal vaccine considers some aspects, including the all of the material used for production, an example, what kind of cell used, what is the composition of the media for culturing the cell and virus, and also the material used for purification, and also and etc. Besides the material used, the audit of halal certification is also check the production process. The process must assure there is no contamination of pig and also its derivative. If during the production process there is some najis involved, the cleaning must be done as Sharia requirement. And finally, the halal certification also requires the implementation of the halal assurance system to assure the continuous production of halal vaccine. So, I invite you to have vaccination using the halal vaccine.
Protect yourself with vaccination.
Protect yourself and others by getting vaccination very soon.
Protect yourself and others by getting vaccine very soon.
Protect yourself by vaccination.
Protect yourself and others by getting vaccination soon.
Protect yourself and others by getting vaccination soon.
Okay, what a amazing video. Ladies and gentlemen, may we all always be healthy and still given the opportunity to spread the benefit to many people. We must win and keep the spirit in defeating the COVID-19 pandemic. Let's get vaccinated together. Yes, we can. Protect yourself and others by getting vaccinated. Ladies and gentlemen, next we will enter the presentation of the participants. The first session is a poster presentation in room 3.B or ruangan 3.B. This session will be divided into two sessions and the duration for this first session is 30 minutes for participants to immediately enter the break room on your Zoom screen. For those of you who want to watch the performance of the participants, you are also welcome to enter the break room that has been provided on your Zoom screen. And after this session, there will be a break, about 30-minute break. And if according to schedule, we will enter at 12:40 p.m. Thank you. Please, for participants to join the breakout rooms. You can see the breakout rooms in your Zoom screen. If you're using phone, you can tap the screen once, and then the break room icon will appear in your screen. I will inform you again. For those of you who want to watch the performance of the participant, you are also welcome to enter the break room that has been provided on your Zoom screen. You can click the breakout room in your menu on your Zoom. Okay, for the moderator in the designated room, I will give you the time. The time is yours.
Faculty of Pharmacy of Universitas Dahlan was established in 1996 with one study program, which becomes the oldest faculty of pharmacy among Muhammadiyah and Ashiah colleges. Since its establishment, the Faculty of Pharmacy has one study program, namely the Pharmacy Study Program with undergraduate level. In 2001, based on a Rector's decree, UAD Faculty of Pharmacy started to open a new study program, namely the Pharmacist Professional Study Program. Along with the education development, in 2015, UAD established a postgraduate program in pharmacy.
Faculty of Pharmacy, Universitas Dahlan is a private Muslim faculty owned by the biggest Muslim organization in the world, namely Muhammadiyah. We already granted A for the National Health Accreditation. Thus, we commit to raise the higher quality in ASEAN level. The integration of moral and intellectual integrity in the higher education system can support the graduates for passing the national examination. The research and publication of the lectures are conducted based on the national and international collaboration. We also serve the community using the pharmaceutical products and our knowledge.
The UAD Pharmacy Study Program commits to the faculty vision of becoming a leading pharmacy higher education institution that is internationally recognized based on Islamic values. Not only that, UAD Pharmacy Study Program implements learning that instills and improves Islamic values in all aspects of campus life. Every year, more than 1,000 students from various provinces in Indonesia study together in the UAD Pharmacy Study Program. UAD Pharmacy Study Program applied curriculum for 4 years of study. It has represented all aspects of the competencies achievements for graduates. UAD Pharmacy Study Program has about 50 teaching staffs with doctor and professor qualification. There are 30 education staffs with appropriate competencies to strengthen the learning, research process, and community service. The institution evaluated their staff annually to improve professional development, monitoring, and evaluation. UAD Pharmacy Study Program built a strong partnership with various parties by involving alumni and stakeholders for the development of higher education. To support the activities of the three Dharma perguruan tinggi, namely learning, research, and community service, UAD Pharmacy Study Program has good, comfortable, and integrated facilities. The UAD Pharmacy Study Program has a laboratory with well-standardized equipment for teaching and learning process. UAD Pharmacy Study Program attempts to improve cooperation network with various parties, including the government, pharmaceutical industry, local higher education, and abroad.
The students' and lecturers' achievements are already reached the national and international level as well. And finally, join with us, get the higher quality, and reach the better moral and intellectual integrity.
With competence that supports various abilities, both theories and soft skills, graduates of UAD Pharmacy Study Program have spread in various areas including in hospitals, government institutions, academics, pharmaceutical industry, pharmacists, and entrepreneurs. The Faculty of Pharmacy of Universitas Dahlan was established in 1996 with one study program, which becomes the oldest faculty of pharmacy among Muhammadiyah and Ashiah colleges. Since its establishment, the Faculty of Pharmacy has one study program, namely the Pharmacy Study Program with undergraduate level. In 2001, based on a Rector's decree, UAD Faculty of Pharmacy started to open a new study program, namely the Pharmacist Professional Study Program. Along with the education development, in 2015, UAD established a postgraduate program in pharmacy.
Faculty of Pharmacy, Universitas Dahlan is a private Muslim faculty owned by the biggest Muslim organization in the world, namely Muhammadiyah. We already granted A for the National Health Accreditation. Thus, we commit to raise the higher quality in ASEAN level. The integration of moral and intellectual integrity in the higher education system can support the graduates for passing the national examination. The research and publication of the lectures are conducted based on the national and international collaboration. We also serve the community using the pharmaceutical products and our knowledge.
The UAD Pharmacy Study Program commits to the faculty vision of becoming a leading pharmacy higher education institution that is internationally recognized based on Islamic values. Not only that, UAD Pharmacy Study Program implements learning that instills and improves Islamic values in all aspects of campus life. Every year, more than 1,000 students from various provinces in Indonesia study together in the UAD Pharmacy Study Program. UAD Pharmacy Study Program applied curriculum for 4 years of study. It has represented all aspects of the competencies achievements for graduates. UAD Pharmacy Study Program has about 50 teaching staffs with doctor and professor qualification. There are 30 education staffs with appropriate competencies to strengthen the learning, research process, and community service. The institution evaluated their staff annually to improve professional development, monitoring, and evaluation. UAD Pharmacy Study Program built a strong partnership with various parties by involving alumni and stakeholders for the development of higher education. To support the activities of the three Dharma perguruan tinggi, namely learning, research, and community service, UAD Pharmacy Study Program has good, comfortable, and integrated facilities. The UAD Pharmacy Study Program has a laboratory with well-standardized equipment for teaching and learning process. UAD Pharmacy Study Program attempts to improve cooperation network with various parties, including the government, pharmaceutical industry, local higher education, and abroad.
The students' and lecturers' achievements are already reached the national and international level as well. And finally, join with us, get the higher quality, and reach the better moral and intellectual integrity.
With competence that supports various abilities, both theories and soft skills, graduates of UAD Pharmacy Study Program have spread in various areas including in hospitals, government institutions, academics, pharmaceutical industry, pharmacists, and entrepreneurs.
Negatif mengenai COVID-19. Oleh sebab itu, apoteker-apoteker di sini lebih berhati-hati dalam mengevaluasi sumber informasi mengenai COVID-19 sehingga dapat membantu keresahan masyarakat dan dapat meminimalisir penyebaran COVID-19. Untuk kesimpulan dalam penelitian ini, lebih dari sebagian apoteker komunitas atau mayoritas apoteker komunitas memiliki pengetahuan yang baik, dan ada beberapa apoteker yang memiliki pengetahuan yang kurang dikarenakan sumber informasi yang didapat kurang tepat. Selain itu juga, apoteker di sini juga memiliki persepsi dan sikap yang positif mengenai COVID-19. Sekian presentasi dari saya mengucapkan terima kasih kepada dosen dan seluruh staf program farmasi Universitas Ahmad Dahlan yang sudah membantu penelitian saya untuk menyelesaikan penelitian ini. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Terima kasih Saudara Wiwin Nopita Putri. Selanjutnya kami persilahkan pada audiens yang hadir pada kesempatan kali ini. Apabila ada yang ingin diklarifikasikan, ingin didiskusikan dengan Mbak Wiwin Nopita Putri, bisa langsung saja, silahkan dibuka mikrofonnya dan diajukan pertanyaannya secara langsung. Atau bisa juga memberikan emot tangan melambai pada gambar di dalam ruangan Zoom meeting ini, kami akan menyapa Bapak dan Ibu. Sepertinya belum ada. Jika begitu, saya ingin bertanya sesuatu kepada Saudara Wiwin Nopita Putri. Kenapa tertarik melakukan penilaian mengenai tingkat pengetahuan, persepsi, dan sikap apoteker, dan dikhususkan pada apoteker komunitas? Kenapa tidak ingin melihat apoteker di komunitas rumah sakit, apoteker di komunitas industri, atau yang lain? Silakan, Saudara atau Mbak Wiwin.
Karena di sini saya, di sini saya tertarik meneliti penelitian ini karena semakin banyak yang terinfeksi COVID-19 dan apoteker komunitas ini yang sangat rentan, Bu, terinfeksi karena kontak langsung terhadap pasien. Jadi apabila pasien merasa gejala seperti COVID-19, jadi terlebih dahulu akan mendatangi apotek, yang itu ada apoteker komunitasnya. sehingga sangat rentan terinfeksi COVID-19. Seperti itu, Bu.
Oh, hanya karena sangat rentan ya. Tidak ada pertimbangan lain apakah sebagai garda terdepan atau sebagai agent of change-nya atau tidak. Jadi hanya mempertimbangkan apoteker lebih rentan terpapar, begitukah yang ada di komunitas? Bapak Ibu yang lain, saudara-saudara sekalian yang ada di ruangan ini, ada yang ingin ditambahkan? Sembari mohon untuk mempersiapkan diri Bapak atau Ibu Saudara Maidina Putri Ananda, karena saya belum melihat Saudara Widya Akmasari hadir di dalam ruangan ini. Mohon untuk mempersiapkan diri Saudara Maidina Putri Ananda tentang tingkat pengetahuan dan perilaku masyarakat di apotek Salaman tentang cara menyimpan dan membuang obat. Baik. Terima kasih Saudara Wiwin Nopita Putri. Ya, semoga sehat selalu dan bahagia selalu. Silahkan terus menyimak presentasi selanjutnya. Maidina Putri, silahkan kami persilahkan untuk share screen atau berkenan kami putarkan videonya.
Baik Ibu, saya akan share screen saja.
Share screen, berarti secara langsung. Baik, bisa dipersiapkan terlebih dahulu 7 menit ya, Saudara Maidina, untuk presentasinya. Ya. Silakan Mbak Maidina.
Baik Ibu, sebentar sedang loading. Apakah layar saya sudah terlihat?
Layarnya terlihat, posternya yang belum.
Oh ya, mohon ditunggu sebentar Bu, karena sudah muncul di tempat saya.
Iya.
Apakah
Atau
poster saya sudah
bisa
terlihat?
membantu menyiapkan backup-nya? Ya. Teman-teman dari panitia bisa membantu siapkan backup-nya. Waktunya sudah running 50 detik, belum muncul. Saya reset terlebih dahulu.
Apakah layar saya sudah terlihat, Ibu?
Layarnya sudah, Mbak Maidina, tetapi
ya?
posternya belum.
Oh, ya. Saya ulangi lagi ya, Bu.
Nah, sudah Mbak Maidina. Kita mulai sekarang ya, saya riset dulu. Silahkan Mbak Maidina.
Baik, terima kasih Ibu atas kesempatannya. Perkenalkan, nama saya Maidina Putri Ananda. Di sini saya akan menyampaikan mengenai tingkat pengetahuan dan perilaku masyarakat di apotek Salaman tentang cara menyimpan dan membuang obat. Latar belakangnya, di sini saya mengambil dari banyaknya masyarakat yang membeli obat di apotek, terutama di masa pandemi COVID-19 seperti ini. Nah, masyarakat membeli obat, kemudian menyimpan obat-obatan tersebut untuk menjadi salah satu pertolongan pertama ketika mengalami suatu gejala seperti itu. Kemudian banyaknya masyarakat yang menyimpan dan membuang obat di rumah itu menimbulkan suatu rasa penasaran saya, bagaimanakah tingkat pengetahuan dan perilaku menyimpan obat dan membuang obat masyarakat. Metode pengambilan data saya yaitu menggunakan observational dengan pendekatan cross-sectional. Di sini, yang pertama kali saya lakukan adalah menentukan calon responden, yaitu masyarakat yang datang membeli obat maupun tidak membeli obat di apotek Salaman. Kemudian, setelah mendapatkan datanya, di sini saya meminta kesediaan kepada responden untuk mengisi informed consent atau persetujuan apakah mereka mau atau menyetujui untuk dijadikan sebagai responden. Kemudian setelah setuju, masyarakat ataupun responden dipersilahkan untuk mengisi kuesioner sesuai dengan peraturannya. Setelah itu, apabila kuesioner sudah diisi, kemudian akan saya analisis apakah kuesionernya sudah diisi semua atau belum. Setelah itu akan dilaksanakan validasi expert atau expert judgment yang dilakukan oleh beberapa orang ahli. Di sini saya mengambil dari dosen psikologi, dan juga farmasi. Setelah itu, saya akan mengecek kesesuaian kuesioner dengan cara memvalidasi dan mengecek uji reliabilitas. Kemudian setelah kuesionernya dianggap valid dan reliable, saya akan mengolah data. Nah, berikut adalah hasil penelitian yang telah saya lakukan. Di sini saya mendapatkan tingkat pengetahuan masyarakat di apotek Salaman tentang cara menyimpan obat itu masih tergolong kurang. Dan juga tentang cara membuat obat, cara membuang obat masih tergolong kurang. Bisa kita lihat pada tabel yang sudah saya tampilkan, dari 100 responden, ternyata hanya 8 orang yang mengisi kuesioner dengan baik, kemudian 41 orang mengisi kuesioner dengan cukup, dan 51 orang mengisi kuesioner dengan kurang. Kemudian untuk membuang obat, dari total 100 responden, hanya 8 orang mengisi kuesioner dengan baik, kemudian 16 orang mengisi kuesioner dengan cukup, dan 76 orang mengisi kuesioner dengan kurang. Kemudian untuk tingkat perilaku masyarakat di apotek Salaman tentang cara menyimpan dan membuang obat, untuk di bagian menyimpan obat, dari 100 orang responden, hanya 2 orang yang mengisi kuesioner dengan baik, kemudian 11 orang mengisi kuesioner dengan cukup, dan 87 orang mengisi kuesioner dengan kurang. Kemudian untuk tingkat perilaku masyarakat di apotek Salaman tentang cara membuang obat, dari 100 orang responden, hanya 1 orang responden yang mengisi kuesioner dengan baik, 44 orang mengisi kuesioner dengan cukup, dan 55 orang mengisi kuesioner dengan kurang. Di sini saya menggunakan kuesioner secara langsung, di mana kuesioner tersebut berisikan 24 soal. Kemudian, untuk kesimpulannya, tingkat pengetahuan masyarakat di apotek Salaman tentang cara menyimpan obat tergolong kurang, tingkat pengetahuan masyarakat di apotek Salaman tentang cara membuang obat tergolong kurang, tingkat perilaku masyarakat di apotek Salaman tentang cara menyimpan obat tergolong kurang, dan tingkat perilaku masyarakat di apotek Salaman tentang cara membuat obat tergolong kurang. Sekian presentasi yang dapat saya sampaikan. Terima kasih atas waktu yang telah diberikan kepada saya. Saya kembalikan lagi ke Ibu Ana.
Ya, terima kasih Saudara Meidina. Bapak, Ibu, Saudara-saudara sekalian, ada yang ingin diklarifikasikan untuk Saudara Meidina? Meidina, ada pertanyaan yang di kolom chat, Mbak Meidina atau Saudara Meidina, gitu.
Iya.
Untuk aktivitas pengambilan data penelitian yang Anda lakukan menggunakan alat ukur berupa kuesioner, ya.
Betul, Ibu.
Nah, kuesioner yang digunakan, apakah menggunakan kuesioner yang sudah ada pada penelitian-penelitian sebelumnya dengan asumsi merujuk pada penelitian-penelitian yang sebelumnya, atau Saudara Meidina mengembangkan kuesioner baru? Kalau mengembangkan kuesioner atau alat ukur yang baru, bagaimana proses dan cara pengembangannya? Silahkan.
Baik, terima kasih Ibu Ana atas pertanyaannya, dan juga dari Saudari
Mahacita.
Oh iya, Mahacita, terima kasih atas pertanyaannya. Di sini saya mengembangkan kuesioner terdahulu. Jadi yang sebelumnya sudah ada, kemudian saya kembangkan dan saya sesuaikan dengan kondisi masyarakat di apotek Salaman, gitu.
Bagaimana proses pengembangannya?
Ya, di sini saya menganalisis terlebih dahulu untuk respondennya. Jadi saya sebelumnya sudah bekerja terlebih dahulu, sebetulnya saya berada di apotek Salaman. Kemudian saya menganalisis bagaimana perilaku atau tingkah laku masyarakat di Salaman. Kemudian dari situ saya bisa mengembangkan melalui kuesioner dari yang terdahulu. Dari yang terdahulu itu saya mengambil dari beberapa sumber, dari ada yang dari masyarakat, ada pula yang di lingkup mahasiswa atau universitas. Jadi di situ saya kolaborasikan, kemudian saya sesuaikan dengan keadaan masyarakat yang ada di Salaman, kemudian jadilah kuesioner yang saya buat tersebut, Ibu.
Berarti kuesionernya adalah kuesioner pengembangan, bukan kuesioner baru, dan pengembangan itu mengelaborasikan dari beberapa penelitian, dari beberapa kuesioner, dari beberapa alat ukur yang pernah digunakan, kemudian tahapan penyesuaiannya dengan penilaian ahli, mungkin dari ahli bahasa, ahli konten media, atau yang lain untuk dilakukan validasi. Begitu ya Mbak Meidina ya, maksud pertanyaan dari Saudara Mahacita. Nah, selanjutnya mohon untuk mempersiapkan diri Saudara, ini Bapak atau Ibu Rahel Pasafika Abdillah, masih seputar pengetahuan, masih seputar sikap, praktek, dan kecemasan tentang vaksinasi COVID-19 antara masyarakat ya, di antara masyarakat. Baik, terima kasih Saudara Meidina Putri. Semoga lancar untuk kegiatan dan aktivitas selanjutnya. Salam sehat dan bahagia. Terima kasih.
Terima kasih Ibu Ana.
Selanjutnya kita sapa. Terima kasih Saudara Rahel Pasafika Abdillah sudah hadir. Saya belum mendengar suaranya, tapi kita cek dulu. Bapak, Ibu, saudara-saudara sekalian yang berada di ruangan ini, di kolom chat sudah terdapat link presensi kehadiran. Mohon jangan dilupakan untuk diisi dan memastikan bahwa nama, gelar, dan semua yang tertulis dalam link tersebut sudah benar untuk penulisan sertifikat Bapak, Ibu sekalian. Saudara Rahel, mikrofonnya silakan dibuka terlebih dahulu, di-unmute dulu. Iya, kita pastikan.
Maaf Bu, ini sebentar.
Ya. Saudara Rahel kemarin mengirimkan video presentasi kepada panitia?
Iya, Bu.
Mengirimkan? Kita pastikan dulu untuk host atau co-host di ruang breakout room ini, masih menyimpan backup dari presentasi atau video presentasi Saudara Rahel. Coba di stop share dulu, Kak Rahel, Mbak Rahel.
Sebentar Bu, ini error.
Ngeheng ya, karena tidak bisa diapa-apakan lagi kalau sudah seperti ini.
Untuk di panitia
tidak bisa di stop share?
Nggak bisa Bu, error.
Kalau ini tidak bisa di stop share, kami juga kesulitan untuk menampilkan video presentasi dari Mbak Rahel. Kami mau ganti, iya benar. Kami mau pindah ke Mbak Nabila, Saudara Nabila Dwi Febrianti juga agak kesulitan ini.
Ya.
Bagaimana ibu-ibu dan saudara-saudara yang ada di ruangan ini atau teman-teman panitia? Kita izinkan Mbak Rahel. Sudah. Iya. Mungkin dari panitia bisa membantu video presentasinya Saudara Rahel Pasafika Abdillah.
Maaf Ibu, untuk Mbak Rahel, videonya tidak dikirimkan ke panitia Ibu. Jadi
gimana? Saya tidak mendengar suaranya, Bu Prita. Kecil banget.
Oh Bu, sudah terdengar Bu suara saya?
Lumayan.
Oh ya, untuk Mbak Rahel tidak mengirimkan video ke panitia Ibu.
Posternya juga tidak dikirim ke panitia?
Tidak dikirimkan Ibu.
Oh begitu.
Jadi
baik. Coba kita cek dulu sebentar. Mbak Rahel, mohon bantuannya bisa mengirimkan soft copy posternya ke panitia. Kita undur Mbak Rahel setelah isoma ya, kita menuju Mbak Nabila dulu, karena kita sekarang sudah pukul 12.00, kita harus mengakhiri sesi yang pertama ini pukul 12:10, jadi kita menuju Mbak Nabila Dwi Febrianti terlebih dahulu. Mbak Nabila, kita sapa dulu Mbak Nabila Dwi Febrianti. Ya, Mbak Nabila Dwi Febrianti bisa share screen dulu? Oke, Bu. Bapak, Ibu, saudara-saudara sekalian, kami mohon bantuannya agar Bapak dan Ibu berkenan mengirimkan soft copy posternya terlebih dahulu kepada kami, untuk menayangkan atau menampilkan poster. Kami juga mengkhawatirkan apabila ada kendala teknis di lokasi Bapak dan Ibu, kami bisa membantu dengan backup. Masih ada waktu 30 menit ke depan, Bapak dan Ibu bisa membantu kami untuk mengirimkan file posternya kepada panitia, sehingga sifatnya hanya backup ya Bapak dan Ibu. Baik, Mbak Nabila Dwi Febrianti sudah muncul posternya. Silakan dipresentasikan mengenai pengetahuan dan kepatuhan minum obat bagi pasien tuberkulosis paru di Puskesmas Kota Yogyakarta. Silakan 10 menit ya, 7 menit presentasi, dilanjutkan dengan tanya jawab.
Terima kasih Bu atas waktunya. Perkenalkan, saya Nabila Dwi Febrianti. Di sini saya akan mempresentasikan tentang pengetahuan dan kepatuhan minum obat pasien tuberkulosis paru di Puskesmas Kota Yogyakarta. Di sini saya dibantu oleh Bu Diah Aryani Pervitasari. Baik, selanjutnya saya akan memperkenalkan tuberkulosis merupakan penyakit infeksi dengan beban tertinggi di dunia. Pada tahun 2019, Indonesia merupakan negara dengan peringkat ketiga beban tertinggi akibat penyakit TB di dunia. Pemerintah Indonesia telah mengusahakan berbagai upaya untuk mencapai target ditemukannya kasus baru TB dan keberhasilan penyembuhan penyakit tuberkulosis. Hal yang telah dilakukan pemerintah antara lain program pendampingan minum obat atau PMO dan program temukan TBC obati sampai sembuh. Salah satu faktor yang menimbulkan kegagalan pengobatan TB adalah rendahnya kepatuhan minum obat dan pengetahuan pasien TB yang kurang mengenai penyakit TB dan pengobatannya. Tujuan penelitian yang saya lakukan yaitu untuk mengetahui kepatuhan dan pengetahuan pasien TB dalam menjalankan proses penyembuhan TB dengan fokus kepada efek samping hepatotoksisitas yang dialami pasien selama minum obat anti-tuberkulosis. Metode penelitian yang saya lakukan yaitu menggunakan metode cross-seksional yang berfokus pada pengambilan data saat pasien datang berobat secara rutin ke puskesmas. Penelitian ini dilakukan di 2 puskesmas di Kota Yogyakarta. Di kriteria inklusinya yaitu pasien TB paru dewasa yang berobat secara rutin ke puskesmas dalam kurun waktu penelitian. Pasien diminta untuk mengisi kuesioner MARS dan kuesioner pengetahuan. Pengisian kuesioner dilakukan secara online. Dan untuk alat dan bahannya yang digunakan dalam penelitian ini berupa kuesioner kepatuhan yang diukur menggunakan kuesioner MARS yang berisi data pasien dan pertanyaan tentang kepatuhan terhadap penggunaan obat. Kepatuhan diketahui dengan hasil jawaban responden. Kemudian, ada kuesioner pengetahuan yang berisi pertanyaan tentang efek samping obat yang mungkin dirasakan pasien. Hasil penelitian yang saya lakukan, yaitu ada sejumlah 28 pasien TB paru dewasa bersedia untuk ikut serta dalam penelitian ini. Rata usia pasien adalah 32 tahun, dan untuk rata berat badannya 46,69 kg, dan untuk rata tinggi badannya adalah 160,53 cm. Dan ada sejumlah 46,4% pasiennya adalah perempuan. Rata skor kepatuhan pasien adalah 24,70 dengan 92,85% pasien patuh terhadap minum obat. Sejumlah 21,42% pasien tidak mengetahui bahwa obat yang mereka konsumsi dapat menyembuhkan penyakit mereka. Dan 42,85% menyatakan bahwa mereka hanya minum obat selama kurang dari 6 bulan. Sejumlah 14,28% pasien tidak mengetahui tentang efek samping obat tuberkulosis dan sejumlah 67,85% mengalami efek samping obat, di mana 50% dari pasien TB mengalami gangguan hati, mual, dan muntah. Kesimpulan dari penelitian saya lakukan adalah menunjukkan bahwa kepatuhan pasien TB sudah sangat baik, meskipun sebagian besar pasien mengalami efek samping obat dan 50% diantaranya mengalami gangguan hati. Demikian presentasi yang saya lakukan, kurang lebihnya. Baik.
Oke, terima kasih Saudara Nabila Dwi Febrianti ya. Iya. Kenapa menyukai penelitian tuberkulosis? Kita simpan dulu jawabannya, kita beri kesempatan terlebih dahulu untuk Bapak Ibu, saudara-saudara sekalian yang hadir di dalam ruangan ini untuk diskusi, klarifikasi, dan beberapa hal yang perlu disampaikan kepada Saudara Nabila. Silakan Ibu dan Bapak. Baik. Saudara Nabila, pengetahuan dan kepatuhan, karena penderita TBC banyak yang putus obat atau gagal terapi karena permasalahan kepatuhan minum obat. Sampai sejauh mana dipastikan mereka tidak patuh minum obat itu karena pengetahuannya, bukan karena tingkat ekonominya, bukan karena faktor-faktor yang lain?
Bisa diulangin sekali lagi, Bu?
Iya, sejauh mana melihat hubungan pengetahuan dan kepatuhan untuk pasien dengan TB paru?
Baik, untuk pengetahuan dilihat sejauh.
Apakah semakin pintar dia semakin patuh? Apakah semakin tinggi pendidikannya dia semakin patuh atau bagaimana?
Tidak, Bu. Jadi,
apakah semakin baik kepatuhan, semakin baik pengetahuan orang, pengetahuannya semakin baik, semakin tinggi atau semakin luas pengetahuannya, kepatuhan minum obatnya semakin baik, atau justru sebaliknya, atau bagaimana?
Tidak, Bu. Jadi tingkat pengetahuan dilihat dari tingkat pengetahuan pasien tentang tuberkulosis, yaitu dilihat dari seberapa
dari seberapa apanya, Mbak Nabila?
tingkat pengetahuan pasien dalam mengetahui penyakit tuberkulosis itu diketahui seberapa tahu tentang penyakit tersebut dalam obat anti tuberkulosisnya itu mencegah penyakit yang dialami pasien.
Oh, semakin tahu dia semakin patuh atau tidak?
Iya, Bu.
Semakin tahu semakin patuh. Iya, berarti kan cukup dijawab. Semakin tinggi pengetahuannya dia semakin patuh. Ya, ya. Oke. Terima kasih, Saudara Nabila Dwi Febrianti. Semoga selalu sehat, selalu dimudahkan untuk setiap aktivitas selanjutnya. Dan Bapak Ibu, saudara-saudara sekalian, kita sudah masuk pukul 12:10. Ya, kita akan akhiri terlebih dahulu untuk sesi yang pertama, presentasi poster bidang Social Behavior and Administration. Kita akan lanjutkan lagi pada pukul 12:40. Mohon mempersiapkan diri dan bisa hadir tepat waktu untuk presenter yang pertama pada pukul 12.40 nanti, kami persilahkan kepada Saudara Sohifa Hafida, dilanjutkan dengan Saudara Rahel Pasa Fica Abdillah. Semoga device-nya sudah mendukung dan bisa lebih pas secara teknis untuk dipresentasikan nanti poster-posternya. Terima kasih. Kita akhiri sesi yang pertama dengan hamdalah. Alhamdulillahirabbil alamin. Silakan dilanjutkan untuk menunaikan ibadah salat zuhur dan mempersiapkan logistik siangnya agar tetap prima, tetap naik imunitasnya. Kita lanjutkan nanti. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Teman-teman, Bapak Ibu, Saudara-saudara sekalian boleh meninggalkan ruangan ini, nanti dapat bergabung lagi dan akan kita setting untuk breakout room-nya. Jika ingin tetap berada di ruangan ini, tetap kami persilahkan. Mana yang nyaman untuk Bapak Ibu dan saudara-saudara sekalian, kami persilahkan.
Dilakukan pembuatan bahan-bahan uji antiinflamasi. Sediaan pembanding sebagai bahan pembanding atau kontrol positifnya itu adalah hidrokortison dari kimia farma yang dibuat balsam stick dengan konsentrasi minimal yaitu 2,5%. Kemudian dilakukan pembuatan larutan karagenan. Setelah itu pengelompokan hewan uji. Kemudian perlakuan hewan uji, setelah itu dilakukan evaluasi sediaan balsam stick. Setelah evaluasi sediaan balsam stick dilakukan pengelompokan dan perlakuan hewan uji, kemudian dilakukan analisis data. Analisis data menggunakan karena tidak memenuhi persyaratan ANOVA, maka dilanjutkan dengan uji Kruskal-Wallis. Kemudian dilihat pengaruh perlakuannya, dilakukan dengan uji Mann-Whitney. Hasil penelitiannya, mudah-mudahan kelihatan, yang Formula 1. Formula 1 ini lembut, kemudian kering, berminyak, khas dari balsam. Formula 1 ini merupakan kontrol positif dengan menggunakan hidrokortison 2,5%. Kemudian kalau Formula 2 hanya balsam stick saja. Kemudian Formula 3 dia menggunakan daun kersen 2,5%, Formula 4 5%. Kemudian warnanya yang formula keempat itu, yang formula 1, 2, 3 khas daun kersen, kemudian bau khas kersen, kemudian uji homogenitas homogen, uji pH seperti yang ada di sini, kemudian hasil persen rata-rata UDEM. Pengamatan dilakukan menggunakan alat plastimometer dengan melihat volume UDEM pada kaki tikus yang diinduksi keragenan. Nah, dari hasilnya, balsam stick dengan konsentrasi 5% dan 10% itu dia memiliki daya hambat lebih dari 50%. Dengan nilai yang 5% 70,27%, yang 10% 95,83%. Kemudian konsentrasi 2,5%, daya hambatnya 44,44%. Kemudian dilakukan uji Kruskal-Wallis, terjadi perbedaan bermakna. Kemudian dilanjutkan uji Mann-Whitney, Formula 2 dan Formula 3 itu berbeda bermakna dengan kontrol negatif dan sebanding dengan kontrol positif. Menurut Sarimana, khasiat antiradang yang dimiliki daun kersen ini karena daun ini mengandung flavonoid. Nah, flavonoid itu sendiri memiliki struktur cincin benzopyron yang dapat berikatan dengan enzim siklooksigenase dan lipoksigenase sehingga dapat menghambat pembentukan asam arakidonat berubah menjadi prostaglandin dan leukotrien yang berperan dalam proses inflamasi. Sehingga ketika pembentukan asam arakidonat dihambat dengan mengikat enzim siklooksigenase, maka inflamasi dapat digagalkan. Kemudian karena dia menggunakan terdapat fase minyak dan surfaktan sehingga dapat berdifusi langsung. Misalkan dia mekanismenya masuk melalui jalur transepidermal, kemudian obat berpartisi ke dalam stratum corneum. Ketika sudah masuk ke stratum corneum, dapat berdifusi melintasi matriks protein lipid dari SC ke lapisan epidermis. Kemudian kalau transpedigial, obat dapat berpartisi ke dalam sebum. Kemudian dia dapat berdifusi melintasi lipid melalui pori sebasea ke lapisan epidermis. Setelah dapat melalui epidermis, kemudian dapat berdifusi kembali melintasi massa seluler dan massa fibrous atau dermis atas. Massa fibrous atau dermis atas itu tempat di mana obat ini berikatan dengan targetnya sehingga dapat berikatan dengan enzim siklooksigenase sehingga dapat memberikan efek anti-inflamasi. Demikian yang dapat saya sampaikan. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Terima kasih Bu Fitri. Kami persilahkan kepada peserta untuk menanyakan terkait presentasi yang telah disampaikan. Kemudian kami infokan bahwasannya ini presentasi sebelum istirahat terakhir. Nanti kami lanjutkan di jam 12.45. Jadi untuk itu setelah ini kami mohon Ibu mau tetap join atau live sementara kami persilahkan untuk isoma.
Baik, karena sudah azan juga ya Pak.
Iya, silahkan ada yang mau menanyakan? Coba saya ini Bu, coba saya menanyakan sedikit mengenai apa yang dipaparkan Bu Fitri ya tadi Bu ya. Baik, Bu Fitri tadi cukup menarik ya presentasinya. Studinya ini secara in vivo ya Bu ya?
Betul Pak, secara in vivo.
Apakah studi in vitronya juga sudah ada, Ibu?
Studi in vitronya kebetulan belum ada, jadi kami langsung mencoba secara in vivo Pak pada tikus jantan.
Begitu ya. Begitu. Ya, tadi disimpulkan bahwasannya tadi ada senyawa flavonoid ya, yang dimana itu sangat berperan dalam antiinflamasi. Nah, di sini mungkin sedikit saya tanyakan, apakah sudah ada mungkin reference yang ditemukan bahwasannya memang jenis golongan flavonoid tersebut yang secara in vitro secara masif memang dia sebagai anti. Ya, baik, kalau tidak ada, kami cukupkan sekian. Kami mohon nanti para peserta bisa hadir kembali di room Zoom yang room 2 untuk sesi poster presentation. Untuk itu kami tutup sementara, kurang lebihnya mohon maaf. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Terima kasih Bu Fitri.
This meeting is being recorded.
The Faculty of Pharmacy of Universitas Ahmad Dahlan was established in 1996 with one study program, which becomes the oldest faculty of pharmacy among Muhammadiyah and Ashiah colleges. Since its establishment, the Faculty of Pharmacy has one study program, namely the Pharmacy Study Program with undergraduate level. In 2001, based on a rector's decree, UAD Faculty of Pharmacy started to open a new study program, namely the Pharmacist Professional Study Program. Along with the education development, in 2015, UAD established a postgraduate program in pharmacy.
Faculty of Pharmacy, Universitas Ahmad Dahlan is a private Muslim faculty owned by the biggest Muslim organization in the world, namely Muhammadiyah. We already granted A for the National Health Accreditation. Thus, we commit to raise the higher quality in ASEAN level. The integration of moral and intellectual integrity in the higher education system can support the graduates for passing the national examination. The research and publication of the lectures are conducted based on the national and international collaboration. We also serve the community using the pharmaceutical products and our knowledge.
The UAD Pharmacy Study Program commits to the faculty vision of becoming a leading pharmacy higher education institution that is internationally recognized based on Islamic values. Not only that, UAD Pharmacy Study Program implements learning that instills and improves Islamic values in all aspects of campus life. Every year, more than 1,000 students from various provinces in Indonesia study together in the UAD Pharmacy Study Program. UAD Pharmacy Study Program applied curriculum for 4 years of study. It has represented all aspects of the competencies achievement for graduates. UAD Pharmacy Study Program has about 50 teaching staffs with doctor and professor qualification. There are 30 education staffs with appropriate competencies to strengthen the learning, research process, and community service. The institution evaluated their staff annually to improve professional development, monitoring, and evaluation. UAD Pharmacy Study Program built a strong partnership with various parties by involving alumni and stakeholders for the development of higher education. To support the activities of the three Dharma perguruan tinggi, namely learning, research, and community service, UAD Pharmacy Study Program has good, comfortable, and integrated facilities. The UAD Pharmacy Study Program has a laboratory with well-standardized equipment for teaching and learning process. UAD Pharmacy Study Program attempts to improve cooperation network with various parties, including the government, pharmaceutical industry, local higher education, and abroad.
The students' and lecturers' achievements are already reached the national and international level as well. And finally, join with us, get the higher quality, and reach the better moral and intellectual integrity.
With competence that supports various abilities, both theories and soft skills, graduates of UAD Pharmacy Study Program have spread in various areas including in hospitals, government institutions, academics, pharmaceutical industry, pharmacists, and entrepreneurs. The Faculty of Pharmacy of Universitas Ahmad Dahlan was established in 1996 with one study program, which becomes the oldest faculty of pharmacy among Muhammadiyah and Ashiah colleges. Since its establishment, the Faculty of Pharmacy has one study program, namely the Pharmacy Study Program with undergraduate level. In 2001, based on a rector's decree, UAD Faculty of Pharmacy started to open a new study program, namely the Pharmacist Professional Study Program. Along with the education development, in 2015, UAD established a postgraduate program in pharmacy.
Faculty of Pharmacy, Universitas Ahmad Dahlan is a private Muslim faculty owned by the biggest Muslim organization in the world, namely Muhammadiyah. We already granted A for the National Health Accreditation. Thus, we commit to raise the higher quality in ASEAN level. The integration of moral and intellectual integrity in the higher education system can support the graduates for passing the national examination. The research and publication of the lectures are conducted based on the national and international collaboration. We also serve the community using the pharmaceutical products and our knowledge.
The UAD Pharmacy Study Program commits to the faculty vision of becoming a leading pharmacy higher education institution that is internationally recognized based on Islamic values. Not only that, UAD Pharmacy Study Program implements learning that instills and improves Islamic values in all aspects of campus life. Every year, more than 1,000 students from various provinces in Indonesia study together in the UAD Pharmacy Study Program. UAD Pharmacy Study Program applied curriculum for 4 years of study. It has represented all aspects of the competencies achievement for graduates. UAD Pharmacy Study Program has about 50 teaching staffs with doctor and professor qualification. There are 30 education staffs with appropriate competencies to strengthen the learning, research process, and community service. The institution evaluated their staff annually to improve professional development, monitoring, and evaluation. UAD Pharmacy Study Program built a strong partnership with various parties by involving alumni and stakeholders for the development of higher education. To support the activities of the three Dharma perguruan tinggi, namely learning, research, and community service, UAD Pharmacy Study Program has good, comfortable, and integrated facilities. The UAD Pharmacy Study Program has a laboratory with well-standardized equipment for teaching and learning process. UAD Pharmacy Study Program attempts to improve cooperation network with various parties, including the government, pharmaceutical industry, local higher education, and abroad.
The students' and lecturers' achievements are already reached the national and international level as well. And finally, join with us, get the higher quality, and reach the better moral and intellectual integrity.
With competence that supports various abilities, both theories and soft skills, graduates of UAD Pharmacy Study Program have spread in various areas including in hospitals, government institutions, academics, pharmaceutical industry, pharmacists, and entrepreneurs. The Faculty of Pharmacy of Universitas Ahmad Dahlan was established in 1996 with one study program, which becomes the oldest faculty of pharmacy among Muhammadiyah and Ashiah colleges. Since its establishment, the Faculty of Pharmacy has one study program, namely the Pharmacy Study Program with undergraduate level. In 2001, based on a rector's decree, UAD Faculty of Pharmacy started to open a new study program, namely the Pharmacist Professional Study Program. Along with the education development, in 2015, UAD established a postgraduate program in pharmacy.
Faculty of Pharmacy, Universitas Ahmad Dahlan is a private Muslim faculty owned by the biggest Muslim organization in the world, namely Muhammadiyah. We already granted A for the National Health Accreditation. Thus, we commit to raise the higher quality in ASEAN level. The integration of moral and intellectual integrity in the higher education system can support the graduates for passing the national examination. The research and publication of the lectures are conducted based on the national and international collaboration. We also serve the community using the pharmaceutical products and our knowledge.
The UAD Pharmacy Study Program commits to the faculty vision of becoming a leading pharmacy higher education institution that is internationally recognized based on Islamic values. Not only that, UAD Pharmacy Study Program implements learning that instills and improves Islamic values in all aspects of campus life. Every year, more than 1,000 students from various provinces in Indonesia study together in the UAD Pharmacy Study Program. UAD Pharmacy Study Program applied curriculum for 4 years of study. It has represented all aspects of the competencies achievements for graduates. UAD Pharmacy Study Program has about 50 teaching staffs with doctor and professor qualification. There are 30 education staffs with appropriate competencies to strengthen the learning, research process, and community service. The institution evaluated their staff annually to improve professional development, monitoring, and evaluation. UAD Pharmacy Study Program built a strong partnership with various parties by involving alumni and stakeholders for the development of higher education. To support the activities of the three Dharma perguruan tinggi, namely learning, research, and community service, UAD Pharmacy Study Program has good, comfortable, and integrated facilities. The UAD Pharmacy Study Program has a laboratory with well-standardized equipment for teaching and learning process. UAD Pharmacy Study Program attempts to improve cooperation network with various parties, including the government, pharmaceutical industry, local higher education, and abroad.
The students and lecturers' achievements are already reached the national and international level as well. And finally, join with us, get the higher quality, and reach the better moral and intellectual integrity.
With competence that supports various abilities, both theories and soft skills, graduates of UAD Pharmacy Study Program have spread in various areas including in hospitals, government institutions, academics, pharmaceutical industry, pharmacists, and entrepreneurs. The Faculty of Pharmacy of Universitas Ahmad Dahlan was established in 1996 with one study program, which becomes the oldest faculty of pharmacy among Muhammadiyah and Ashiah colleges. Since its establishment, the Faculty of Pharmacy has one study program, namely the Pharmacy Study Program with undergraduate level. In 2001, based on a rector's decree, UAD Faculty of Pharmacy started to open a new study program, namely the Pharmacist Professional Study Program. Along with the education development, in 2015, UAD established a postgraduate program in pharmacy.
Faculty of Pharmacy, Universitas Ahmad Dahlan is a private Muslim faculty owned by the biggest Muslim organization in the world, namely Muhammadiyah. We already granted A for the National Health Accreditation. Thus, we commit to raise the higher quality in ASEAN level. The integration of moral and intellectual integrity in the higher education system can support the graduates for passing the national examination. The research and publication of the lectures are conducted based on the national and international collaboration. We also serve the community using the pharmaceutical products and our knowledge.
The UAD Pharmacy Study Program commits to the faculty vision of becoming a leading pharmacy higher education institution that is internationally recognized based on Islamic values. Not only that, UAD Pharmacy Study Program implements learning that instills and improves Islamic values in all aspects of campus life. Every year, more than 1,000 students from various provinces in Indonesia study together in the UAD Pharmacy Study Program. UAD Pharmacy Study Program applied curriculum for 4 years of study. It has represented all aspects of the competencies achievement for graduates. UAD Pharmacy Study Program has about 50 teaching staffs with doctor and professor qualification. There are 30 education staffs with appropriate competencies to strengthen the learning, research process, and community service. The institution evaluated their staff annually to improve professional development, monitoring, and evaluation. UAD Pharmacy Study Program built a strong partnership with various parties by involving alumni and stakeholders for the development of higher education. To support the activities of the three Dharma perguruan tinggi, namely learning, research, and community service, UAD Pharmacy Study Program has good, comfortable, and integrated facilities. The UAD Pharmacy Study Program has a laboratory with well-standardized equipment for teaching and learning process. UAD Pharmacy Study Program attempts to improve cooperation network with various parties, including the government, pharmaceutical industry, local higher education, and abroad.
The students and lecturers' achievements are already reached the national and international level as well. And finally, join with us, get the higher quality, and reach the better moral and intellectual integrity.
With competence that supports various abilities, both theories and soft skills, graduates of UAD Pharmacy Study Program have spread in various areas including in hospitals, government institutions, academics, pharmaceutical industry, pharmacists, and entrepreneurs. The Faculty of Pharmacy of Universitas Ahmad Dahlan was established in 1996 with one study program, which becomes the oldest faculty of pharmacy among Muhammadiyah and Ashiah colleges. Since its establishment, the Faculty of Pharmacy has one study program, namely the pharmacy study program with undergraduate level. In 2001, based on a rector's decree, UAD Faculty of Pharmacy started to open a new study program, namely the Pharmacist Professional Study Program. Along with the education development, in 2015, UAD established a postgraduate program in pharmacy.
Faculty of Pharmacy, Universitas Ahmad Dahlan is a private Muslim faculty owned by the biggest Muslim organization in the world, namely Muhammadiyah. We already granted A for the National Health Accreditation. Thus, we commit to raise the higher quality in ASEAN level. The integration of moral and intellectual integrity in the higher education system can support the graduates for passing the national examination. The research and publication of the lectures are conducted based on the national and international collaboration. We also serve the
menggunakannya karena analisa ini dengan cara melihat perbandingan dua variabel atau lebih. Jadi hasil dari penelitian saya pertama yaitu usia pasien, ada 3 golongan. Ini didasarkan dari kehamilan beresiko atau tidak. Jadi golongan beresiko adalah di bawah 20 tahun dan di atas 35 tahun. Sedangkan usia produktif untuk wanita hamil adalah pada usia 20 sampai 30 tahun. Jumlah pasien ini, jadi jumlah pasien pada penelitian ini yang sudah masuk dalam kriteria inklusi dan eksklusi ada 50 orang. Dengan frekuensi tertinggi pada umur 20 sampai 35 tahun, sebanyak 41 pasien. Dan yang paling rendah adalah usia di bawah 20 tahun, yaitu 2 pasien. Untuk variabel selanjutnya yaitu usia kehamilan, ada 3 golongan yaitu trimester 1, trimester 2, dan trimester 3. Jadi kategori tertinggi untuk wanita hamil mendatangi rumah sakit ada pada trimester pertama yaitu 21 pasien dengan persentase 42, yang terendah yaitu kategori trimester ketiga sebanyak 13 pasien. Selanjutnya ini adalah perbandingan persepan sebelum pandemi dan selama pandemi. Jadi sebelum pandemi ada 4 jenis sebelum melakukan comparison ada 4 produk yang digunakan pada rumah sakit Nasrul Ummah untuk diresepkan kepada wanita hamil, yaitu produk A, produk B, produk C, dan produk D.
Bisa
Sebelum
dipercepat
pandemi,
sedikit, Mbak Sofia?
ya, sebelum
Bisa
pandemi,
dipercepat sedikit.
jadi sebelum pandemi ini produk A itu yang paling banyak, yaitu sebanyak 84%. Sedangkan selama pandemi produknya itu 36% pada produk B. Jadi pada kesimpulannya produk A dengan persentase sebelum pandemi 8% mengalami penurunan sebanyak 40%. Dan juga terdapat kenaikan jumlah persepan vitamin dialami oleh produk B sebanyak 28%, produk C sebanyak 4%, dan produk D sebanyak 8%. Di sini saya juga melihat mengevaluasi dua variabel yaitu dosis dan jenis vitamin. Jadi dosis yang sesuai dengan yang dianjurkan pada ibu hamil ini yang sesuai ada 5
Mbak Sofia bisa langsung pada kesimpulannya, Mbak, sudah 7 menit. Silakan.
Jadi kesesuaian dosis vitamin sebanyak 5 persepan dan ketidaksesuaian sebanyak 45 persepan, yaitu 90%. Untuk jenis vitamin sendiri sebanyak 0 persepan vitamin jenis vitamin yang sesuai dan 50 persepan yaitu 100% tidak sesuai. Kesimpulannya ada perbedaan persepan vitamin pada wanita hamil sebelum dan selama COVID-19 dengan persepan vitamin yang kurang rasional. Jadi saya menggunakan referensi dari buku.
Baik, terima kasih Mbak Sofia. Kesimpulannya sudah disampaikan. Ya, bisa kita langsung kepada Bapak Ibu audiens. Barangkali ada yang perlu diklarifikasikan untuk Saudara Rahel Pasa. Sudah disiapkan posternya? Ya, insya Allah baik-baik saja ya. Tadi sudah sempat di-restart.
Iya, Bu.
Bapak Ibu yang lain ada yang perlu diklarifikasikan untuk Saudara Sofia Hafidah dari Lamongan ya Mbak, Universitas Muhammadiyah Lamongan ya? Iya, penelitiannya tentang persepan vitamin pada wanita hamil sebelum dan selama masa pandemi COVID-19. Ada yang ingin diklarifikasikan Ibu dan Bapak? Jika belum ada yang diklarifikasikan, saya ingin menyampaikan sesuatu untuk Mbak Sofia.
Iya, Bu.
Dalam presentasi hanya disampaikan produk A, produk B, dan produk C.
Ya.
Boleh disampaikan produk A, produk B, dan produk C-nya itu seperti apa? Mungkin tidak harus merek, tapi bisa disampaikan bahwa produk A itu adalah asam folat dan zat besi, mungkin produk B adalah kombinasi asam folat, zinc, dan zat besi, mungkin dari produk C adalah kombinasi asam folat, zinc, kalk atau kalsium laktat dan zat besi. Mungkin bisa disampaikan?
Oh ya, baik Bu. Jadi dari 4 produk ini, dari produk A ini sendiri adalah produk yang mengandung vitamin. Jadi ini adalah suplemen vitamin dan mikronutrien ya, seperti mineral juga. Tapi yang saya lihat adalah vitaminnya. Jadi di produk A, vitaminnya itu terdiri dari vitamin di produk A terdiri dari vitamin A, B6, B9, B12, vitamin D. Sedangkan di produk B lebih lengkap, yaitu vitamin A, B1, B2, B3, B6, B9, B12, vitamin C, dan D. Untuk produk C terdiri dari asam folat saja. Untuk produk D hanya terdiri dari vitamin B9, B12, dan juga vitamin C.
Baik. Untuk dosisnya sendiri dilihat dari mana, Mbak?
Dosisnya itu dilihat dari standar acuannya, Bu. Jadi yang menjadi standar acuan dari penelitian ini dari buku mikronutrien.
Maksud saya Anda menghitung dosis, bukan menghitung, membuat definisi operasional atau kriteria istilah dosis itu apa? Potensi, durasi, dan frekuensi, atau hanya potensi saja, atau hanya durasi dan frekuensi saja, atau bagaimana?
Jadi untuk dosis ini saya lebih melihat ke setiap produk kan ada dosisnya, Bu. Jadi saya mempunyai standar acuan, jadi ada range dosis.
Bukan standar acuannya, Mbak Sofiha. Mbak Sofiha, bukan standar acuannya. Dari produk A, produk B, dan produk C, menentukan dosisnya, kan pasti ada membuat sebuah definisi operasional.
Oh iya.
Definisi dosisnya itu apa? Potensi, durasi, dan frekuensi, atau frekuensi dan durasi saja, atau hanya potensinya saja?
Oh, sepertinya frekuensi sama ini, Bu. Frekuensinya, Bu.
Frekuensinya saja, berarti hanya melihat ya, asam folat sekian mikrogram, asam folat sekian international unit 1 kali sehari selama 30 hari. Ya, dosisnya seperti itu.
Ya,
berarti potensi, frekuensi, dan durasi.
Oh iya, Bu, benar.
Baik, terima kasih Mbak Sofiha.
Ya, Bu.
Selamat sudah
sama-sama.
menyelesaikan tahapan ini. Kita berpindah pada Mbak Rahel Pasa Visa Abdillah, panjang sekali namanya. Silahkan share screen terlebih dahulu untuk menyampaikan hasil pengamatan mengenai pengetahuan. Langsung share screen saja, Mbak. Saya mengisi waktu, Anda share screen, saya sebut judul, tidak perlu menunggu lebih detail lagi mengenai pengetahuan, sikap, praktik, dan kecemasan tentang vaksinasi COVID-19 pada masyarakat Kabupaten Wonosobo. 7 menit, silahkan Mbak Rahel.
Ya, bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Di sini saya akan mempresentasikan hasil penelitian saya yang berjudul Pengetahuan, Sikap Praktik, dan Kecemasan tentang Vaksinasi COVID-19 pada Masyarakat Kabupaten Wonosobo. Latar belakang penelitian saya yaitu penerimaan vaksin COVID-19 dipengaruhi oleh banyak faktor seperti pengetahuan, persepsi mereka tentang kemungkinan yang dirasakan dari penyebaran COVID-19, persepsi keamanan vaksin, logistik, kemanjuran, dan risiko yang vaksin yang dirasakan. Oleh karena itu, sangat penting untuk menafsirkan masyarakat mengenai pengetahuan, sikap praktik, dan kecemasan tentang vaksinasi COVID-19 untuk meningkatkan penerimaan massal. Objektif penelitian saya yaitu masyarakat Kabupaten Wonosobo, dan metode penelitian saya yaitu menggunakan desain penelitian observasional dengan pendekatan cross-sectional. Sampel yang digunakan adalah sebanyak 201 sampel. Untuk hasil dari penelitian saya yaitu mayoritas masyarakat Kabupaten Wonosobo 56,7% tidak mengetahui bahwa masyarakat wajib secara hukum mengikuti program vaksinasi COVID-19. Kemudian ada tenaga kesehatan merupakan sumber informasi yang paling dipercaya oleh sebagian responden dengan presentasi 48,3%. Kemudian, sebagian besar responden merespon positif terhadap vaksinasi COVID-19 dengan presentasi 69,1%. Vaksin COVID-19 dapat membantu melawan infeksi COVID-19 sebanyak 73,6%. Kemudian, mayoritas responnya merespon positif untuk vaksin COVID-19 tersedia secara gratis sebanyak 80,6%. Lalu ada mayoritas responden merespon dengan sikap negatif untuk ketersediaan vaksin dengan presentasi 60,2%, efek samping 52,2%, kemungkinan terjadi kerusakan pada vaksin COVID-19 sebanyak 57,7%, kemudian efek tak terduga 58,7%, dan untuk kepentingan komersial perusahaan farmasi sebanyak 51,7%. Untuk kesimpulannya, mayoritas masyarakat Kabupaten Wonosobo bersikap positif terhadap vaksin COVID-19, namun masyarakat juga masih khawatir akan keamanan dan efek samping dari vaksin COVID-19. Sudah, Bu.
Singkat padat jelas ya, Mbak Rahel.
Penelitian saya di sini yaitu untuk mengandung hidroklorida pada produk herbal pangsing yang tersebar di Banjarmasin. Kemudian untuk metode penelitian di sini desainnya eksperimental, kemudian teknik pengambilan sampelnya purposive sampling, tempatnya dilakukan analisis di laboratorium kimia analisis Universitas Muhammadiyah Banjarmasin. Kemudian waktunya November 2020 hingga Februari 2021. Ini dihitung dari pengumpulan sampel. Kemudian alat dan bahan di sini alatnya menggunakan spektrofotometer UV vis merek Genesis. Kemudian ada juga bahan-bahan kaca seperti yang tertera. Kemudian bahannya di sini digunakan baku pembanding Farmakope Indonesia butramin hidroklorida. Kemudian ada juga di sini menggunakan pelarut dan untuk fase gerak di KLT nanti yang pastinya 15 sampel dari produk herbal pangsing. Kemudian di sini saya akan melakukan uji kualitatif dengan KLT dari pembuatan larutan standar hingga penyiapan preparasi sampelnya. Kemudian ada juga uji kuantitatif. Uji kuantitatif di sini dimulai dari penentuan panjang gelombang maksimum, operating time, kemudian penentuan kurva baku. Sebelumnya juga dilakukan validasi metode dari uji linearitas, LOD, LOQ, kemudian presisi, dan akurasi. Analisisnya di sini untuk analisis kualitatif dengan perhitungan nilai RF. Untuk analisis kuantitatif nanti akan diperoleh persamaan kurva baku yang akan membantu perhitungan pada kadar. Kemudian untuk hasil di sini, untuk hasil uji kualitatif dapat kita lihat di sebelah sini ada plat KLT-nya. Di sini dibantu dengan sinar UV, yaitu dengan panjang gelombang 254 nanometer dan 366 nanometer. Pada penelitian sebelumnya, memang si baku sibutramin ini terlihatnya di panjang gelombang 254 nanometer. Nah, dapat kita lihat di sebelahnya ini dihitung nilai RF-nya. Jadi yang muncul pada panjang gelombang 254 nanometer ini hanya ada 4 sampel sama seperti si standarnya. Dan keempat sampel ini tidak ada yang sama dengan standar. Jadi untuk melanjutkan ke kuantitatif, ini berdasarkan studi literatur sebelumnya pada penelitian pada penelitian Putra dan Wisnu itu pernah didapatkan nilai RF yang sama seperti sampel K, J, K, dan M. Maka dilanjutkan ke uji kuantitatif. Nah, di sini uji kuantitatif panjang gelombang maksimumnya didapat 223 nanometer. Kemudian untuk operating time ini operating time-nya 1-10 menit. Lalu untuk uji linearitas ini memenuhi standar dengan nilai koefisien determinasi 0,998 dan nilai koefisien korelasinya 0,993. Kemudian untuk LOD di sini didapatkan nilai 0,5 ppm dan LOQ di sini 1,68 ppm. Untuk presisi di sini
Nilai RSD-nya itu 1,47%, dan untuk akurasi di sini didapatkan 126,13%. Lalu kita hitung untuk hasil absorbansi sampel. Untuk sampel J ini kadar rata-ratanya itu didapatkan 5,8278%. Kemudian untuk sampel K itu kadar rata-ratanya 8,5351%, untuk sampel M ini 8,9342%. Lalu kita bandingkan dengan penelitian sebelumnya, yaitu pada penelitian Wisnu et al. Pada tahun 2017 ini semua sampelnya itu terdeteksi mengandung sibutramine hidroklorida, di mana pada sampel B itu kandungan terkecilnya yaitu 1,772%, dan pada sampel F ini yang terbesar yaitu 9,759%. Jadi kesimpulannya di sini kadar sibutramine hidroklorida pada sampel J, K, dan M adalah 5,8278%, 8,5351%, dan 8,9342%. Ini referensi yang saya gunakan. Sekian dan terima kasih. Mohon maaf apabila ada salah kata. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Terima kasih kepada Saudari Nadia atas presentasinya. Baik, dari rekan-rekan audiens ada yang ingin bertanya kepada hasil dari Saudari Nadia tadi? Ada? Boleh sambil di chat ya di kolom untuk komentar Zoom-nya. Berarti tadi ini ya, identifikasi ya terhadap beberapa produk pelangsing yang ada di Kota Banjarmasin ya, betul ya?
Iya, betul.
Oke. Baik kawan-kawan, silahkan ditanyakan.
Harus ada yang bertanya nih.
Izin bertanya, Bu.
Iya,
boleh, silahkan.
Tadi kan panjang gelombang yang digunakan itu apakah sudah sesuai kayak gitu dengan literatur yang sebelumnya?
Baik, silahkan dijawab, Saudari Nadia.
Terima kasih atas pertanyaannya. Jadi sudah sesuai dengan literatur sebelumnya, yaitu pada Maluf et al. Pada tahun 2007 itu didapatkan juga panjang gelombangnya 223 nanometer. Dan sebelumnya juga memang ada pada tahun 2016 itu Simamere itu panjang gelombangnya didapatkan 224 nanometer. Jadi masih sekitaran panjang gelombang itu. Jadi sesuai.
Oke, baik. Ada lagi mungkin yang ingin bertanya? Baru satu ya. Masih ada waktu sekitar 2 menit.
Saya kasih pertanyaan ya Mbak Nadia ya. Oke. Untuk mencari, mungkin bisa dijelaskan tadi cara mencari menentukan akurasi bagaimana Mbak?
Iya Bu, izin share screen.
Sama parameternya kok besar gitu ya, parameter keberterimaannya.
Jadi untuk uji akurasi ini pertama kita timbang 10 mg sibutramin ini secara duplo. Kemudian ada dua kan labu ukurnya masing-masing ditambahkan 10 mili aqua bidestilata. Kemudian salah satunya ini ditambahkan 9 mili larutan standar 1000 ppm yang sebelumnya sudah dibuat. Kemudian ini kita homogenkan, masing-masing larutannya ini kita ambil itu 150 mikroliter dengan mikropipet, kemudian dimasukkan ke labu ukur 10 mili, dietkan dengan aqua bidestilata. Lalu ini dilihat di spektrofotometernya dan dilakukan pengulangan sebanyak 6 kali. Kenapa 6 kali? Ini berdasarkan literatur dari pada tahun 2004, lupa namanya Bu, tapi ada 6 kali. Kemudian untuk keberterimaannya itu ada pada slide. Oh ya, pada Hermita 2004 tadi Bu, yang 6 kali pengulangan, syaratnya itu 80 hingga 110%.
Oke, mungkin nanti di ini ya, mungkin masukan aja dari saya untuk nanti dicek lagi. Coba Mbak tetap aja di slideshow. Tadi di kesimpulan, saya mulai dari kesimpulannya, Anda mendapatkan kadarnya itu kan sekitar 5% ya. 5%, kemudian ada 6%, ada 8%. Nah, kalau kadar segini, tentunya ini yang harusnya dijadikan syarat keberterimaannya. Syarat keberterimaannya harus ini, kemudian tidak mungkin syarat untuk kadar segini itu kemudian syarat keberterimaannya sampai 80 sampai 100 berapa tadi Mbak? 80 sampai 110% kalau nggak salah ya. Nah ini mesti range-nya kecil, biasanya sekitar 2%, 98 sampai 102% saja. Ya, tapi dicek lagi ya. Terus kemudian kalau untuk akurasi mungkin juga perlu dibaca lagi, mungkin kalau baca itu ya ada. Oh ini yang dilakukan di sini Anda pakai larutan baku yang yang hanya dibuat begitu ya. Nah, padahal kalau kita mau memvalidasi suatu metode, apakah dia itu bisa digunakan untuk menetapkan sibutramin dalam misalnya dalam campuran obat herbal begitu ya, maka sangat penting ada matriks atau placebo itu. Nah, padahal yang Anda gunakan untuk pengukuran akurasi tadi itu zat murni. Jadi saya kira ada dua metode ya dalam hal akurasi, itu dengan metode spike placebo atau dengan standar adisi standar. Nah, mestinya memang harus ada matriks atau placebo yang ada di situ. Kalau placebo nggak bisa dibuat, maka harusnya dengan metode adisi standar. Mungkin nanti diinikan lagi ya Mbak ya, masukkan aja.
Terima kasih Bu.
Baik, waktunya sudah habis Bu ya. Kita lanjut pada presentator selanjutnya. Selanjutnya Saudari Lirih Indriarini. Saudari Lirih Indriarini sudah bisa dipersiapkan.
Izin share screen.
Saya jadi kangen diajar Prof Nur Hasanah lagi nih.
Nah, nanti kembali lagi ke UAD aja.
Bentuknya video ya, Saudari Lirih ya. Baik, silahkan.
Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan saya Lirih Indriarini, perwakilan dari Universitas Muhammadiyah Malang. Di sini saya akan
Bisa diperbesar mungkin suaranya?
penelitian yang berjudul Perlindungan UV dan Aktivitas Antioksidan dari Ekstrak Kulit Jeruk atau Citrusinensis L. Osbeck untuk formulasi nanogel tabir surya. Kulit buah jeruk atau Citrusinensis L. Osbeck merupakan limbah organik yang mengandung senyawa polifenol, di mana senyawa polifenol memberikan fotoproteksi dalam penyerapan sinar UV dan sebagai antioksidan pada tabir surya. Nah, yang cocok untuk tabir surya yaitu gel yang dibuat sistem nanopartikel atau nanogel dengan ukuran partikel yang kecil yaitu 10-1000 nanometer. Ukuran partikel yang kecil ini memberikan kemampuan permeasi yang baik dan diharapkan dapat menyerap tabir surya sampai ke bagian kulit terdalam sehingga memaksimalkan kinerja produk tabir surya. Tujuan penelitian ini untuk mengetahui karakteristik fisikokimia, aktivitas antioksidan, nilai SPF dari sediaan nanogel tabir surya yang dikembangkan menggunakan ekstrak kulit jeruk. Untuk metode pembuatan sediaan nanogel, di sini ada dua, yaitu basis gel dan fase air. Basis gel disiapkan dari aquades, TEA, dan karbomer yang dikembangkan 24 jam. Lalu fase airnya terdiri dari propilen glikol, natrium metabisulfit, metilparaben, propilparaben, dan sisa aquades. Lalu keduanya dilakukan pengadukan. Selanjutnya untuk basis gel dilakukan pengecekan pH. Dan untuk fase air dilakukan pengadukan kembali sambil ditambahkan ekstrak sedikit demi sedikit. Lalu untuk fase air yang sudah jadi, dicampur ke dalam basis gel dan dilakukan pengadukan kembali. Lalu sedia nanogel sudah jadi, dilakukan pengadukan lagi selama 10 menit dengan kecepatan 1200 rpm dan diulang lagi sebanyak 3 siklus sampai terbentuk sedia nanogel yang baik. Berikut untuk tabel formulanya, di sini ada 3 formula dengan perbedaan konsentrasi ekstrak, yaitu formula 1 1%, formula 2 3%, dan formula 3 5%, dan juga bahan-bahan aktif yang lain untuk pembuatan sedia nanogel. Selanjutnya, untuk evaluasi karakteristik fisikokimia, telah dilakukan pengujian ukuran partikel, PDI dan potensial zeta, organoleptis, homogenitas, penentuan nilai pH, nilai viskositas, dan daya sebar. Untuk uji aktivitas antioksidan dengan metode DPPH, yaitu sampel nanogel dan asam ascorbat sebagai kontrol positif, direaksikan dengan DPPH. Lalu untuk sampel, dicampurkan dengan DPPH dengan konsentrasi 200 ppm. Larutan blanko dibuat dengan mencampurkan DPPH dan etanol. Kontrol positifnya dibuat dengan asam ascorbat yang dicampur dengan DPPH dan etanol. Lalu campuran-campuran tersebut dihomogenkan menggunakan vortex dan diinkubasi selama 30 menit pada suhu ruang dengan kondisi gelap. Lalu absorbansi diukur pada panjang gelombang 517 nanometer menggunakan spektrofotometer. Aktivitas antioksidan berupa presentasi inhibisi diukur menggunakan rumus yang sudah tertera di bawah. Lalu dilakukan uji SPF terhadap ekstrak dan sediaan dengan konsentrasi ekstrak 5000 rpm yang dilarutkan dalam etanol. Begitu pula dengan sediaan yang dilarutkan dalam etanol dan disonikasi selama 5 menit. Lalu dilakukan analisis menggunakan spektro dengan absorbansi pada panjang gelombang 290 sampai 320 nanometer dengan interval 5 nanometer. Lalu dihitung untuk nilai SPF-nya. Selanjutnya masuk ke hasil penelitian. Yang pertama, hasil evaluasi fisikokimia nanogel tabir surya ekstrak kulit jeruk. Untuk organoleptisnya, nanogel ekstrak kulit jeruk ini memiliki tekstur yang kental, berwarna coklat, dan bau khas jeruk. Warna coklat berasal dari warna ekstrak kulit jeruk, dan formula 3 memiliki warna yang paling pekat dibandingkan formula lainnya. Dapat dilihat gambar di bawah. Secara visual, ketiga formula ini tidak mengandung partikel yang kasar, sehingga mengindikasikan bahwa sediaan tersebut telah homogen. Yang kedua yaitu hasil ukuran partikel, PDI atau indeks fluidispersitas, potensial zeta, pH, viskositas, dan daya sebar. Untuk ukuran partikelnya di sini memasuki ukuran partikel yang nano, di mana memasuki rentang 10 sampai 1000 nanometer. Lalu untuk hasil indeks fluidispersitasnya mengindikasikan bahwa partikel memiliki ukuran yang kurang homogen atau polidispersi, yaitu ditunjukkan dari hasil lebih dari 0,3. Lalu untuk potensial zetanya diindikasikan sistem nanogel tersebut memiliki stabilitas dispersi koloid yang baik karena tidak adanya agregasi dan flokulasi. Lalu untuk pH-nya di sini terlihat mengalami penurunan dikarenakan semakin tinggi konsentrasi bahan aktif yang ditambahkan maka semakin rendah pH yang dihasilkan. Hal ini juga berkaitan dengan senyawa asam yang terkandung dalam jeruk sehingga memiliki pH yang cenderung asam. Akan tapi pH ini masih memenuhi kualifikasi untuk nilai pH yang ditolerasi oleh kulit. Lalu untuk hasil viskositasnya di sini mengalami penurunan seiring dengan penambahan ekstrak. Hal ini juga disebabkan sifat dari ekstrak yang ditambahkan bersifat cair, sehingga semakin tinggi ekstrak kulit jeruk yang ditambahkan semakin kecil pula viskositasnya. Lalu untuk daya sebarnya di sini sesuai dengan teoritis, yaitu berbanding terbalik dengan viskositas. Semakin kecil nilai viskositas, maka semakin besar daya sebarnya. Selanjutnya untuk hasil antioksidan dan proteksi UV-nya, di sini didapatkan untuk hasil antioksidan dengan nilai SI50 ekstrak lebih tinggi dari kontrol positifnya yaitu asam ascorbat. Sedangkan untuk nilai SPF ekstrak itu didapatkan nilai 37,36 di mana nilai ini dapat memberikan perlindungan yang tinggi. Lalu untuk hasil pengecekan antioksidan dan nilai SPF pada formula didapatkan bahwa di dalam formula 3 memiliki hasil persen inhibisi yang tinggi dan nilai SPF yang tinggi. Hal ini dikarenakan semakin tinggi ekstrak yang ditambahkan, maka semakin tinggi juga aktivitas antioksidan dan nilai SPF yang dihasilkan. Kesimpulannya yaitu menunjukkan ekstrak kulit jeruk dapat diformulasikan dalam bentuk nanogel dengan karakteristik yang baik. Lalu, sediaan ini juga memiliki nilai persen inhibisi yang menunjukkan adanya aktivitas antioksidan serta berpotensi sebagai sediaan tabir surya dengan proteksi sedang terhadap UV yang terlihat pada formula 3. Sekian presentasi dari saya, mohon maaf apabila ada salah kata. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Pas ya, 10 menit ini. Baik, mungkin kita buka untuk satu orang penanya ya, atau 2 orang penanya deh, nggak apa-apa. Baik, rekan-rekan yang ingin bertanya mungkin silahkan.
Pak Rizkyanti tuh raise hand.
Ya, oke, Bu.
Mbak Rizkyanti raise hand.
Mbak Rizkyanti?
Raise
Itu
hand.
tadi untuk
ya,
yang
baru muncul
ini,
tempat saya.
Yang sebelumnya, tapi
Oh, yang sebelumnya.
iya, tapi
bukan
sudah
yang
habis
ini ya.
waktunya.
Baik, yang ini mungkin rekan-rekan ada yang ingin ditanyakan. Ini raise hand-nya nggak bisa hilang berarti ya?
Bisa itu diklik itu bisa hilang.
Oh iya, diklik 2 kali. Silahkan, mungkin ada yang ingin ditanyakan? Atau mungkin dari Prof. Nur Hasanah ada yang ingin mengomentari dari penelitian yang dilakukan oleh peserta?
Oke, ya terima kasih. Mungkin ini aja sih tadi dari kesimpulan terakhir itu ternyata ini ya apa namanya ekstrak itu SPF-nya jauh lebih tinggi dibandingkan bentuk formula ya Mbak ya.
Iya, iya.
Tuh. Nah, Anda membandingkan nggak dengan ini apa namanya? Saya tidak melihat ya. Anda membandingkan nggak dengan hasil penelitian yang lain gitu. Artinya kenapa ternyata dibuat dalam bentuk nanogel ya, kemudian dibuat formula seperti itu, kok kemudian malah jadi lebih kecil begitu loh, SPF-nya lebih kecil kan nggak menguntungkan. Nah, apakah memang selalu terjadi seperti itu, ataukah mungkin sebenarnya di penelitian-penelitian lain itu bentuk nanogel itu akan lebih baik SPF-nya begitu?
Kalau sebenarnya ini dari
secara global di seluruh dunia dan digolongkan oleh WHO sebagai pandemi. Virus ini memiliki tingkat mutasi yang tinggi menjadi berbagai variasi genetik, sehingga virus SARS-CoV-2 ini digolongkan menjadi varian of concern dan varian of interest. Adanya mutasi dari virus SARS-CoV-2 ini menimbulkan pertanyaan apakah vaksin yang saat ini ada itu dipengaruhi efikasi dan keamanannya oleh mutasi dari virus SARS-CoV-2. Seperti yang telah saya sebutkan sebelumnya, bahwa virus SARS-CoV-2 ini digolongkan menjadi varian of interest dan varian of concern. Virus yang masuk ke dalam varian of interest adalah virus-virus yang mengalami perubahan pada ikatan reseptor, lalu adanya pengurangan netralisasi antibodi, adanya pengurangan efikasi treatment, adanya prediksi peningkatan transmisi atau keparahan penyakit, serta adanya potensi bahwa virus yang masuk ke dalam varian of concern ini dapat mempengaruhi diagnosis dari penyakit COVID-19. Beberapa contoh dari virus SARS-CoV-2 yang masuk ke dalam varian of interest diantaranya ada ETA, LOTA, Kappa, dan Lambda. Kategori yang kedua itu adalah varian of concern. Virus yang masuk ke dalam varian of concern diantaranya memiliki kriteria bahwa dia memiliki bukti atau evidence peningkatan transmisi serta keparahan penyakit. Lalu adanya pengurangan signifikan netralisasi antibodi, adanya penurunan keefektifan dari pengobatan atau vaksin, serta adanya bukti bahwa virus yang masuk ke dalam golongan ini dapat mempengaruhi uji diagnosis seperti adanya kesalahan diagnosis. Contoh virus yang masuk ke dalam varian of concern diantaranya ada alpha, beta, lalu ada gamma, dan juga ada delta. Untuk varian alpha ini pertama kali ditemukan di Amerika Serikat. Untuk varian beta ini pertama kali ditemukan di Afrika Selatan. Untuk gamma ini pertama kali ditemukan di Jepang dan Brazil. Kemudian yang terakhir ada delta, ini pertama kali ditemukan di India ketika adanya lonjakan kasus yang cukup signifikan di India, begitu. Lalu varian delta ini saat ini menjadi fokus utama masyarakat karena dikabarkan memiliki transmisi yang sangat tinggi. Hanya dengan berpapasan 5 hingga 10 detik saja tanpa masker, kita dapat tertular COVID-19 yang disebabkan oleh varian delta ini. WHO sendiri saat ini sedang fokus pada virus yang masuk ke dalam varian of concern dikarenakan virus-virus yang masuk ke dalam varian of concern ini dapat mempengaruhi vaksinasi yang saat ini sedang digalakan. Kemudian beberapa vaksin yang telah dikembangkan, diantaranya ada whole virus vaksin, contohnya ada Sinovac dan Sinopharm. Lalu ada viral vector vaksin, contohnya ada AstraZeneca dan Johnson & Johnson. Ada juga vaksin yang dibuat dari asam nukleat virus seperti Pfizer dan Moderna. Lalu ada juga vaksin yang dibuat dari protein subunit, contohnya adalah Novavax. Dari hasil review yang telah kami lakukan, untuk vaksin Pfizer, ini secara keseluruhan memiliki efikasi sebesar 95%, dengan efikasi pada varian alpha sebesar 90%, untuk varian beta dan gamma ini 88%, dan untuk varian delta, vaksin Pfizer ini memiliki efikasi sebesar 87,9%. Untuk vaksin Moderna, secara keseluruhan memiliki efikasi sebesar 94,1%, untuk varian alpha sebesar 88,1%, varian beta sebesar 61,3%, dan varian delta sebesar 79 hingga 96%. Untuk vaksin Sinovac yang pertama kali digunakan di Indonesia, ini secara keseluruhan memiliki efikasi sebesar 65,3%. Untuk varian gamma yang bergejala ringan, ini efikasinya sebesar 50,7%. Yang bergejala sedang sebesar 83,7%, dan bagi yang bergejala berat ini cukup tinggi, efikasinya sebesar 100%. Kemudian untuk vaksin Sinopharm secara keseluruhan memiliki efikasi sebesar 79%. Untuk vaksin Johnson & Johnson secara keseluruhan memiliki efikasi sebesar 66,9%. Untuk varian beta yang bergejala sedang, vaksin ini memiliki efikasi sebesar 64%, dan bagi yang bergejala berat memiliki efikasi sebesar 81,7%. Untuk varian delta memiliki efikasi sebesar 68,1% bagi yang bergejala sedang dan 87,6% bagi yang bergejala berat. Untuk vaksin AstraZeneca yang juga digunakan di Indonesia, ini secara keseluruhan memiliki efikasi sebesar 81%, kemudian untuk varian alpha sebesar 70,4%, beta sebesar 10,4%, dan varian delta sebesar 59,8%. Lalu ada vaksin Novavax dengan efikasi secara keseluruhan sebesar 89,7%, untuk varian alpha sebesar 86,3%, dan varian delta sebesar 93%. Lalu efek samping umum yang dirasakan oleh individu setelah menjalani vaksinasi pada umumnya mengalami nyeri pada tempat.
Bisa pada langsung kesimpulannya, Mbak Hasna?
Oh iya, Bu. Nyeri pada bagian yang disuntik, panas. Lalu ada
langsung kesimpulannya aja. Jadi kesimpulannya adalah
jadi untuk virus SARS-CoV-2 ini memiliki dua varian, yaitu varian of interest dan varian of concern, di mana untuk mencegah, salah satu cara untuk pencegahan si virus SARS-CoV-2 yaitu adalah vaksinasi dengan efikasi vaksin terbesar yaitu adalah Pfizer sebesar 95%. Baik, terima kasih.
Baik, terima kasih Mbak Hasna. Teman-teman ada yang ingin disampaikan? Ini menarik loh tabel itu. Kalau jeli, aduh aku nyesel kemarin divaksin Sinovac, kenapa dia nggak spesifik ke alpha, beta, dan delta begitu. Harusnya aku ikut yang AstraZeneca aja, jangan ikut yang Sinovac gitu. Ada nggak yang berpikiran seperti itu? Teman-teman yang lain ada? Sambil kita menunggu persiapan dari Arisa Astiwi Maulani. Teman-teman, karena kesimpulan yang diambil Mbak Hasna di sini hanya mengangkat yang paling efektif tertinggi adalah vaksin dari Pfizer, begitu ya. Tapi kalau di Indonesia yang masuk baru Astra dan Sino, gimana?
Untuk Astra dan Sinovac itu masih lebih tinggi di Astra sih, Bu. Menurut penelitian yang sudah review yang sudah kami lakukan.
Iya, ini review dari berapa artikel, Mbak Hasna?
Ada 10.
Coba lihat daftar pustakanya. Coba kita lihat daftar pustakanya. Ditarik posternya. Iya, yang ditampilkan hanya 3 daftar pustaka, jadi asumsinya di sini mini reviewnya hanya dari 3 jurnal ya?
Sebenarnya ada banyak, Bu, cuma yang nggak masuk semua daftar pustakanya, nggak cukup gitu. Jadi cuma diambil 3.
Padahal kan banyak ya untuk mendapatkan komparasi seperti ini kan bukan sesuatu yang mudah. Ya,
Iya.
Selamat ya Mbak Hasna ya. Kita sapa dulu Mbak Arisa Astiwi Maulani. Anda sudah hadir Mbak Arisa?
Sudah Bu, suara saya masuk ya Bu?
Mbak Arisa Astiwi Maulani ada loh, tadi saya lihat loh.
Iya Bu, suara saya masuk nggak ya Bu?
Kecil sekali Mbak Arisa. Mbak Arisa coba Anda setting. Di bagian video itu Anda klik, kemudian Anda choose virtual background, bagian audionya diklik. Anda pakai laptop kan?
Iya Bu, pakai laptop.
Bagian audio diklik, kemudian ada speaker output level, bagian volumenya dinaikkan.
Sudah
Coba
Bu,
Mbak.
suara
Lagi, naikkan lagi.
Udah Bu, udah pol.
Iya, dan ini lebih terdengar daripada yang tadi.
Iya,
Oke.
baik.
Boleh share screen Mbak Arisa Astiwi Maulani?
Iya, saya coba.
Mbak Arisa, ini nggak salah kamar?
Enggak Bu, emang di sini Bu. Saya juga.
Oh baik. Baik, oke. Kalau begitu tentang sesuatu yang sedikit berbeda ya, ada pengaruh pemberian sediaan X selama 28 hari terhadap jumlah leukosit dan diferensial leukosit pada tikus jantan galur spague Delhi.
Iya. Baik.
Silakan, Mbak.
Apakah poster saya sudah baik? Baik.
Sudah cantik terlihat seperti Mbak Arisa ya, pinggi-pinggi.
Baik.
10 menit, 7 menit untuk presentasi, 3 menit untuk tanya jawab, silahkan.
Baik Bu. Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera, Om Swastiastu, Namo Buddhaya, salam kebajikan. Baiklah, di sini saya akan mempresentasikan terkait pengaruh pemberian sediaan X selama 28 hari terhadap jumlah leukosit dan diferensial leukosit pada tikus jantan galur SD. Yang mana pada penelitian ini ada dosen pembimbing saya yaitu Bapak Dr. Akrom MKes. Yang mana di sini untuk sediaan X ini merupakan kombinasi dari jintan hitam, temulawak, dan madu. Ya, adanya sediaan X ini tentu perlu dilakukan uji keamanan. Nah, salah satu uji keamanan yaitu adalah uji toksisitas. Untuk uji toksisitas ini nantinya dilakukan pada 6 kelompok, yaitu 1 kelompok kontrol dan 5 kelompok variasi dosis. Kemudian untuk kenapa menggunakan darah atau leukosit di sini dikarenakan darah sendiri merupakan sebagai fungsi untuk transportasi, pengaturan ataupun pertahanan tubuh. Salah satu profil dari hematologi atau darah yang bisa digunakan sebagai sistem pertahanan imunitas yaitu adalah leukosit. Sehingga di sini dilakukan untuk pengamatan dari pengaruh sediaan dari jumlah leukosit ataupun diferensial leukosit atau jenis-jenis leukosit. Kemudian untuk tujuannya di sini adalah yaitu untuk mengetahui pengaruh pemberian sediaan X selama 28 hari terhadap jumlah leukosit dan diferensial leukosit pada tikus jantan. Nah, untuk lokasi penelitiannya yaitu di sini dilakukan di LPPT Unit 4 UGM pada bulan September sampai Oktober tahun 2020 dengan kelompok penelitian. Kelompok 1 adalah kelompok kontrol, kemudian kelompok 2 adalah dosis yaitu 0,018 mili per 200 gram berat badan, kelompok 3 0,09 mili per 200 gram berat badan, kelompok 4 adalah 0,45 mili per 200 gram berat badan, dan kelompok 5 2,25 mili per 200 gram berat badan, dan kelompok terakhir yaitu 4,5 mili per 200 gram berat badan. Untuk bahan-bahannya sendiri di sini ada X yang mana merupakan kombinasi dari inang hitam sebanyak 0,5 mili, kemudian 300 mg temulawak, dan madu at 1 gram. Kemudian juga ada EDTA, ada aquades, ketamin, dan giemsa. Untuk metodenya, sampel darah di sini akan diambil dari darah tikus melalui sinus orbitalis kurang lebih sebanyak 1,5 mili, kemudian akan ditambahkan EDTA untuk mencegah koagulasi, kemudian akan diukur secara dengan mesin ya, yaitu dengan hematologi analyzer untuk pengukuran jumlah leukosit dan diferensial leukosit. Hanya saja adanya keterbatasan alat di sini untuk pengukuran dengan hematologi analyzer, diferensial leukosit di sini tidak terlalu jelas, tidak terlalu banyak gitu parameter diferensial leukosit yang teramati, sehingga juga dilakukan pengamatan dengan menggunakan mikroskop dengan pewarnaan menggunakan giemsa. Untuk hasilnya, di sini data hasil jumlah leukosit tikus jantan dari semua kelompok, baik kelompok 1, kemudian kelompok 2, 3, 4, dan 5, dan 6. Di sini menggunakan 5 ekor untuk perlakuan per kelompoknya, yang mana dapat diketahui berdasarkan uji statistik ANOVA yang telah dilakukan bahwasanya persediaan X ini tidak memberikan pengaruh terhadap jumlah leukosit apabila diberikan selama 28 hari. Kemudian, untuk diferensial leukosit di sini yang teramati adalah limfosit, neutrofil, dan MXD. MXD ini merupakan gabungan antara monosit, eosinofil, dan basofil, sehingga adanya keterbatasan alat dari hematologi analyzer di sini dilakukan analisis menggunakan mikroskop, sehingga teramati bahwasanya di bawahnya yaitu terdapat penampakan sel monosit dan eosinofil. Kemudian MXD ini merupakan gabungan dari monosit, eosinofil, dan basofil. Basofil tidak terlihat pada saat pengamatan mikroskop dikarenakan giemsa yang mungkin kurang selektif terhadap adanya basofil ataupun jumlah basofil yang sangat sedikit, sehingga basofil tidak terlihat pada pengamatan dengan mikroskop. Berdasarkan literatur yaitu Halal tahun 2017, yang mana jumlah limfosit dan neutrofil ini setelah dilakukan uji MX, dilakukan uji LSD, tidak terdapat perbedaan untuk pengaruh pemberian sediaan X pada limfosit, tetapi pada neutrofil di sini memberikan pengaruh terutama pada dosis kedua, yaitu dosis 0,09 mili per 200 gram berat badan. Kemudian untuk MXD-nya, kemudian untuk pengamatan dengan mikroskopnya yang teramati adalah monosit dan eosinofil. Berdasarkan literatur yaitu Halal tahun 2017, bahwasanya jumlah monosit dan eosinofil yang teramati masih sesuai range normal, sehingga dapat diketahui bahwasanya sediaan X ini tidak memberikan pengaruh pada monosit, eosinofil, kemudian juga tidak memberikan pengaruh pada limfosit. Kemudian kenapa pada dosis 0,09 mili ini neutrofilnya mengalami peningkatan? Dikarenakan tikus berarti menandakan bahwasanya telah adanya sistem pertahanan tubuh atau imunologi, yang mana bisa disebabkan karena kondisi yang stres karena pengambilan pemberian sediaan itu kan setiap hari, sehingga bisa jadi pada tikus-tikus yang ada pada dosis 2 ini mengalami kondisi stres sehingga bisa mempengaruhi kenaikan jumlah neutrofil. Untuk kesimpulannya, sediaan X ini tidak memberikan pengaruh terhadap jumlah leukosit, kemudian tidak memberikan pengaruh juga terhadap limfosit, eosinofil, dan monosit, tetapi memberikan pengaruh terhadap persentase neutrofil, terutama pada dosis 0,09 mili per 200 gram berat badan. Terima kasih, Bu Ana. Mungkin itu saja dari saya, Bu.
Iya. Terima kasih. Nyaris saya ingatkan ya, pas di 6 menit.
Baik, Bu.
Mbak siapa ini? Mbak Arisa. Ada pertanyaan dari teman-teman yang siang hari ini bergabung ya. Mengapa intervensi sediaan X ini dilakukan dalam 28 hari?
Baik, terima kasih Bu Ana yang sudah mewakili untuk pertanyaan dari peserta, karena saya tidak dapat melihat pesertanya seperti itu, Bu. Jadi
lihat saya saja.
Baik, ya Bu Ana selalu cantik kok, Bu. Iya, baik.
Alhamdulillah. Ini energinya mendapatkan semuanya dari teman-teman yang hadir pada siang hari ini. Ya,
baik.
energinya naik ya, imunnya juga naik begitu.
Oke.
Gimana Mbak Arisa? Kenapa 28 hari?
Baik, di sini kenapa 28 hari? Karena di sini yaitu dilakukan untuk uji toksisitas subkronik atau pemakaian jangka panjang. Nah, sebenarnya toksisitas subkronik ini kan jangka range-nya itu 28 sampai 90 hari. Kemudian pada penelitian ini, kenapa 28 hari? Karena diambil syarat minimal untuk uji toksisitas subkronik seperti itu, Bu. Jadi karena syarat minimal
saya sebenarnya tadi juga saya juga ingin bertanya gitu ya. Kalau 14 hari itu kan akut gitu ya, 7 sampai 14 hari akut. Ini saya juga sudah menduga kenapa 28 hari ini subkronik gitu. Tapi kenapa 28 tidak dimaksimalkan di 90 hari gitu. Boleh tahu ini X-nya itu apa? Stat aktifnya ini dipapar apa? Sebenarnya tikusmu ini kamu papar apa sih?
Untuk sediaan X ini dia kombinasi dari jintan hitam, kemudian temulawak sama madu, Bu. Jadi ingin mengetahui kira-kira apabila sediaan ini kan berfungsi untuk sebagai preventif gitu ya. Nah, apabila digunakan jangka panjang, itu apakah memberikan pengaruh nantinya gitu. Sehingga di sini kenapa 28 hari ya karena syarat toksisitas subkronik itu memang 28 hari itu minimalnya seperti itu, Bu.
Iya, sayang banget hanya 28 hari ya, padahal ini untuk penggunaan jangka panjang. Pertanyaan dari saya pribadi, Mbak Arisa. Menurut Mbak Arisa, seandainya nih judulnya saya rubah uji toksisitas subkronik pada mencit atau pada tikus jantan galur Sprague Dowley, bisa nggak? Uji toksisitas subkronik sediaan X gitu?
Bisa saja, Bu, sebenarnya. Cuma di sini karena kan untuk kenapa saya menggunakan pengaruh pemberian sediaan, karena untuk uji toksisitas subkronik ini kan sebenarnya parameternya banyak, Bu. Jadi kita bisa untuk profil hematologi tentu banyak hal yang bisa kita pakai. Nah, karena di sini saya ini adalah penelitian berkelompok seperti itu, jadi tentunya kan saya tidak bisa memakai semua parameter karena harus berbagi dengan teman-teman saya yang lain sehingga untuk menghindari
karena Anda parameternya hanya jangka 28 hari sedangkan ada banyak parameter sehingga Anda tidak berani untuk mengangkat bahwa topik Anda adalah uji toksisitas gitu ya. Terima kasih Mbak Arisa, boleh ditutup screen-nya. Terima kasih. Ya, kemudian ini agak berat temanya. Firen Ramadan, Anda di mana? Firen, saya membacanya judul ini wow sekali ya. Deg-degan saya membacanya. Firen, bisa dibuka videonya, Firen? Iya. Dibuka mikrofonnya, Firen.
Oh ya, hadir. Mohon maaf.
Ini judulnya membuat saya ciut nyali.
Wah, enggak juga, Bu. Masih review.
Iya. Review the relationship of CYP2C19 gene polymorphism with clopidogrel resistance in cardiovascular and stroke patient.
Yes.
Correct?
Oke. Tapi hari ini saya menggunakan bahasa Indonesia ya, Bu. Soalnya kemarin juga sudah disubmit bahasa Indonesianya.
Oh, I think you will be conduct or delivered your article or your poster with in English. So today you will give information for us and present your article in bahasa.
Yes, in bahasa.
Okay.
In my poster also in bahasa.
I think it's better for us in bahasa.
Okay, ya.
Yeah. Okay, Firen Ramadan, time it's yours just for 7 minutes.
7 minutes. Artikelnya, posternya sudah saya kirimkan.
Baik, posternya sudah dikirimkan.
Ya, atau saya aja yang nge-share, Bu?
Boleh, daripada terlalu lama, di-share screen ulang saja.
Baik.
Selanjutnya mohon bersiap ya, Saudara Yolan Nisa. Studi literatur juga tentang evaluasi penggunaan obat indomitasin pada pasien GOUT. Mohon bersiap setelah Mas Firen menyampaikan artikelnya atau reviewnya, kita akan menuju ke Mbak Yolanisa. Sudah terlihat di layar Mas Firen, silahkan.
Ya, sudah terlihat ya, Bu ya?
Sudah.
Baik, terima kasih atas kesempatannya. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Alhamdulillahirrahmanirrahim. Puji dan syukur kita panjatkan atas kehadiran Allah, karena pada hari ini saya diberi kesempatan oleh rekan-rekan dari UAD, yaitu Bu Ana Hidayati, yang memberikan kesempatan kepada saya untuk mempresentasikan artikel atau literatur review melalui media poster yang saya bikin. Dengan judul Review Hubungan Polimorfisme Gen CYP2C19 dengan resistensi clopidogrel antiplatelet ya pada pasien kardiovaskular dan stroke. Baik, kita mengetahui bahwa farmakogenetika dapat diaplikasikan dalam perbedaan variabilitas respon terhadap suatu obat. Saat ini kita ketahui bahwa individualisasi terapi menjadi concern yang sangat penting oleh clinical pharmacy. Sama-sama kita ketahui bahwa cardiovascular disease seperti acute coronary syndrome, ya, penyakit-penyakit saraf seperti stroke, termasuk penyakit-penyakit yang prevalensinya cukup tinggi di Indonesia, dan juga menjadi masalah saat ini ya terkait dengan pandemi COVID-19 yang komorbiditas penyakit ini juga dapat membahayakan pasien jika tidak tertangani dengan baik. Menurut guideline of American College of Cardiology atau American Heart Association (AHA), clopidogrel dan aspirin ya, merupakan antiplatelet standar yang diresepkan pada pasien ACS, terutama pasien-pasien yang mendapatkan pemasangan stent. Ya, itu mendapatkan sekitar kurang lebih 1 tahun ya, bisa juga sampai hingga 2 tahun. Kita ketahui bersama bahwa clopidogrel merupakan inactive drug, ya, prodrug yang dikonversikan oleh sitokrom P450 di hepar menjadi metabolit aktif. Clopidogrel memblok P2W12 reseptor sehingga menjadi agen antiplatelet. Untuk CYP2C19 sendiri merupakan salah satu subfamily dari sitokrom P450. Salah satu problem seorang klinisi ya, atau dokter yang merawat pasien ACS dengan stroke adalah adanya resistensi dari clopidogrel. Resistensi clopidogrel sendiri ini terjadi juga karena mungkin terkait dengan dosis. Maka dari itu ya menarik untuk dilakukan riset bagaimana ya kita clinical pharmacy dapat berperan untuk memberikan rekomendasi pada dokter berapa awal mula dosis yang akan diberikan, terutama dari segi genetika orang-orang Indonesia. Tujuan dari artikel ini adalah bertujuan mengkaji dari beberapa artikel tentang bagaimana hubungan antara polimorfisme gen CYP dengan resistensi clopidogrel pasien kardiovaskular dan stroke. Untuk pembahasannya sendiri, efisiensi terapi clopidogrel dikontrol oleh mekanisme aksi enzim sitokrom. Penyakit jantung iskemik dan stroke merupakan penyumbang terbesar kesehatan di dunia, sekitar 31%, kematian pada tahun 2021. Suatu penelitian menunjukkan hasil klinis yang lebih baik dengan strategi farmakogenomik, yaitu mengetahui genotype pasien dengan ACS yang memiliki alel star 2 and star 3. Ya, karena kita ketahui bahwa alel pada CYP ada star 2, 3, 4, 5, 6, 7, and 8. Namun yang menarik untuk kita ketahui adalah alel 2 dan 3 ini karena dia poor metabolizer. Dan juga alel ini cukup banyak ya di populasi Asia. Populasi Asia sendiri prevalensinya itu sekitar 18% dan merupakan prevalensi tertinggi dibanding populasi-populasi lain di seluruh dunia. Begitu juga dengan Indonesia ya, studi di Indonesia baru-baru ini terutama hanya di satu daerah ya, hanya ambil sampling di satu daerah di Sumatera Selatan, ras Melayu, itu sekitar 46,7%. Jadi memang clopidogrel ini resistensi klopidogrel ya sangat dipengaruhi oleh orang-orang yang memiliki polimorfisme CYP2C19 ini dari alel STAR2 dan STAR3, sehingga dia kecenderungan untuk menjadi resisten oleh klopidogrel tinggi. Maka dari itu ya, adanya hubungan polimorfisme gen CYP2C19 dengan kejadian resistensi klopidogrel, sehingga perlu dilakukan penelitian lebih lanjut agar dapat membuat pedoman tata laksana pencegahan dan penanganan resistensi klopidogrel, ya, termasuk adjustment dose-nya melalui kajian-kajian farmakogenetik, sehingga ya tidak mengganggu prognosis pasien dan dokter, klinisi maupun clinical farmasi ya mampu memperbaiki kualitas hidup pasien pada pasien-pasien kardiovaskular dan stroke. Mungkin cukup penyampaian dari saya. Saya kembalikan kepada Bu Ana. Terima kasih, Bu Ana.
Terima kasih Bapak, Ibu, Saudara Firen Ramadan. Ini Bapak, Ibu, Saudara Firen Ramadan ini kalau layar yang di depan saya kayak orang pantomim ini, jadi yang terlihat hanya wajahnya saja.
Oh,
jadi
iya,
goyang-goyang berasa topeng itu ya.
Bagaimana
kalau
ini ya?
videonya aja ini ya pakai masker. Dari Universitas Muhammadiyah Kudus,
Iya, dari Universitas
Alhamdulillah.
Muhammadiyah
Kudus
Kudus.
Mandali, Mas atau Pak Firen?
Apa Bu? Mohon maaf.
Mandali ya, di Kudus aman terkendali.
Oh, Mandali aman terkendali. Alhamdulillah aman Bu,
Iya.
terkendali.
Kudus sudah lewat, sekarang Jogja yang luar biasa.
Iya.
Iya, sambil kita menunggu barangkali ada respon atau ada feedback dari teman-teman dan seluruh rekan-rekan yang hadir dalam ruangan ini. Pak Firen atau
Ya.
Mas Firen, kenapa tertarik di klopidogrel? Apa karena klopidogrel itu lebih banyak digunakan sebagai first line-nya kalau digunakan untuk terapi-terapi yang hubungannya dengan penyakit kardiovaskuler?
Ya, tertarik dengan klopidogrel ya. Pertama ya memang merupakan first line ya. Kita ketahui bersama dari guideline ya, dari guideline internasional maupun guideline sendiri dari Persatuan Dokter ya Jantung atau Kardiovaskular Indonesia juga meresepkan klopidogrel ya. Dan ini memang juga sangat ya, banyak digunakan ya, terkait dengan efektivitasnya juga cukup baik ya pada pasien-pasien yang mendapatkan terapi pemeliharaan ini. Namun ada beberapa sedikit problem ya, tapi nggak semuanya. Dari beberapa pasien juga butuh individualisasi terapi. Belum lagi kita bicarakan terkait dengan kombinasi dengan aspirin ataupun ticagrelor, seperti itu Bu. Jadi ya memang cukup menarik ini. Untuk tahun 2010 sendiri sebenarnya di Amerika Serikat sudah mulai dilakukan penelitian untuk adjustment dose bagi mereka yang memiliki polimorfisme ini ya. Namun untuk di Indonesia ini menarik nih, jika belum dilakukan ya. Mungkin kedepannya rencananya aja saya juga mau concern di sini, insyaallah.
Benar sekali, karena banyak sekali kondisi-kondisi, sebenarnya nggak cuma di kardiovaskuler aja ya Mas atau Pak Firen ini ya, karena beberapa juga kita jumpain pada kasus-kasus antidepresan misalnya, pada kasus-kasus depresi seperti itu sudah diberikan dosis yang sesuai, sudah diberikan potensi, durasi, dan frekuensinya juga sudah sesuai, tetapi tidak memberikan efektivitas yang baik, masih aja tetap seperti itu. Ternyata setelah dikaji, polimorfisme juga. Dan akhirnya melihat juga untuk yang kardiovaskuler ya, setelah beberapa presentasi teman-teman yang lain melihatnya dari TBC sehingga muncul multidrug resisten, kemudian dari antidepresan dan dari jenis-jenis obat psikosis yang lain. Terima kasih Mas atau Pak Firen Ramadan dari Universitas Muhammadiyah Kudus, menarik sekali ya, sesuatu yang berbeda. Kita menuju Bapak, Ibu atau Saudara Yaulan Nisa mengenai studi literatur evaluasi penggunaan obat indometasin pada pasien GERD.
Ya, Bu.
Ada Mbak Yulan di sini?
Ya, Bu.
Oke, silakan. Setelah ini kita menuju ke Mbak Widya Akmasari ya. Mbak Widya Akmasari sudah hadir, akan menyampaikan informasi tentang tingkat pengetahuan dan perilaku masyarakat ya. Mbak Widya Akmasari. Oh, bukan. Survival rate. Oke, ya. Oke, sudah muncul Mbak Yolan.
Baik.
Mbak Yolanisa sudah muncul, silahkan 7 menit waktu Anda untuk presentasi.
Assalamualaikum warahmatullahi wabarakatuh. Saya Yolanisa dari Universitas Muhammadiyah Banjarmasin. Di sini saya ingin mempresentasikan tentang penelitian yang saya lakukan, yaitu studi literatur evaluasi penggunaan obat indomethacin pada pasien GERD. Untuk pendahuluannya, GERD akut merupakan kasus yang cukup banyak terjadi di Indonesia dan di negara-negara lain. Menurut WHO, pada tahun 2017, GERD akut di dunia terjadi sekitar 34,2%. Untuk di Indonesia dilihat dari hasil diagnosa tenaga kesehatan yaitu 11,9% yang terjadi. Menurut Riskesdas pada tahun 2013, penderita yang paling banyak terserang yaitu pada wanita 27,5% dibandingkan pada pria 21,8%. Untuk obat golongan NSAID merupakan terapi lini pertama yang efektif mengobati GERD akut. Untuk obatnya yang sering diresepkan yaitu salah satunya indomethacin. Indomethacin dapat menghambat peradangan pada GERD akut serta dapat mengurangi rasa nyeri yang terjadi. Banyak pasien yang memberitahukan bahwa penggunaan indomethacin banyak sekali efek sampingnya. Oleh sebab itu, banyak peneliti yang mengevaluasi penggunaan obat indomethacin dari segi keefektifan dan efek sampingnya. Untuk tujuan penelitian yang saya lakukan yaitu untuk mengetahui evaluasi penggunaan obat indomethacin pada pasien GERD dengan metode studi literatur. Untuk metode penelitiannya, jenis penelitian ini studi literatur, dan untuk studi pustakanya dilakukan dengan cara mengumpulkan data dengan menelaah karya tulis dan hasil penelitian yang telah dipublikasikan. Untuk waktu penelitiannya mulai bulan Oktober 2020 sampai Maret 2021. Untuk dokumentasinya menggunakan Mendeley, dan ini merupakan hasil penelitian yang saya lakukan. Untuk jurnal yang didapat yaitu sebanyak 10 jurnal, akan tetapi yang dibahas ada 7 jurnal saja karena yang memenuhi persyaratan dengan penelitian saya hanya 7 literatur yang digunakan. Untuk hasilnya, di sini banyak di antara 7 jurnal tersebut, penelitian yang dilakukan membandingkan dengan cara memberikan obat indomethacin versus prednisolon dan versus eterokosip. Di sini dari hasil tersebut banyak yang menghasilkan bahwa indomethacin lebih banyak menghasilkan efek samping daripada obat prednisolon dan eterokosip. Untuk efektivitasnya, di sini dari semua literatur mengatakan bahwa efektivitasnya sama antara indomethacin, prednisolon, dan eterokosip sama untuk mengurangi rasa sakit karena GERD. Untuk kesimpulannya, penggunaan obat indomethacin efektif dalam mengatasi GERD setara dengan obat lain seperti prednisolon dan eterokosip. Efek samping dari penggunaan obat indomethacin dalam jangka pendek maupun jangka panjang lebih banyak terjadi dan banyak menyebabkan pasien berhenti melakukan pengobatan. Ini merupakan daftar pustaka yang saya gunakan, dan saya mengucapkan terima kasih kepada Ibu Apoteker Risha Mulyani, MSc selaku pembimbing penelitian saya, dan Ibu Apoteker Tuti Mulyani MSc selaku pembimbing juga, dan Bapak Apoteker Andika MPAM selaku Kaprodi pada kampus saya, dan pada Bapak Apoteker Dede Hartanto MSc selaku pembimbing saya juga. Sekian Bu Ana, terima kasih.
Terima kasih Mbak Yolanisa dari Universitas Muhammadiyah Banjarmasin. Mohon disampaikan salam kami untuk Bapak Ibu pembimbingnya. Semoga selalu sehat dalam lindungan Allah Subhanahu wa taala. Dan Mbak Yolanisa, studi literatur tentang evaluasi penggunaan obat indomethacin pada pasien GERD. Yang dievaluasi tadi sebatas efektivitasnya, sebatas apa lagi Mbak?
Efek samping.
Efek samping juga tadi ya?
Ya.
Okay. Mungkin dari teman-teman yang lain silakan. Ada speechless semua ya ini teman-teman yang lain. Obatnya tadi yang direview sebatas ini ya Mbak Yolan ya, indomethacin, kemudian ada prednisolon, kemudian ada dari siklooksigenase 2, ada dari CHOX2. Ya.
Yep.
Nggak nemu artikel yang isinya tentang allopurinol. Katanya nih, kalau untuk GERD itu banyak juga pakai allopurinol. Statement saya ini bener nggak? Katanya atau beneran?
Mungkin banyak orang mengatakan lubrinol lebih banyak digunakan pada pasien GERD, akan tetapi yang saya baca, kebanyakan lubrinol digunakan pada pasien yang mengalami hiperesemia sebelum GERD.
Jadi beda ya? Apa beda hiperurisemi dengan GERD menurut Mbak Yolan?
Kalau hiperurisemi itu merupakan asam urat yang asam uratnya cukup tinggi, akan tetapi belum mengalami peradangan dan bengkak, Bu. Sedangkan untuk GERD itu sudah mengalami peradangan dan bengkak.
Ada bedanya, sehingga yang banyak dilihat di sini adalah penggunaan obat-obat antiinflamasi dan juga obat-obat yang antiinflamasinya spesifik ya, yaitu pakai CHOX2. Kemudian ada yang ingin menanyakan kepada Mbak Yolan, kenapa tertarik pada kasus-kasus GERD gitu? Kenapa nggak seperti Pak Firen tadi yang cardiovascular disease gitu? Kenapa Mbak?
Karena ya di lingkungan saya sendiri juga lumayan banyak yang mengalami GERD. Jadi kemarin awalnya cuman mau menganalisis tentang obat NSAID secara keseluruhan. Akan tetapi untuk jurnalnya lebih banyak yang didapat tentang indomethacin, prednisolon, sama metrokosid. Nah, itu mungkin salah satu alasannya.
Oke.
Saat misalnya terbentuknya mungkin ada riset baru gitu ya, itu apakah nanti efeknya ke dalam tubuh terutama itu seperti apa? Gitu aja mungkin Pak Rizky.
Terima kasih Ibu atas pertanyaannya. Satu lagi mungkin masih bisa ini ya. Satu lagi, ada pertanyaan dari rekan-rekan audiens? Tidak ada? Karena waktunya ini sudah habis ya. Izin untuk saya lanjutkan ke presentator selanjutnya. Terima kasih Mbak Ani Sintara atas presentasinya. Selanjutnya kita panggil terakhir ini ya berarti ya. Mbak Galuh ya. Mbak Galuh ya. Mbak Galuh Purnamaningrum. Kepada Mbak Galuh Purnamaningrum, hadir? Hadir ya. Nah, silahkan dipersiapkan Mbak Galuh untuk presentasinya. Bentuknya presentasi atau video?
Presentasi.
Presentasi. Silahkan di-share screen. Baik, judulnya analisis dari rodamin ya, pada kolang-kaling. Oke, Mbak Galuh, waktunya 7 menit kepada para audiens untuk bisa dipersiapkan pertanyaan untuk Mbak Galuh. Silahkan Mbak Galuh.
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Perkenalkan, saya Galuh Purnamaningrum. Di sini saya akan mempresentasikan hasil penelitian saya yang berjudul Analisis Rodamin B pada kolang-kaling yang dijual pedagang ronde dengan metode spektrofotometri visible. Pendahuluan, makanan yang sehat adalah makanan yang mengandung 5 nilai gizi, yaitu karbohidrat, protein, lemak, vitamin, dan mineral, serta tidak mengandung unsur-unsur yang berbahaya bagi tubuh. Produsen makanan memiliki ciri khas pada produk makanannya, baik dari segi warna, rasa, dan aroma, terutama yang memiliki pengaruh besar yaitu bahan pewarna. Di era ini masih banyak pedagang yang menambah bahan tambahan berbahaya pada makanan sehingga dapat menimbulkan berbagai macam penyakit. Contohnya adalah rodamin B. Rodamin B ini adalah zat pewarna sintetik yang memiliki sifat fisik kristal berwarna kehijauan dan larutan berwarna merah keunguan pada konsentrasi tinggi dan merah terang pada konsentrasi rendah. Zat warna ini memiliki banyak keuntungan sehingga dipakai para pedagang, yaitu harganya yang murah, stabil, praktis, warnanya yang beragam, mudah disimpan, dan lain-lain. Selain itu, rodamin B ini dapat menyebabkan efek berbahaya, contohnya iritasi saluran pernafasan, kulit, mata, saluran pencernaan, keracunan, gangguan hati, dan kanker. Tujuan dari penelitian ini adalah untuk mengidentifikasi kandungan rodamin B pada kolang-kaling yang dijual pedagang ronde
terutama di alun-alun kota Yogyakarta. Untuk bahan dan alatnya sendiri, bahannya ada spektrofotometer UV 1800, alat-alat gelas, plat benang wol, reagen kit tes rodamin B, dan timbangan analitik. Untuk bahannya ada kolang-kaling, amonia 2%, etanol 96%, asam asetat 10%, aquades, amonia 10%, dan rodamin B standar. Untuk metodenya sendiri diawali dengan analisis kualitatif, yaitu dengan dua metode. Yang pertama, metode perbandingan panjang gelombang dan menggunakan reagen kit tes rodamin B. Kemudian analisis kuantitatifnya dengan metode spektrofotometri visible. Kemudian ekstraksi sampelnya menggunakan penarikan warna dengan benang wol. Kemudian pembuatan kurva baku pada konsentrasi 1, 1,5, 2, 2,5, 3, 3,5, dan 4 ppm. Kemudian melakukan validasi metode, yaitu linearitas, presisi, LOD, dan LOG. Kemudian analisis statistik dengan SPSS 25 menggunakan uji Kolmogorov-Smirnov dan ANOVA. Hasil dan pembahasan. Yang pertama, pada analisis kualitatif menggunakan reagen kit tes rodamin B. Uji kualitatif ini dilakukan pada 6 sampel, yaitu sampel A, B, C, D, E, dan F, masing-masing 2 kali replikasi. Pada setiap sampel ditambah 2 buah reagen. Untuk reagen yang pertama berisi SbCl3 dan reagen yang kedua berisi C7H8. Dapat dilihat pada gambar, ini standar rodamin B yang telah ditambah reagen, yaitu berwarna merah keunguan. Kemudian untuk salah satu contoh pada sampel yang positif, yaitu sampel A, B, dan D, ini setelah ditambah 2 buah reagen tersebut memiliki warna merah keunguan seperti pada standar rodamin B. Kemudian untuk yang sampel negatif, yaitu sampel C, E, dan F ini tidak mengalami perubahan warna pada setelah ditambah reagen tersebut. Untuk mekanisme reaksinya, rodamin B ditambah SbCl3 ditambah C7H8 akan menjadi kompleks rodamin antimon yang berwarna ungu. Kemudian, perbandingan panjang gelombang. Sampel hasil ekstraksi ini dianalisis pada spektrofotometer visible pada range panjang gelombang 400 sampai 600 nanometer. Dapat dilihat pada gambar bahwa panjang gelombang maksimum standar rodamin B yaitu 557,4. Dan salah satu contoh pada sampel B ini memiliki panjang gelombang 557,6. Sehingga dapat dinyatakan bahwa dari perbandingan panjang gelombang ini, sampel B memiliki panjang gelombang yang mendekati dari standar rodamin B. Kemudian pembuatan kurva baku dengan konsentrasi berikut dan dianalisis dengan spektrofotometer visible. Didapatkan panjang persamaan regresi linear, yaitu Y sama dengan 0,1137 X plus 0,147 dengan nilai koefisien korelasi 0,9971. Kemudian masuk ke validasi metode, yaitu uji presisi. Ini untuk menunjukkan ketepatan suatu seri pengukuran ulangan dengan konsentrasi yang sama. Untuk metode presisi ini digunakan metode keterulangan sistem dengan membuat 6 larutan dan didapat nilai RSD-nya. Hasil RSD dari data tersebut yaitu 1,583% atau kurang dari 2%, yang mana memenuhi syarat POPP-CPOP tahun 2013. Kemudian uji linearitas ini bertujuan untuk mengukur apakah kurva tersebut memiliki hubungan yang linear atau tidak. Uji ini dilakukan pada 7 seri kurva baku. Hasil dari data ini didapatkan persamaan regresi linear Y sama dengan 0,1137X plus 0,147
dengan nilai koefisien korelasi sebesar 0,9971 yang memenuhi syarat POPP-CPOP, yaitu R lebih dari atau sama dengan 0,99. Kemudian uji selanjutnya yaitu uji LOD dan LOG. Untuk LOD ini digunakan untuk menentukan konsentrasi terkecil dari suatu analit yang dapat terdeteksi. Sedangkan untuk LOG ini untuk menentukan konsentrasi terkecil yang dapat ditentukan dengan presisi dan akurasi yang dapat diterima. Pada penelitian ini dibuat konsentrasinya di bawah konsentrasi kurva baku. Dan diperoleh persamaan regresi linier 0,1335x minus 0,1291. Kemudian dimasukkan ke dalam rumus LOD dan LOG. Untuk LOD-nya didapat 0,115 ppm dan LOG-nya 0,384 ppm. Dan analisis
silakan dipercepat.
Untuk analisis kuantitatifnya, untuk sampel A didapat kadar 1,893 plus minus 0,1064 ppm. Untuk sampel B 2,0084 plus minus 0,089 ppm. Dan sampel D 1,8100 plus minus 0,0113 ppm. Untuk analisis statistiknya ini didapat nilai signifikansi pada uji Kolmogorov yaitu 0,084. Untuk uji homogenitas didapat nilai signifikansi 0,125 dan untuk uji ANOVA didapat nilai signifikansi sebesar 0,001. Untuk kesimpulannya, pada penelitian ini disimpulkan bahwa 3 sampel dari 6 sampel mengandung rodamin B. Kemudian hasil validasi metode ini memenuhi syarat POPP-CPOP tahun 2013 dan kadar rata-rata yang didapat sebagai berikut. Terima kasih, kurang lebihnya mohon maaf. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Baik, pada hadirin yang ingin bertanya, kami persilahkan. Ya, masih kita punya waktu 2 menit lagi. Bisa dibuka mungkin ya kesimpulannya. Berarti sini bisa disimpulkan bahwa ada 3 sampel yang terdeteksi mengandung rodamin ya, Mbak Galuh ya. Silahkan kepada para audiens untuk bertanya.
Pak Riki, ya, masuk ya. Coba Mbak Galuh di judulnya Mbak Galuh.
Iya, Bu.
Ini samplingnya berapa jumlahnya, Mbak? Kembali ke judul.
Mohon maaf Ibu, bisa diulang.
Kembali ke judul.
Iya.
Halaman 1 mungkin ya. Macet Mbak, atau tempat saya ini, saya nggak stabil.
Tempat saya juga masih halaman 1 Ibu.
Oke, oke. Ini analisis rodamin dalam kolang-kaling. Ya, ini di mana Mbak Galuh?
Di alun-alun selatan.
Iya, jadi judulnya kurang begitu spesifik gitu ya. Oke, jadi karena tadi di dalam penelitiannya kan dari sampel berapa? 6? 6 ya. 6 cuma ada 3, tapi kan itu kalau menurut saya kamu tidak menyebutkan judulnya. Di judul itu di mana? Di Yogyakarta kah? Mungkin di Semarang kah? Atau di Eropa kah? Atau di Malaysia gitu kan? Nah, seperti itu. Jadi ini apa ya, masukan saja. Jadi kalau nggak jelas judulnya kan takutnya nanti kok ini ya apa melebar gitu. Ya, kalau di Yogyakarta ya tadi kayak kemarin alun-alun kayak gitu ya. Itu. Oke, jadi kalau tadi sampelnya cuma 6, kamu ngambilnya di mana? Kalau tidak dijelaskan di sini ya nanti nggak nyambung gitu kan. Kamu mau publikasi kalau nggak ada judulnya nggak kurang pas gitu kan, nanti malah ya nanti diperbaiki ya.
Iya, Bu.
Oke, itu aja mungkin Pak Riki masukannya.
Baik, terima kasih.
Iya.
Mungkin satu lagi dari rekan-rekan ya, sebelum kita tutup, karena ini adalah peserta terakhir ya. Tidak ada ya yang belum maju. Ada satu lagi mungkin dari rekan-rekan. Oh ya, saya pengen tanya dulu di sini dari yang pada room 4 ini mahasiswa semua ya? Atau ada yang sudah berprofesi sebagai dosen atau posisinya sedang studi S2 mungkin? Mahasiswa semua ya, berarti ya? Baiklah. Kalau tidak ada pertanyaan lagi, maka kita tutup ya untuk sesi pada room 4. Terima kasih kepada para hadirin yang sudah hadir pada hari ini. Kemudian juga terima kasih kepada reviewer yaitu Profesor Nur Hasanah. Juga pada para peserta. Nah, rekan-rekan peserta, setelah ini silahkan kembali masing-masing ya ke main room, ke room utama, karena nanti sesi selanjutnya akan kita umumkan untuk the best oral presentation di tiap-tiap kategori dan juga untuk the best poster presentation. Saya Muhammad Riki Sindi Praristia, saya mohon maaf kalau misalkan pada penyelenggaraan atau ketika saya menjadi moderator mungkin belum sempurna atau ada suatu kesalahan ya, karena kalau boleh jujur saya dadakan dijadikan moderator ini. Jadi mohon maaf kalau misalkan memang ada yang salah, semoga nanti pada pertemuan berikutnya jika ada kesempatan kita bisa lebih baik lagi. Baik, itu saja mungkin. Saya pamit dulu, kita kembali ke main room. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
The recording has stopped.
Faculty of Pharmacy of Universitas Dahlan was established in 1996 with one study program, which becomes the oldest faculty of pharmacy among Muhammadiyah and Asyiah colleges. Since its establishment, the Faculty of Pharmacy has one study program, namely the Pharmacy Study Program with undergraduate level. In 2001, based on a rector's decree, UAD Faculty of Pharmacy started to open a new study program, namely the Pharmacist Professional Study Program. Along with the education development, in 2015, UAD established a postgraduate program in pharmacy.
Faculty of Pharmacy, Universitas Dahlan is a private Muslim faculty owned by the biggest Muslim organization in the world, namely Muhammadiyah. We already granted A for the National Health Accreditation. Thus, we commit to raise the higher quality in ASEAN level. The integration of moral and intellectual integrity in the higher education system can support the graduates for passing the national examination. The research and publication of the lectures are conducted based on the national and international collaboration. We also serve the community using the pharmaceutical products and our knowledge.
The UAD Pharmacy Study Program commits to the faculty vision of becoming a leading pharmacy.
Keuntungan masing-masing. Nah, belum ada penelitian mengenai perbandingan macam-macam sediaan topikal yang efektif untuk penyembuh luka. Selanjutnya yaitu kerangka konseptual. Dari penjelasan luka di atas, jenis luka menurut Ariningrum 2019 ada luka sayat, luka lecet, luka robek atau parut, luka tusuk, luka gigitan, luka bakar dan luka laserasi. Dari jenis luka-luka tersebut, yaitu perlulah dilakukan penyembuhan luka yang efektif. Penyembuhan luka dapat diberikan obat sediaan oral maupun obat sediaan topikal. Nah, jenis obat sediaan topikal yaitu ada gel, hidrogel, emulgel, salep, krim, patch, cairan antiseptik, serta sabun. Dari jenis sediaan topikal tersebut, perlulah dikaji literatur efektivitas macam-macam sediaan topikal pada proses penyembuhan luka. Nah, dengan rumusan masalah yang pertama yaitu macam-macam bentuk sediaan topikal yang dapat dijadikan penyembuh luka dan basis formulasinya yang dapat dijadikan sebagai pembawa sediaan untuk penyembuh luka yang efektif. Selanjutnya yaitu metode penelitian. Metode yang pertama yang digunakan yaitu ada studi literatur. Sumber literatur ada PubMed, Sun-Direct, Google Scholar. Keywordnya formulasi sediaan topikal untuk penyembuh luka dan topical formulation of wound healing. Selanjutnya yaitu menentukan kriteria inklusi. Kriteria inklusi yang pertama yaitu jurnal tahun publikasi 2010 sampai 2020, bahasanya Indonesia dan Inggris, objeknya sediaan topikal, jenis jurnalnya research artikel original dengan teks lengkap, tema jurnalnya macam-macam sediaan topikal yang digunakan dalam penyembuh luka. Selanjutnya yaitu kriteria eksklusi. Yang pertama tidak dapat diakses secara lengkap atau full text. Yang kedua yaitu format penulisan jurnal tidak sesuai dengan IMRAT, Introduction, Method, Result, and Discussion. Selanjutnya yaitu alur penulisan jurnal. Berdasarkan pencarian pada Sun-Direct itu ada 7.387, PubMed 565, dan Google Scholar ada 1.320 dengan jumlah 9.272. Nah, dari 9.272 tersebut dilakukan kriteria inklusi dengan tahun 10 tahun terakhir yaitu 2010 sampai 2020 didapatkan 8.233 jurnal. Dari 8.233 jurnal tersebut dilakukan screening literatur sejumlah 122 jurnal. Nah, 62 jurnal dieksklusi karena tidak bisa di-download atau diakses dengan full text sehingga didapatkan hanya jurnal 60. Dari 60 jurnal tersebut dilakukan eksklusi karena penulisan format tidak sesuai dengan IMRAT yaitu Introduction, Method, Result, and Discussion, sehingga jurnal akhir yang dapat dianalisa sesuai target, tujuan, dan pemecahan masalah yaitu 31 jurnal. Selanjutnya yaitu hasil penelitian. Nah, pada hasil di atas itu dapat dilihat bahwa bentuk sediaan gel banyak digunakan, yaitu terdapat 6 penelitian di mana satu jurnal itu sediaan spray gel. Nah, kategori 6 jurnal tersebut itu diakumulasikan seluruhnya menggunakan basis formulasi HPMC, karbopol, dan CMCNA. Tetapi basis yang paling sering digunakan yaitu ada karbopol. Nah, selanjutnya yaitu sediaan hidrogel. Pada sediaan hidrogel itu banyak digunakan pada penyembuh luka, yaitu terdapat 6 penelitian. Kategori 6 penelitian ini itu menggunakan basis formulasi karbopol, CMCNA, dan kombinasi karbopol serta citosan. Akan tetapi basis formulasi yang paling banyak digunakan yaitu menggunakan karbopol. Selanjutnya yaitu ada tabel M.Biotech, di mana tabel ini didapatkan obat M.Biotech untuk penyembuh luka itu 2 jurnal penelitian. Hal ini membuktikan bahwa tidak banyak jurnal penelitian yang menggunakan sediaan M.Biotech dalam obat penyembuh luka. Nah, pembuatan sediaan M.Biotech ini menggunakan basis formulasi HPMC dan karbopol. Bahan aktif yang digunakan yaitu ada Osimum basilicum dan minyak ikan gabus, di mana kedua bahan tersebut itu memiliki zat aktif yang berupa minyak. Selanjutnya yaitu tabel bentuk sediaan salep. Nah, pada tabel ini dilihat bahwa bentuk sediaan salep banyak digunakan dalam penyembuh luka, yaitu terdapat 11 penelitian. Basis formulasi yang digunakan pada penelitian tersebut yaitu ada adapselame, parafin padat, parafin lunak, parafin putih, seraflava, vaselin abu, dan vaselin kuning. Tetapi basis salep yang paling banyak digunakan yaitu ada vaselin, adapselame, dan parafin. Nah, penelitian yang menggunakan basis vaselin itu ada 6 jurnal, yang menggunakan basis adapselame ada 6 jurnal, sedangkan yang menggunakan basis parafin ada 4 jurnal penelitian. Nah, terdapat 2 jurnal yang menggunakan basis eucerin. Selanjutnya yaitu ada tabel sediaan krim. Nah, untuk tabel sediaan krim ini itu dapat dilihat bahwa sediaan krim itu memiliki 3 penelitian. Nah, hal ini membuktikan bahwa tidak banyak jurnal penelitian yang menggunakan sediaan krim dalam obat penyembuh luka. Penelitian tersebut menggunakan basis formulasi asam stearat, PEA, parafinilik, adapselane, span, dan twin. Terdapat 1 jurnal penelitian yang menggunakan kombinasi span dan twin, serta terdapat 2 jurnal penelitian yang menggunakan kombinasi basis asam stearat dan TEA. Selanjutnya yaitu pada tabel plaster dapat dilihat bahwa bentuk sediaan plaster ini memiliki 3 jurnal penelitian. Hal ini membuktikan bahwa sediaan plaster ini sedikit digunakan dalam obat penyembuh luka karena termasuk inovasi yang baru. Penelitian tersebut menggunakan basis formulasi citosan, HPMC, dan PVA. Selanjutnya yaitu ada pembahasan. Pembahasan yang pertama yaitu ada gel, di mana berdasarkan hasil diperoleh informasi bahwa jenis sediaan topikal yang banyak digunakan dalam penelitian sebagai penyembuh luka yaitu ada sediaan gel dengan jumlah 6 penelitian. Nah, sediaan ini merupakan sediaan semi padat yang mengandung zat aktif yang terdiri dari suspensi yang dibuat dari partikel anorganik kecil atau anorganik besar yang terpenetrasi oleh suatu cairan. Pemilihan sediaan gel dalam penyembuh luka ini adalah sediaan ini terdiri dari komposisi air yang banyak dan dapat melembabkan kulit karena kelembapan kulit itu berpengaruh pada proses penyembuhan luka. Faktor penyembuhan luka akan mempengaruhi proses angiogenesis. Nah, proses ini itu akan terbentuk pembuluh darah baru sebagai tempat untuk menyuplai nutrisi dan oksigen pada luka. Hal ini sesuai dengan keuntungan sediaan gel yang memberikan sensasi dingin ketika diaplikasikan, mudah diaplikasikan pada kulit, memiliki kemampuan merata dan menyebar yang baik pada kulit, serta melembabkan kulit. Nah, dalam tabel hasil basis formulasi gel yang digunakan yaitu ada karbopol, CMCNA, dan HPMC. Hal ini sesuai dalam teori Rowe bahwa remaining agent yang memiliki 4,6 pada hasil penelitian dilakukan MAPA, konsentrasi CNA-nya itu 5%. Hal ini digunakan terdapat 6 jurnal penelitian. Sediaan hidrogel ini terdiri dari 80-95% air dan gelling agent yang dapat membengkak dan mempertahankan sebagian besar air dalam strukturnya. Nah, hidrogel ini sering digunakan dalam obat penyembuh luka dengan alasan sediaan ini itu sangat baik digunakan sebagai sediaan penutup luka karena berperan dalam membantu menghilangkan jaringan yang mati. Hidrogel juga mampu memberikan kelembapan pada area yang terkena luka sehingga memberikan rasa dingin yang dapat mengurangi edema pada luka sehingga meningkatkan efektivitas proses penyembuhan luka. Basis formulasi yang digunakan yaitu ada karbopol, CMCNA, PVA, dan kombinasi ketosan plus karbopol. Tetapi paling banyak digunakan yaitu karbopol. Nah, hidrogel dengan basis PVA akan terbentuk gel dengan konsentrasi air yang tinggi dan terdapat film tipis yang memiliki kelupas sehingga sesuai dengan penyembuhan luka. Selanjutnya yaitu ada M.Biotech. Pada tabel hasil M.Biotech diperoleh informasi bahwa jenis sediaan topikal M.Biotech itu hanya sedikit digunakan dalam penyembuh luka. Hanya didapatkan 2 juta penelitian. Sediaan ini terbentuk dari emulsi yang diberi jaring agent. Pemilihan sediaan M.Biotech pada hasil tabel itu karena bahan aktif yang digunakan berupa minyak sehingga dipilih sediaan M.Biotech. Nah, berupa minyaknya yaitu pada Oxalium basilicum terhadap minyak atsiri dan minyak ikan gabus. Nah, basis formulasi yang digunakan pembuatan sediaan M.Biotech yaitu ada karbopol dan HPMC. HPMC itu digunakan dalam pembuatan M.Biotech pada jurnal peneliti ahli menggunakan basis karbopol dengan konsentrasi 0,25 sampai 0,75%. Hal ini sesuai dengan teori Rowe bahwa rentang konsentrasi karbopol yang digunakan yaitu 0,5 sampai 2%. Pada penelitian yang dilakukan oleh Novrianti menggunakan basis HPMC sebesar 3%. Selanjutnya yaitu ada salep. Berdasarkan hasil tabel diperoleh informasi bahwa jenis sediaan topikal yang banyak digunakan dalam penyembuh luka yaitu salep. Pembuatan sediaan salep.
Ini langsung saya ke kesimpulan ya.
Ya.
Ini yaitu ada kesimpulan. Dari penjelasan pembahasan di atas dapat ditarik kesimpulan yang pertama yaitu jenis sediaan topikal yang digunakan dalam obat penyembuh luka yaitu ada gel, hidrogel, emulgel, krim, salep, dan plester. Selanjutnya basis formulasinya yang digunakan pada sediaan penyembuh luka adalah karbopol, CMCNA, HPMC, PVA itu digunakan dalam basis sediaan gel. Basis sediaan salep yang digunakan yaitu ada eucerin, adiflanil, vaselin album, parafin cair, dan parafin padat. Pada sediaan krim yaitu ada spantun, asam stearat, dan TEA. Pada sediaan plester yaitu ada citosan dan PVA.
Ya, terima kasih. Ya.
Terima kasih Bapak atas pemaparan dari video PPT saya. Kurang lebihnya mohon maaf.
Ya.
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Ya, waktu sudah 10 menit. Silakan kalau memang ada yang ingin ditanyakan bisa disampaikan. Oke. Kita cukupkan dulu untuk presentasi dari Alfia. Terima kasih, Saudari Alfia. Selanjutnya ada atas nama Galuh Trinidad Syah. Mohon maaf, ini ibu atau mahasiswa? Galuh Trinidad Syah. Apakah ada orangnya? Oke, yang persentase kelima tadi atas nama Vicky Malinda, saya panggil ulang. Apakah sudah bergabung?
Cek apakah suara saya terdengar?
Oh baik, ya, ya terdengar. Mohon maaf, ini ibu Galuh ya, betul ya?
Mahasiswa Pak saya.
Oh baik, mahasiswa. Persentase melalui video atau PowerPoint?
Pakai PowerPoint saja. Yeah.
Ya, PowerPoint. Silakan Saudari Galuh Syah.
Ya, izin share screen.
Ya.
Baik, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, saya Galuh Trinidad Syah dari Universitas Ahmad Dahlan. Akan mempresentasikan review kami dengan judul Sistem Penghantaran Obat Tertarget Berbasis Nanopartikel pada terapi kanker payudara. Salah satu jenis kanker yang paling sering menyerang wanita ialah kanker payudara. Pertumbuhan sel yang abnormal dan tidak terkendali menjadi pemicu terjadinya kanker payudara. Pengobatan konvensional menggunakan obat-obatan kemoterapi dapat menimbulkan terjadinya efek samping dan toksisitas yang tinggi. Oleh karena itu, dikembangkan suatu pengobatan tertarget, non-toksik, aman, dan memiliki efek terapi yang maksimal, yaitu dengan nanopartikel. Nanopartikel terdapat lapisan hidrofilik, lipofilik, dan amfifilik. Metode yang digunakan dalam review ini ialah review dengan penelusuran database dari Google Scholar, PubMed, Web of Science, dan Scopus, di mana sumber pustaka yang digunakan adalah untuk review dan riset orisinil yang terbit di tahun 2019 hingga 2021. Pengembangan terapi dengan nanopartikel memiliki berbagai keuntungan, di antaranya dapat memperbaiki profil farmakokinetik dengan peningkatan distribusi dan perbaikan bioavailabilitas obat. Ukurannya yang kecil, yaitu 1 hingga 100 nanometer, dan partikel yang fotoluminescent dapat mendukung pemuatan obat menuju target terapi. Jenis-jenis nanopartikel yang telah dikembangkan dalam terapi kanker payudara terdapat nanopartikel organik dan anorganik. Berikut nanopartikel anorganik liposom. Pada nanopartikel liposom terdapat kolesterol, di mana mekanisme aksi dari liposom akan berikatan dengan sel kanker melewati permukaan sel, sehingga akan melalui ikatan permukaan sel yang hidrofobik dan hidrofilik. Dengan demikian, obat dapat dihantarkan secara tertarget ke dalam sel kanker. Kelebihan dari liposom yaitu stabil, non-imunogenik, tidak beracun, biokompatibel, dan biodegradable. Kemudian, nanopartikel dendrimer dengan kombinasi obat, di mana enkapsulasi dapat dilakukan dengan sistem targeting secara pasif, sedangkan kompleksasi dapat dilakukan sistem targeting secara aktif. Pada nanopartikel misel terdapat lapisan amfifilik polimer yang dikombinasikan dengan obat hidrofobik, yang mana mekanisme aksi dari misel adalah permukaan sel kanker yang mengandung permukaan ion negatif dan permukaan misel yang mengandung ion positif akan membentuk ikatan dan dapat menghantarkan obat secara tertarget ke dalam sel kanker. Berikutnya, nanopartikel organik, contohnya ialah nanogold. Nanogold terdiri atas gold dan ikatan pitosan yang dikombinasikan sehingga akan berpengaruh ke dalam sel kanker oleh adanya radiasi. Atom gold akan menembus pembuluh darah. Dengan demikian, penghantaran tertarget dapat terjadi ke dalam sel kanker. Pembuatan dari karbon nanotube terdiri dari beberapa material seperti nanografit dan nanoblack material, di mana mekanisme aksinya dapat melewati kompartemen seluler dan dapat terlokalisasi ke dalam sel kanker. Nanopartikel iron oxide terdiri atas inti oksida besi dan lapisan polimer, di mana iron oxide dapat dimodifikasi dengan lipid untuk menghantarkan obat secara tertarget ke dalam sel kanker.
Kemudian anorganik nanopartikel lain yaitu mesoporous silica nanopartikel atau biasa disingkat dengan MSN. MSN ini efektif sebagai pembawa anti kanker dengan penghantaran secara endositosis. Efikasi dan keamanan nanopartikel ditujukan oleh kemampuannya dalam menghantarkan obat secara tertarget ke dalam sel kanker, sehingga dapat mengurangi degradasi obat, memudahkan penyerapan obat secara selektif ke dalam situs target, dan dapat menurunkan toksisitas daripada formulasi konvensional. Selain itu, profil farmakokinetik dan eflux intraseluler dalam sel kanker akan meningkat. Kesimpulannya, sistem penghantaran nanopartikel akan meningkatkan akumulasi obat dalam sel kanker payudara akibat peningkatan permeabilitas yang memungkinkan terjadinya ekstravasasi nanopartikel dari jaringan darah ke sel payudara. Nanopartikel dengan ukuran kecil, apabila terjadi degradasi obat, maka risikonya kecil terjadi toksisitas, dan nanopartikel dengan ukuran 10 hingga 100 nanometer tidak difiltrasi ginjal sehingga akan terakumulasi ke dalam sel kanker. Sekian presentasi yang dapat disampaikan. Kurang lebihnya mohon maaf. Terima kasih. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Baik, luar biasa, cukup menarik terkait nanopartikel pada kanker payudara. Silakan bagi yang ada ingin didiskusikan, kami persilahkan. Bapak, Ibu, dan mahasiswa sekalian, apakah ada yang ingin didiskusikan? Masih ada waktu. Bapak Zainal, Ibu Uce, Ibu Iis, silakan. Jadi mahasiswa sekalian. Baik. Kalau tidak ada, saya cukupkan ya. Terima kasih kepada presenter terakhir kita, Saudari Galuh Syah. Semoga ilmunya bisa bermanfaat ya. Baik, dengan berakhirnya presentasi dari Saudari Galuh Syah, maka berakhirlah sesi oral presentation hari ini.
Ya, sebentar Pak Silvian. Berarti ada dua peserta yang tidak hadir, nggih? Betul nggak?
Satu, Ibu.
Yang
nama
PT 7 yang Nurul Fadil itu nggak ada, memang nggak ada ya?
Mohon maaf, Nurul Fadil ya, Muhammad Nurul Fadil.
PT 7.
PT 7.
Tapi yang dihadir memang nggak ada sih PT 7, sama yang Vicky Malinda ya. Coba kita cek terakhir.
Iya. Ya mungkin panggilan terakhir ya kepada Saudari Vicky Malinda dan Saudara Muhammad Nurul Fadil.
Iya. Nampaknya nggak ada. Oke. Di peserta nggak ada sih.
Baik.
Iya. Oke,
Begitu,
Pak.
Iya. Terima kasih sekali lagi kepada Ibu Iis Wahyuningsi selaku reviewer, dan terima kasih kepada presenter yang telah memaparkan hasil presentasi hasil penelitian yang sudah dilakukan. Semoga ilmu yang telah dibagikan bisa bermanfaat bagi kita semua. Oke, untuk selanjutnya adalah saya serahkan pada ruang utama. Nanti best presenter akan diumumkan di ruang utama. Untuk sesi kali ini, saya selaku moderator memohon maaf yang sebesar-besarnya atas ketidaknyamanannya. Apabila saya memiliki banyak kesalahan, mohon dimaafkan. Dan saya cukupkan dengan mengucapkan terima kasih. Wabillahi taufiq wal hidayah. Assalamualaikum warahmatullahi wabarakatuh. Selamat siang. Untuk selanjutnya saya serahkan ke panitia nggih.
Pak Silvian tadi udah balik ya?
This meeting is being recorded.
Ladies and gentlemen, all participants, we hope that all the participants will be able to return to main room. Check, 1, 2, 3. We hope that all participants will be able to return to the main room because we will step on into the next session shortly. Kepada para partisipan diharapkan untuk bisa kembali ke main room karena sebentar lagi ya kita menunggu sedikit lagi. Lalu selanjutnya kita akan masuk pada sesi berikutnya. Okay, ladies and gentlemen, I'm already have the result. So finally, let's keep on the next agenda, announcement of the best oral presenter and the best poster presenter. Hasil sudah saya terima, Bapak-Bapak dan Ibu-Ibu, maka kita masuk pada sesi selanjutnya dan the best oral presenter. Patient to Mulyani Rizka Arjuna. Congratulations to Mulyani Rizka Arjuna for the best presentator in room 1. Okay, congratulations. And then in room 2 for oral presentation is Yulia Indah Widianti. Congratulations to Yulia Indah Widianti. And for room 3, for the best oral presentation is Rizky Rahmadi. Congratulations to Rizky Rahmadi. And for the best oral presentation in room 4 is Alfi Syahriatun Nida Meli. Congratulations to Alfi Syahriatun Nida Meli. And for the best poster presentation in room 1B is Ade Kristi Sugianto. Congratulations to Ade Kristi Sugianto. For the best poster presentation in room 2B is Liza Wardani. Congratulations to Liza Wardani. For the best poster presentation in room 3B is Arista Astiwi Maulani. Congratulations to Astiwi Maulani. Congratulations. For best poster presentation in room 2 is I think that's it. Congratulations to the winners. You can see the certificate displayed on the screen. So once again, congratulations. Finally, we have stepped on into the last session, which is closing and the post-test. And finally, from deepest of my heart, I do apologize for my mistakes in presenting in this event. Thanks to the organizing committee to invite me and give me amazing experience. And thank you very much for your kind attention. See you later. Wassalamualaikum warahmatullahi wabarakatuh. And I want to remind you, ladies and gentlemen, that post-test will be closed at 8 p.m. on West Indonesian time, or GMT +7. Ladies and gentlemen, let me remind you that post-test will be closed at 8 p.m. West Indonesian time. Thank you.
The Faculty of Pharmacy of Universitas Dahlan was established in 1996 with one study program, which becomes the oldest faculty of pharmacy among Muhammadiyah and Ashiah colleges. Since its establishment, the Faculty of Pharmacy has one study program, namely the Pharmacy Study Program with undergraduate level. In 2001, based on a rector's decree, UAD Faculty of Pharmacy started to open a new study program, namely the Pharmacist Professional Study Program. Along with the education development, in 2015, UAD established a postgraduate program in pharmacy.
Faculty of Pharmacy, Universitas Ahmad Dahlan is a private Muslim faculty owned by the biggest Muslim organization in the world, namely Muhammadiyah. We already granted A for the National Health Accreditation. Thus, we commit to raise the higher quality in ASEAN level. The integration of moral and intellectual integrity in the higher education system can support the graduates for passing the national examination. The research and publication of the lectures are conducted based on the national and international collaboration. We also serve the community.
More than 1,000 students from various provinces in Indonesia study together in the UAD Pharmacy Study Program. UAD Pharmacy Study Program applied curriculum for 4 years of study. It has represented all aspects of the competencies achievement for graduates. UAD Pharmacy Study Program has about 50 teaching staffs with doctor and professor qualification. There are 30 education staffs with appropriate competencies to strengthen the learning, research process, and community service. The institution evaluated their staff annually to improve professional development, monitoring, and evaluation. UAD Pharmacy Study Program built a strong partnership with various parties by involving alumni and stakeholders for the development of higher education. To support the activities of the three Dharma perguruan tinggi, namely learning, research, and community service, UAD Pharmacy Study Program has good, comfortable, and integrated facilities. The UAD Pharmacy Study Program has a laboratory with well-standardized equipment for teaching and learning process. UAD Pharmacy Study Program attempts to improve cooperation network with various parties, including the government, pharmaceutical industry, local higher education, and abroad.
The students' and lecturers' achievements are already reached the national and international level as well. And finally, join with us, get the higher quality, and reach the better moral and intellectual integrity.
With competence that supports various abilities, both theories and soft skills, graduates of UAD Pharmacy Study Program have spread in various areas including in hospitals, government institutions, academics, pharmaceutical industry, pharmacists, and entrepreneurs.